तुझ्याला सी टाईपचं आहे का?
हां USB वाला आहे का बघ?
एक मिनिट हा USB वालाच हाच
HDMI to USB पण आहे का? लॅपटॉपचं आहे का? हां
त्याला लावून परत फक्त व्हिडिओसाठी सगळं काढायला पाहिजे.
एकच कुठला तरी लॅपटॉप ते USB to HDMI कनेक्टर असतं ते आहे का? आता सी टाईप आहे ना तसं USB वाला असतं.
क्या चाहिए?
अरे वो
व्हिडिओ
कनेक्ट नाही
कनेक्ट
USB वाला आहे का
आपल्याकडे HDMI आहे का?
हां USB to सेकंड स्क्रीन केलं ना तर
हां नाही येस नहीं मेरे ऊपर ये जिम्मेदारी नहीं लेते.
नहीं उसके सारे दिन में इंट्रोडक्शन
किराने बैठे हेलो येतोय ना? डिले हेलो
काही औषध कुठलं वाढवायचं आहे का? सगळं लिहिलं रोज
they will come from
आम्ही सीआयची कामं एवढी केलीत ना
चालू आहे सर.
बंद कर, बंद करून ठेव.
जरा टेस्टिंग.
ठीक आहे. 1 मिनिट चालू आहे. अभी बंद हो गया सर. 1 मिनिट चालू आहे सर. बंद.
हॅलो हॅलो. हेलो 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 माइक टेस्ट. हेलो. अभी आ रहा है. हेलो 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 माइक टेस्ट. हेलो माइक टेस्ट 1 2 3. हेलो हेलो. ठीक है क्या.
Education is a fundamental human right that can change the trajectory of one's life. However, for many bright and self-motivated students from rural areas, financial conditions can come in the way of realizing their dreams. Recognizing this issue, in 1956, the eminent social worker Dr. Achyut Rao Apte, along with like-minded individuals, established Vidyarthi Sahayak Samiti. The aim was to provide subsidized but high-quality accommodation, nutritious food and self-development opportunities to needy students coming to Pune for college education from rural areas. Emphasis on self-development activities is what sets Samiti apart from other hostels. These initiatives are organized under Vidyarthi Vikas from rural areas. Emphasis on self-development activities is what sets Samiti apart from other hostels. These initiatives are organized under Vidyarthi Vikas Kendra and include that can change the trajectory of one's life. However, for many bright and self-motivated students from rural areas, financial conditions
So a very, very good morning to each of you, and welcome to NCL for this one-day symposium. And it's being organized by the Organic Chemistry Division at CSIR-NCL, together with the Professor K.N. Ganesh Endowment Trust, and with a lot of help from our friends at ISER Pune as well. It is a part of the CSIR 1 week, 1 lab program. This program was conceptualized by the Honorable Minister of Science and Technology, Dr. Jitendra Singh Ji, to showcase and propagate the diverse work and contributions of the 37 CSIR labs that are spread across the country. And this is a nationwide campaign. At NCL, it's being held during this week of 22nd to the 27th of May, when the activities, contributions, the technological breakthroughs, and innovations are being highlighted in the form of several programs and outreach programs, symposia, exhibitions, including symposia such as this one. I would now like our dignitaries for today I invite them to kindly come up on the dais. Dr. Lele, our director, Professor Ganesh, our guest of honor for today, Professor Bhagwat, Professor Dr. Shivram, Dr. Nathu, please. Okay. Thank you. So I request Dr. Ramana to kindly welcome our director by handing over a potted plant. And then and now I request our director, sir, to please welcome our guests for today, Professor Ganesh. Dr. Shivram. Professor Bhagwat. Dr. Nathu. Dr. Lele, I request you to please, I will call upon you to kindly give the opening remarks.
Namaskar and a very, very good morning to all of you. I welcome you all today formally to today's one-day symposium on advances in DNA RNA Therapeutics, which is organized here in NCL, as Monisha said, by our Organic Chemistry Division and the Professor K.N. Ganesh Endowment Trust, with a lot of help from ISER Pune. Today's symposium, as you will read, is focused on the recent developments in the field of oligonucleotides, drugs, and nucleic acids, particularly from a chemist's perspective. This special symposium is also organized to honor and felicitate our own Professor K.N. Ganesh, a great scientist who has epitomized excellence in path-breaking research, a visionary institution builder who exemplifies every aspect of leadership, and a thorough gentleman who personifies generosity, empathy, integrity, equity, and all of those values. We in NCL are extremely proud that Professor K.N. Ganesh, who was part of us for about 7 years, from 1987 to 1994, has been conferred the Padma Shri in Science and Technology by the Government of India. Please join me in this great applause. It is also wonderful that Professor K.N. Ganesh celebrated his 70th birthday just yesterday. And so we are, so very, very happy birthday to you, Professor K.N.G., and many, many, many happy returns of the day. During Professor K.N.G.'s tenure at NCL, he undertook absolutely top-class research on chemistry and biology of nucleic acids, therapeutic and diagnostic applications of DNA analogs, structural biology of collagen peptides, DNA nanotechnology, and PNA analogs. All of these at that time were so much more, futuristic. And today, especially nucleic acids and oligonucleotides is one of the hottest growing areas in the industrial biotechnology, not just in India, but across the world. And I think this, I believe truly that this is the hallmark of NCL. We at NCL take immense pride in saying that time and again, our scientists and visionary leaders have managed to foresee the future well ahead of time, undertake top-class research in those areas, and make every conscious effort to translate that science into technologies just in time to make them market ready. So thanks to you, Professor K.N. Ganesh, we today at NCL have at least a platform on which we can deliver what the industry expects from us. And indeed, we today in NCL are displaying in a way this confluence and synergy of what Dr. Mashelkar said beautifully, the excellence and relevance, the confluence and synergy of excellence and relevance. And this is done as part of our 1 week, 1 lab campaign. As Monisha told you, we have a fairly large exhibition organized in the main building. In the foyer, you will see a display of our relevance of what we are doing. We have 7 themes in our roadmap, and each theme is displayed in the form of a stall where some of our recent technological innovations and breakthroughs that we have achieved are displayed there, and you get a feel of what it is that we are doing for our stakeholders, whether it is industries, startups, farmers, and so on and so forth. On the back side, in front of the library, you will see a display of some of our competencies in the form of our 6 divisions and our about 10 support functions. All of them, their recent activities are displayed there. So I request all of you to spend some time and take about half an hour to go through that small exhibition that we have. It's the first time we are doing this at this scale. We've had almost 1,500 people, perhaps more. I have lost count of how many students have been coming on a daily basis. People tell me at least 250 to 300 students, just students, and so many others who have been coming and sort of witnessing what we have done, and I'm getting really great reviews of what they see. So it gives us a lot of confidence that we are doing the right things, and we are also bringing all these stakeholders to our laboratory to showcase what we are doing. So please do, I appeal to you to take some time out of this wonderful symposium, sometime maybe just after lunch or something like that, to go around in the main building and see what it is that we are doing. So once again, I welcome all of you to this one-day symposium and to the 1 week, 1 lab exhibition at NCL today. I'm really looking forward to attending all the lectures, although I might miss some because there are parallel events going on as part of the OWOL. But I do want to attend as many of these as possible. So once again, thank you for coming. Congratulations, Professor K. N. Ganesh, and happy birthday to you.
Thank you, Dr. Lele. Yes, we have included a small time slot for people to visit the exhibition as well, and we can also do it during the lunch break because we have a fairly long time then. So I request now the chair of our Organic Chemistry Division, Dr. Ramana, to please say something about our division.
Yeah, distinguished guests on the dais and guests, students and friends. Good morning to all of you. Maybe first I would like to say something about the Organic Chemistry Division. I'm very happy to see the two HODs of the former Organic Chemistry Technology and Organic Chemistry Synthesis, which was merged maybe after Professor Ganesh left to ISR Pune and become one division Organic Chemistry. So, first I would like to talk something about Professor Ganesh. Actually, my association with Ganesh goes back almost 22 years. In the same month, when I joined actually NCL as a new entrant, I have to meet Professor Ganesh. Then actually we had almost more than an hour discussion. Basically, it was revealing actually what I did during my postdoc and what I did during my PhD. And at the end of the day, what advice he gave me is, you work for the properties. Anyway, to some extent, I can do it, but most of my time has been gone actually in chasing the molecules. Then actually, I have the opportunity of working with Professor Ganesh on several occasions. Either it was designing the coursework for organic chemistry. Indeed, actually, Organic Chemistry Division is one of the first divisions to start coursework in NCL way back in 2004. So initially what we did is we called all the professors from outside the institutes for giving the lectures or the classes. And at the end of the session, actually, we didn't do any exam. It was an interview. It's not interview, but it's a oral examination like. So Professor K. N. Ganesh, he sat 2 days along with us. I don't know whether Hota is here. Myself, Professor Ganesh, then Hota and Seshizar. We four sat 2 days, and he was all the time asking the questions. But the beauty of him is the magnitude of the question, it depends upon the caliber of the student. At the end of the session, the student feels that actually he did answer all the questions very well, and he is very happy. Anyway, maybe last year, I met Professor Ganesh at ISR Tirupati. So we sat almost for more than an hour. And I was amazed to see his passion and perfection in attending the minute things, either it was the design of a building or even the aesthetics of a road. It was indeed a great lesson for us, all of us. With my experience, what I could say is Professor KNG is not a godfather. He won't be a godfather for anybody. He'll be like a great grandfather who narrates his experience as a story and whose passion is contagious. And his pursuit of perfection inspires those around him to strive for the greatness. Once again, on behalf of NCL in general and on behalf of the organic chemistry in particular, we congratulate Professor Ganesh and express our deepest appreciation for his exceptional contributions. Professor Ganesh, may your journey continue to inspire generations to come, and finally, many, many happy returns of the day. Thank you.
Thank you, Dr. Ramana. In continuing in the celebration mood, I understand that Professor Bhagwat would like to say a few words and felicitate Professor Ganesh. Please, sir.
Professor Ganesh, other dignitaries on the dais, and various guests, ladies and gentlemen. I am perhaps one of the newest person between these two campuses to be here. And I got that privilege to be here on a beautiful campus thanks to Dr. Ganesh. So it's a very special feeling for me that I get to greet you on this occasion. An institution where it started from being almost a barren land, forest land, to a beautiful institute that it stands today is substantially because of your contributions to it. So we are grateful to you for that. And so this is from ISER as an institute and specifically the chemistry department there.
Thank you, Professor Bhagwat. Moving on to with the program of today, I now request Dr. Nathu, who's president of the Professor K.N. Ganesh Endowment Trust, to tell us a little more about the trust and the award.
Hello, good morning, dignitaries on the dais and in the auditorium. It gives me a great pleasure to introduce the trust that has been founded 10 years back when, under the title Professor K.N. Ganesh Endowment Trust. So on behalf of the trust, I'd like to welcome you all. And just in 2 or 3 minutes, I'll just give the introduction what we are doing. Next. Keep on. Yeah. So this was basically started 10 years back when Ganesh became 60. From his figure and all other things, it is very difficult to judge his age, that is for sure. So, but he became 60 at that time, and then there was a gathering of all the students, collaborators, researchers, and connected people. And they thought in just celebrating and having a liquid and solid diet in the night, let us not take it on. So we thought that we'll be able to do something concrete. And with the idea then was floated that the mission, all the students collaborated, they came together and established the K.N. Ganesh Endowment Trust. Main mission, there will be a lot of visions and missions, so I don't want to, but there were 2 things. One is to establish the endowment lectures. I'll just go through endowment lectures to encourage this one. And second is to help the underprivileged children in their education, which have been completely deprived of. So generally, next. So these are some of this one. Most important thing is to foster excellence and creativity and provide recognition of significant contribution to individual groups and organizations. To organize endowment lectures in the cutting edge of research areas, and to support the young researchers in shaping their careers through seminars and personal counseling. So when we started, we thought let us start with the endowment lectures, and one for the senior gentleman and another for the upcoming researcher. And this we have been continuing now. Next. Next. Okay. These are some of the eminent personalities or the scientists who has given lectures earlier. One was Professor Deepankar Chatterjee from ISC Bangalore, Dr. Rajesh Gokhale from Secretary of Biotechnology, Shekhar Mande, former DG CSIR, Nicola Nifintinev, he is a professor from Russian Academy of Science, and of late Dr. Mukesh, from Professor of Indian Institute of Science, Bangalore. Next. We were thinking of young aspiring researchers, so we invited Dr. Yamuna Krishnan. At that time, we thought that she is aspiring, but now she has grown into matured now and full professor. And Dr. Savik Maiti from CSIR. Next. This year, it gives me a pleasure to announce this year's 2 awards. The senior award goes to Dr. Manoharan. He is a senior vice president and a distinguished research scientist at LIM Pharmaceuticals. And as my young aspiring researchers award goes to Dr. Jyotirmoy Das of IIS Calcutta. There is one common thread between the two, that is the application of nucleic acids. Whereas Professor Manoharan has introduced several new drugs into the market which are based on the RNA therapeutics, Jyotirmoy Das also is trying to use them for medicinal and pharmaceutical research. So there is a common thread between the two. This photo I am just showing you how we were 10 years back when we started our activities. Next. We were not at loggerheads, but just started the activity in a very positive way. So I thought I'll just share this picture. Next. 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 These were some of the founder members, you can say, we gathered together and then started the activity. Next. Next. Yeah, this is also one of the group photos that we were together. Next. And thus, I would like to thank you, all of you, for attending this and respecting to the invitation of the Professor K.N. Ganesh Endowment Trust. I hope we'll continue to do it. If there are any suggestions in terms of activities or anything, you just let me know. Thank you very much.
So now we will have the presentation of the award. So yes, as Dr. Nathu announced, the Professor K.N. Ganesh Endowment Award goes to Dr. Mano Manoharan. I request you to kindly come up, and I request, yes. Wherever. Maybe, no, we will first felicite. Dr. Manoharan is a senior vice president and distinguished research scientist at RNA Pharmaceuticals, Cambridge, Massachusetts in the US. In 2003, he was the first chemist hired at RNA. He and his team pioneered the discovery and development of the chemical modifications that make RNA interference-based human therapeutics possible. His work and his team's work led to Onpattro, that is patisiran, which is the first RNAi therapeutic that was approved by FDA in 2018. He has a distinguished career as a world-renowned chemist in the area in the areas of oligonucleotide chemical modifications, conjugation chemistry, delivery platforms, especially lipid nanoparticles, etc. And he's very well known for the GalNAc conjugates that are widely acknowledged. We are very happy that he has consented to accept this award and welcome him to NCL, which he has traveled here against all odds to be with us today. Thank you, sir. And congratulations. Thank you. I request, the Junior Professor K.N. Ganesh Endowment Award this time is Professor Jyotirmayi Dash from IACS Kolkata. She's currently Senior Professor at the School of Chemical Sciences, IACS, and her research interests include developing new strategies for targeting nucleic acids, designing artificial ion channels, and developing new synthetic methods for therapeutic and biomedical applications. Her group developed biomolecular probes to selectively target DNA secondary structures, nucleic acid template-directed synthesis of selective ligands, DNA quadruplexes, I motifs, and HIV-IR RNA. Professor Dash, thank you for accepting this award from the Trust, and congratulations to you. Thank you all. I request the dignitaries to kindly occupy their seats in the hall.
We start with the session.
No, now we will have the social organization.
Okay. Please come.
Yes, please.
Yeah.
Please. Thank you very much. As mentioned by Dr. Nathu, another important aspect of the Professor K.N. Ganesh Endowment Trust is the contribution towards social organizations, towards the development and education of underprivileged and specially abled children. This year, 2 social organizations have been chosen by us. The Vidyarthi Sahayak Samiti and Maitri. We will play a short clip for each of them, and then we will call upon their representatives to kindly come up on stage to collect the Vidyarthis. Yeah. Can you play the... Thank you. I now call upon the representative from Maitri, Mrs. Jayashree Shidore, who is the founder and coordinator of Maitri, to kindly come up on stage and receive the contribution from us. The next is the Vidyarthi Sahayak Samiti.
Education is a fundamental human right that can change the trajectory of one's life. However, For many bright and self-motivated students from rural areas, financial conditions can come in the way of realizing their dreams. Recognizing this issue, in 1956, the eminent social worker Dr. Achyut Rao Apte, along with like-minded individuals, established Vidyarthi Sahayak Samiti. The aim was to provide subsidized but high-quality accommodation, nutritious food and self-development opportunities to needy students coming to Pune for college education from rural areas. Emphasis on self-development activities is what sets Samiti apart from other hostels. These initiatives are organized under Vidyarthi Vikas Kendra and include yoga training, mandatory volunteering at Samiti, mentorship by experts, English speaking, interviewing skills, public speaking, earn and learn scheme, and entrepreneurship development programs. Through these programs, cleanliness, equality, dignity of labor, and self-reliance are the values imbibed in students at Samiti. Samiti's journey that began with 5 students now provides hostel facilities to 450 boys and 350 girls. Samiti has helped transform the lives of thousands of students. Many of these students are contributing to nation building through leadership in their chosen fields. The Samiti bond is so strong that many ex-students are drawn back to Samiti to contribute as volunteers, trustees, and management. Demand for hostel accommodation at Samiti is so high that today it can only admit 1 in 5 deserving applicants. Hence, Samiti has embarked on an expansion program to increase the hostel capacity by 500. Given the acute need, over 2/3 of new capacity addition is for girls. We invite you to join the mission. Your support as a karyakarta, a mentor, or a financial contributor can make a world of difference to the future of these students. Join us on this journey of growth, hope, and possibility. Let us help change lives, one student at a time.
Thank you, Dr. Singh. Yes. I request the representative to kindly come up on stage. Mr. Tushar Ranjankar was,
Okay. Since there
as he's not here, we will move on. So now we will take a short break for tea, and we'll join back at 5 minutes to 11. Thank you all. welcome back to this session of the Professor K. N. Ganesh Endowment Trust lectures. And I request Dr. Shivram, our own former dynamic director of NCL, to kindly chair the session. He is currently associated with ISER Pune as well as several industry houses and continues actively to be engaged with several of us here at NCL still. Dr. Shivram, please.
Just push everybody from outside in. So, my name is Shivram. It's a pleasure to be here. So, I have heard about you, but it's the first time I came. Manisha? Yeah. Okay. Thank you very much. My thanks to the organizers of this meeting, especially the K.N. Ganesh Endowment Trust, the Organic Chemistry Division of NCL, for having given me this platform this morning. I think Nathu called me almost a month and a half ago and blocked my date. So that was helpful, Nathu, because sometimes dates do clash. So I wouldn't have missed this day for any other event in my life. Welcome, Dr. Ganesh, back to NCL. And I can sit here and talk for 1.5, 2 hours, but that's not the purpose today. All I can tell him is that welcome to the club of septuagenarians. So, and I can tell you, 70 is the new 30s, okay? So I have finished 6 years in my 70th year of my life. And I think when you are free of all other, encumbrances in your professional life, and all you can do is sit and contemplate and think and do what you like to do, that's the best phase in your life. No bosses, no telephone calls asking you to do something. You want to do something, you choose. If you don't want to do something, you can say no. And I think you have reached that stage in life where you are the boss, and I think that's the best phase in your life. So welcome. And I always said that Ganesh and I have gone back. It's 40 years. I knew him. Actually, I knew him much earlier, but we became professional colleagues in 1989 in NCL Pune. And I'm extremely, thankful for the National Chemical Laboratory to have organized this event, because I always say that Ganesh's Janma Bhumi was NCL, okay? It was. And his Karma Bhumi was ISER, okay? So, and I think this is where Ganesh became who he was and who he is at National Chemical Laboratory. And I hope that this lab, by organizing this event in his honor, I think has done Ganesh proud. It has also done the National Chemical Laboratory proud. And I will not take much time because we are running behind schedule by 10 minutes. I'd like to invite Dr. Manoharan, Muttaya Manoharan, to the podium, please. I think I don't need to introduce him. His profile is here. But all I wish to say is, if there is an ideal profile of a scientist, in my opinion, an aspiring profile for a young scientist, I think that is here. Okay, and I think all we need to do is to follow him on his footsteps. And I think that's the most fulfilling journey of a scientist: excellence in science, relevance in application, close to 250 scientific papers, close to 200 patents. What more do you want in life? Muttaya Manoharan, welcome to NCL, welcome to this function.
And also I was very much touched by the type of things what this KNG Trust is doing for this country and for the needy people and students. Professor Nata, you are doing a wonderful job. Amazing. Thank you. And for some personal reasons at the last minute, I was not sure whether I will be able to be here in person. By looking at everybody, but it's okay. I wanted to say happy birthday to our dear Ganesh. So that is the reason I gave the title of my talk as Growing Together with the Double Helix and Living in the World of Nucleic Acid Therapeutics, as Monisha pointed out. Now we are living in the oligonucleotide therapeutics thanks to the origin of double helix. Having said that, I want to thank all of you being here and those who have joined online and listening to, and every one of you who helped me to be here. I want to thank you. Particularly for the science part I will be talking about, I want to thank my colleagues at Alnylam, and it has been a 20 years walk, 20 years journey at Alnylam, and all the collaborators, including Professor K.N. Ganesh, who made this science possible, and particularly about the therapeutic areas. I would like to thank the patients, the caregivers, and the clinicians who trusted our drug and treated them to get the outcome. And I was given this big award. Maybe this is the biggest award I ever got in my life today. I was wondering, and I was just thinking about this Forrest Gump line. Miracle happens every day. Some people think they don't, but they do. Like this big miracle I got today. I'm not a very smart man, but I know what passion is. That is what keeps me going. And thank you, Ganesh, Dr. Natu, Monisha, and everybody who was behind this gift what you gave me. I come from Tamil Nadu, and we are all here for one noble mission. When we were talking about the nucleic acid medicine, I want to point out two things. One is what the Hippocrates said, wherever the art of medicine is loved, there is also a love of humanity. Without the love of humanity, the art of medicine cannot be done, cannot be practiced. And this is, in a different way, was conceived about 2,500 years ago by Kaniyan Pungundana in Purananuru, and he said, Yavarum Ure, Yadum Ure, Yavarum Keli. For all of us, it's the same village. We come from the same village, and we are all related. And this is the time when all our Indians must have had connections around the world. Otherwise, he would not have even thought about such a line. So we are here for that noble thinking, how each of us can contribute to human health. So I want to recollect that phrase, Yadum Ure Yavarum Kelir. And this is inscribed in UNESCO. Then I'm coming to the beautiful state of Maharashtra. And thanks to my friend Kaliyappan, there was an organic chemistry symposium happened earlier this year. And I was taken into, for the first time, into the caves of Ajanta. And in the caves of Ajanta, I was stunned to see this picture of young Buddha. And I was told that this is nearly 2000 years old. And I could see still clearly the flower, the face, the beauty surrounding Buddha in this cave picture 2000 years old. What came to my mind is, as a chemist, the chemistry went behind this picture. The pigments which went behind this picture, still very fresh. It gives that beautiful look of expressions of Buddha. So that is happening in this state of Maharashtra. This is to point out how our Indians 2000 years ago were thinking about chemistry and putting the pigments together for such a wonderful painting. Now, I want to come to this growing with double helix. With, as I mentioned, prayers to Lord Ganesha and Lord Krishna and my friend Krishna Ganesh. And in this one, you know, Ganesh is the one who removes all the obstacles. As a friend, I have discussed with him on many occasions what needs to be done practically. He will always, as the Lord Ganesha, he will have a solution. He will tell it in a very nice way to make that happen. And he's a very jovial person, like young Krishna. But what is more important, in writing the Bhagavad Gita, he was there to lead, to give the guidance to his friends and his students. That is what depicted in this thinking about the Krishna Ganesh. And Krishna Ganesh is connected with human genome to the double helix because it happened in 1953. The year in which Ganesh was born is the year in which Watson-Crick double helix was described. It was formulated. And that is an interesting year because that's the time Everest was climbed, 1953. Air India and Indian Airlines joined. Staudinger got his Nobel Prize for this. We are talking about this ligation reactions, but for his chemistry, he got his Nobel Prize in 1953. For people who are not going technically, you can look at, that is the year Tony Blair was born, the actress Kim Basinger was born, and Zinken was born, and my dear Gargi pointed out that is the year Casino Royale was written by Ian Fleming, the first James Bond story. So 1953 is a very important year. And that is the year of double helix. That is the year of Krishna Ganesh. As we talked before, nothing in biotechnology without chemistry. Only if the chemistry happens, biotechnology can happen. And Ganesh has contributed a lot. And these are some of the pictures what I got from, thanks to a lot of you who contributed to this. And I want to give my respect and salute to Ganesha's mom. And I want to appreciate this handsome guy here. I was stunned, a movie star like Ganesh. I was told that this is during his early days in graduate school, namely after setting up his lab at NCL. And here with Mrs. Ganesh, and of course of the lectures when he was giving in ACS, I took this picture when he was so excitingly describing his chemistry. And his chemistry is connected with some of the ancient important chemistry of nucleic acids. He is in the academic tree, he is connected with Professor Govind Khurana, who created so much, who did so much for nucleic acids chemistry. Because Govind Khurana taught Mike Gates, and K.N. Ganesh worked with Mike Gates, and they published a paper very nicely with U3 groups and Gates, everybody in the nice paper back in 1987. And here you can see when Mike Gates was given the Lifetime Achievement Award by the RNA Therapeutics Society, he was remembering his contributors, his postdoc. And here you can see Mike Gates is pointing out to Ganesh. Here is Professor Dan Brown. Here is Ganesh. So this is how the teachers remember his contributions. And Ganesh, on the other hand, he doesn't forget this either. When he visited last year Al-Nilam to give a lecture on the PNA chemistry, he was remembering this connection from Lord Todd, Govind Khurana, Mike Gates, and this work, and this academic tree. I know Professor Ganesh for the past 30 years. Even before meeting him, I was interested in repeating John Moffat's chemistry of the tin-mediated uridine to make the 2', 3' isotopes. I was able to repeat that even until today. My colleagues like Dhruv Datta suffer or enjoy this chemistry every time when we want the 2', 3' modifications. But I was so fascinated to see K.N. Ganesh has modified this chemistry, improved this chemistry using base transfer catalysis. This is the paper which I saw even before I knew about Ganesh. That is how my acquaintance started. Then that worked out very favorably when I saw an application from T.P. Prakash, who joined in my group later at ISIS Pharmaceuticals. And in the early days of nucleic acid chemistry, Ganesh explored all possible modifications, sugar modifications, backbone modifications, and base modifications. And here he is exploring spermine-spermidine modifications as T5. And these cationic modifications, when you bring the anionic backbone containing oligonucleotides, it increases the kinetics of binding to the target RNA and target DNA. But Ganesh was a visionary, and David Corey, after 10 years later, when he did similar work, he pointed out these positive charges increases the binding affinity, rate of binding by 45,000-fold improved. But using a very difficult chemistry, Ganesh introduced this, which is shown here in this slide. Yet another beauty of making the spermine going into this pyrrolidine type structures is done by another favorite superstar from Ganesh lab, Rajiv, Kallantottathil Rajiv. And these things taught me what are the modifications we should use in the oligonucleotide chemistry. And this is probably the best place at NCL to explore all kinds of peptide nucleic acids. Even more than what was done in Denmark, the birthplace of DNA. So many chiral variations, so wanted to have a paper, a co-author publication with KNG. So we did that by bringing our GalMax and his DNA at ISER Tirupati and ISER Pune, and we were so successful in making this happen as a publication, as you can see in JOC. We love conjugates. Now I want to go a little bit into what kind of things Ganesh has done. I mentioned about the Purananuru. Now I want to go into Tirukkural. This is also 2000 years old book. In this one, I will probably read the Tamil one first. Takkar Thagavilar என்பது அவரவர் எச்சத்தார் காணப்படும். So somebody asks Thiruvalluvar the question, how do you differentiate a great man from a so-so man? And the answer from Thiruvalluvar was, the difference between great and so-so people may be known by what they build and what they give to the universe. So in that sense, of course, Ganesh has a great family. That is some of his creations. Apart from that, look at all his students. This was a picture taken, part of his students, taken 10 years ago, and some of his colleagues. And here you can see young Ganesh with some of his students. And most of the people are here right now and otherwise listening to the online. And these are the students he created, developed. And I could think of at least 3 educational institutions he built. NCL Pune, ISER Pune, ISER Tirupati. And I had the fortune and pleasure of visiting all these 3 places and enjoying the chemistry and the chemists who built these organizations. And I would point out that NCL Pune, when KNG organized the lecture, that was the first time I got any opportunity of meeting Ram Zerflo or people at that caliber. Everybody was here in that meeting, which was a big organization done by KNG. Not only the educational institutions, he is building ACS journals, ACS Omega, he is the chief editor. Here you can see his contributions. Now coming back to his students, I had the personal pleasure and fortune of working with so many of Ganesh's students. I had the pleasure, but the field was immensely blessed by that. The current state of oligonucleotide therapeutics was blessed by every individual you are seeing here, along with Ganesh. Now I want to come back again to Tirukkural. The first one is Tandai Magarkattrum Nandri Avayattu Mundi Iruppach Seyal. This talks about the parents and the children, what the parent has to do for the child. The same thing can go for the teacher and the child. So the translation goes like that. The teacher should do everything for their students in such a way they get well recognized in any assembly of the voice. And that is what Ganesh has done for all his 50 plus students. If it is more, I don't know exact number, Ganesh, but you have done that. You have brought them in such a way, when they sit in the Sabha, people get exclaimed, where this person got trained to create such an outstanding scientific thing. On the other hand, to the same Thiruvalluvar, asking what the child has to do for the father, or what the student has to do for the teacher. Magan Tandaikkattrum Udavi, Ivan Tandai Ennotran Kollum Kollenum Sol. The duty of the students is to make other people exclaim what prayers has the teacher has done to get such wonderful students. So that is the kind of thing the students should do, so that everybody will praise the teacher. And I see this chemistry book going back in everybody whom I work with. I can talk as the chair pointed out, Dr. Shiram pointed out, each person for 2 hours, at least 2 hours, of their work ethics, scientific contributions, and so on and so forth. And here is young Rajiv. Now let me move on to nucleic acid therapeutics. How these students came from Ganesh's lab, Ganesh's training, contributed to nucleic acid based medicine. Beginning 1998 until today, there are 19 nucleic acid drugs have been approved, including the 2 mRNA vaccines. Those are all shown here. And for the youngsters in the room, I would like to point out, as a kind of a shameless advertisement, in March of this year in the nucleic acid research, my friend Martin Egley and I, we wrote a complete review about all these compounds, the basic chemistry, how it works, and what are the advantages and disadvantages, and where does it stand, and so on and so forth. Those compounds are all there. I can point out that chemists whom I point out earlier from Ganesh's lab, they have touched each one of these compounds in one way or other. And more recently, our lab has also been successful in making these drugs work not only in the systemic administration, but working on the devastating disease models, something like Alzheimer's, dementia. For that one, we used simple 2' chemistry. Like the tin chemistry what I was talking about, instead of having a methyl group, we have a C16 alkyl chain. This makes the siRNA distribute broadly into the brain so that it can control, it can knock down the APP protein, and that is helpful in curing Alzheimer's disease. And this has been shown in the phase 1 study, very successful progress, the dose-dependent effect going over 3 months' time after a single administration. And this involves, these are the basic chemistries which go into these drugs: DNA, RNA, 2' fluoro, 2' methyl, methoxyethyl, and methoxyethyl with the 5-methylcytidine, and phosphorothioate, N-methyl pseudouridine, which goes into mRNA vaccine for the splicing modulations, PMOs, with the 2 delivery systems. One is lipid nanoparticles, and the lipid nanoparticles containing siRNA, or lipid nanoparticles containing mRNA. And then, as Monisha pointed out earlier, our own GalNAc conjugate, which takes the molecules and delivers efficiently into liver hepatocytes. And this is coming back to the story of Prakash. He was not satisfied with the methoxyethyl. He made a lot of 2' modifications. Prakash is the guy who would come every year. He will write 10 different papers to go, and he will make that happen at the end of the year. He's still doing it at Ionis. And these are all the modifications, because if you make chemistries, if you make that work, if you build it, they will come. The biologists would come. The pharmacologists would come. They will make use of it. And these are some of his publications. And in the case of when I was at ISIS, we worked on 4 hydrogen bonding systems like the G-class, and the 5 hydrogen bonding systems like the G-guanidinium structure. We can see when you make a tricyclic structure with an amino ethoxy group, that can form 4 hydrogen bonds. when you make that amino group into a guanidinium group, you can make that as a 5-hydrogen bond system. And this is the only 5-hydrogen bond system known, crystallographically shown from our laboratory. And this is the crystal structure. Why I'm showing this? When Rajiv worked on this DNA, by taking the C into a C mimic as the G clamp, he showed that by introducing a single modification, one can change the delta Tm by 18 degrees against RNA, or going up to 24 degrees against the DNA. Such amazing chemists came from Ganesh's group. And when we joined at 2003, when I moved from ISIS to Alnylam, the story was nothing known about the RNAi drugs. Our CEO was giving the interview to Forbes magazine saying that it depends on how we figure out the delivery. That is where the success is going to be. And we started with the lipophilic conjugates. Again, through the C16, we introduced, thanks to Rajiv and his team working with us, cholesterol conjugates. And this was the very first in vivo application of RNAi drugs, and which we demonstrated in mouse. And this is happening because the cholesterol molecules make RNAi bind to LDL, HDL, and albumin, gives a broad distribution, takes into liver where we can see the silencing. Then we suffered for a while where we take this molecule, how do we deliver this? So delivery was an issue. We decided to focus on the delivery to liver. And in this case, after this 20 years in the RNAi mechanism, we are successfully enabled RNAi because we are making a double-stranded molecule. We are entering into the cell that goes and loads into RISC, and once it's loaded into the RISC, the sense strand is peeled off. The antisense strand looks for the target mRNA to be knocked down, to be killed. And once that happens, the Argonaut slicer comes and cuts across the 10th position. That way that mRNA is destroyed. And this catalytic mechanism goes on and on and on because the Argonaut is a natural enzyme. It can continue this process as long as you make the right kind of siRNAs. Thanks to this mechanism, we have made 5 approved drugs by the past 20 years effort. And this has happened in the past 5 years. So this is the slicer enzyme, which has the different domains, and this mid domain is the one which binds to the 5' end. And this is our summary of all the compounds what we have discovered and which went and which is helping patients now. The first one is the Onpattro, which is based on the lipid nanoparticle delivery, and all other 4 compounds are based on the trivalent GalNAc delivery. In the interest of time, I will just quickly explain their functions here. Onpattro controls the disease called the TTR polyneuropathy. And as of last week, it has also been approved for TTR cardiomyopathy. And TTR is a protein which is synthesized in the liver. And it is distributes and it is helpful in transporting vitamin A or thyroxine. But when the mutant TTR happens in the liver, the milk becomes curd. So you get, instead of free flowing protein, it becomes like the fibers, amyloid fibers. And that's not the case what you want. So we were able to silence the TTR protein using the siRNAs and loaded into the LNPs, and that helped the patients. That's our first product. The second one is Givlari, and this is to control acute hepatic porphyria. This is a rare disease, and it attacks mainly female population. And when they get that attack, that pain, it happens very frequently, they cannot do anything else. They don't know the origins of that pain. It completely stops their function. It is due to a liver malfunction. A protein called ALAS1 produced in the liver accumulates heme intermediates into the liver and into the blood system, and that causes this pain. So we were able to silence that ALAS1 gene because this is again a liver problem. We were able to control that, and this helped for the first time cure for acute hepatic pharmacuria. The third one is oxlumo. This is a simple calcium oxalate problem, but this is not a simple calcium oxalate problem. Sometimes kidney stones happen due to the food we take or something like that. But some people, genetic mutation makes this oxalate, calcium oxalate, due to misfunction in the liver. Even in the infants, when that happens, that calcium oxalate deposit can happen everywhere in the body, totally causing this oxaluria conditions. The only cure is double transplantation, namely you have to cure the liver and also the affected kidney. So we were able to control the primary hyperoxaluria and able to help even the pediatric populations so that they don't need to go through this liver transplant. And that is also through the GalNAc conjugate. And another example is the Lequio, which controls this protein called the PCSK9, hypercholesterolemia. We have, in Indians, including myself, we have statin controlling hypercholesterolemia. But not everybody is responsive to statins. In those cases, cholesterol level goes up. In those cases, by knocking down PCSK9, one can bring down the LDL cholesterol. And we have shown, and this compound has been approved, by a single injection, subcutaneous administration, for 6 months, 60% of LDL cholesterol can be brought down. So these are all the compounds what we have made, and we have made it into the market thanks to oligonucleotide technology and thanks to oligonucleotide chemists. And so this is the lipid nanoparticles what I was talking about. And the lipid nanoparticles take the material into liver cells and make this endosomalytic release happening efficiently. And that gives the cure for this TTR polyneuropathy and TTR cardiomyopathy. And this I point out because I met this doctor in UK, and when we met in a scientific conference, he came and introduced himself, and he said, so far I was just maintaining my patients for the past 5 years or 10 years, because the average lifetime once after the TTR amyloidosis is detected, the lifetime is only 5 to 10 years. Only now, thanks to Onpattro, I am giving their life back. They are able to live. They are getting their normal life. And this is true for each compound what I talked about so far. And Onpattro validated the lipid nanoparticles. Lipid nanoparticles validated the mRNA vaccines, and mRNA vaccines helped us through the pandemic. And just like Carolyn Petruzzi says, when the world is in trouble, chemistry comes to rescue. Let me quickly point out the contribution of the GalNAc from Rajiv and other NCL scientists. What we wanted to deliver siRNA is using a trivalent GalNAc, and this trivalent GalNAc, subcutaneously administered, has receptors known as ACLO glycoprotein receptor in the liver hepatocytes. These are the tulip-looking liver hepatocyte ACLO glycoprotein receptors, and we are sending the trivalent GalNAc. And that is a naturally existing receptor which takes galactose containing dead proteins so that the dead proteins can be degraded in the endosomes and lysosomes. we were able to take this mechanism, attach siRNA so that these molecules will be taken into the liver hepatocytes so that they can do the therapeutic function. And that is what happening, but we are giving a naked siRNA, not within the lipid nanoparticles, so we need to give the metabolic stability for the molecule without compromising their potency or without affecting their safety. For that reason, we have to make siRNA conjugates in larger, sufficient quantities with right kind of chemistries. And here are my heroes who made this chemical conjugate, GalNAc conjugate, possible. You can see Rajiv, you can see Prakash Nair, who are these NCL people. And I would say if Rajiv hasn't worked with me, this GalNAc would not have solved. Because from the day one of 2003, we worked together in various ways planning. It took a while. We made this first in 1 gram quantities, then 100 gram quantities, maybe in a kilogram quantities, and shown here. And now, in this country, thanks to the laboratories in our place, in our mother country, GalNAc conjugate has been made to 100 kilogram quantities. It is going to be made in ton quantities, helping thousands of patients around the world. For example, the inclisiran, PCSK9 inhibitor, has been, just for the clinical trials, has been used for 18,000 patients. And the starting material comes from India, Trivalent GalNAc. Thanks to the process developed by Rajiv and others. So I will quickly go through the rest of the chemistries what we use for the trivalent GalNAc conjugate. We use 2'-O-methyl, 2'-fluoro to protect against the endonucleases, and we put phosphorothioates at the terminal ends, and we bring vinyl phosphonate in the antecedent strand so that it will load properly into the mid-domain of the organoid. And we also make specificity improvement by bringing a xenonucleic acid called glycol nucleic acid, which pushes away unnecessary, unrelated mRNA so that the on-target effect would be maintained. So these are all the chemistries which are used, and this is our recipe for making effective siRNA therapeutics today. And that's how the vinyl phosphonate interacts in the mid-domain and mimics a natural phosphate. And as you can imagine, the vinyl phosphonate doesn't get cleaved, so it's metabolically stable. So I mentioned about the story of acute hepatic porphyria and how it helps this woman who had this attack, frequent attacks. And historically, this was misunderstood disease, and King George III was blamed to be a mad guy because he had this acute hepatic porphyria. All these people were victims of this disease. As I mentioned, Oxlumo helps the infants in coming over the glycolate oxidase problem, and PCSK9 reduces the 60% of their cholesterol by once every 6 months injection. So we can control similarly blood pressure using the siRNAs, and we are doing this in a study called Cardio for knocking down the angiotensin gene, and we can connect two different siRNAs with one linker, with one GalNAc, so that in a metabolic disease we can control both hypercholesterolemia and also high blood pressure. More recently, we are moving into making one application. It's called amino-oxy conjugation chemistry. instead of keeping just one amine and acid to bring an amide bond, we are using amino-oxyclick chemistry so that we can bring two ligands to the same nitrogen. In addition to forming the oxenes, we can make homo-bis conjugation or hetero-bis conjugation. And for example, here you can see the folate molecule which targets to the cancer cell also has a C16 protein binding molecule. Such conjugation chemistry is being developed in our laboratory by Dhruv Datta, another person from NCL Pune, ISER Pune combination. Thanks to Ganesh. I have taken too much of time, but I want to leave with few comments. In spite of this 40 years of work in the oligonucleotide therapeutics, we haven't solved everything. There is no other GalNAc beyond GalNAc which we have identified which will take the molecules into different tissues. So that is an issue. And then similarly, we cannot make oligonucleotide drugs which can be taken as pills. Oral delivery is not possible. Oligonucleotide drugs don't cross blood-brain barrier penetration. And microRNAs, targeting microRNAs has not been successful. mRNA works as vaccines, but mRNA has not been made as drug molecules. So there are a lot of opportunities exist, as mentioned earlier during this session, that nucleic acids offer a lot of promise to go into different tissues, different modalities. So there's a lot of opportunities for all of you. Whenever I come to India, when I meet these youngsters, I think they should go and contribute some extent into this nucleic acid therapeutics. And I very fondly remember my friend Jean Albert's comment in Accounts of Chemical Research, chemists get back to the bench. With that, I have taken too much of time and explaining, but I covered few aspects. I believe I want to thank you for your patience and putting up with me. Appreciate the opportunity. Thanks for the award. Thank you very much.
Thank you, Dr. Manoharan, for this scintillating journey that you have so lucidly presented. Even somebody like me who doesn't understand anything about oligonucleotide chemistry and drugs, I think I felt as if I have understood something today. Thank you very much. I think we have a few minutes to ask a few questions. If anyone has or any comments or any thoughts, the floor is open. Is there any question? Any comments? Yeah.
What are the approaches you think about the which can enter into the cells rather than the liver? So,
That is a great question. We are working on it. One of the things which we are using, particularly in going into the brain, is just the lipid conjugate, because that takes the molecule and gives the broader perspective, broader distribution into different parts of the brain. So that is one thing. Alternatively, we can think of folic acid, which takes it into the cancer cells, mannose ligands, which takes it into the dendritic cells or Kupffer cells. So those are the types of ligands which we are thinking. And people now taking antibody conjugates or FAB fragments or centerings or peptides. So all types of conjugates are being tried and evaluated. So as far as we can find the right kind of high expressing, copy numbered, high capacity receptors and the correct ligands, we can attach, we can overcome this part of that. That's a good question.
Great talk. You know, in reference to that question, have you tried, conjugating? You have conjugated with cholesterol and long 16 chain and all those. What about tocopherol? This is because this is known to go to hepatic cells. And we had done some work based on those kinds of lipid construction and found they are particularly targetable to hepatocellular carcinoma.
Yeah, we have evaluated tocopherol also. Interestingly, in this amino oxide chemistry also, tocopherol has been tried as one of the ligands. And what we believe is the hydrophobicity of these molecules, the lipid spectrum, plays a role, rather than some other receptor-mediated processes. But tocopherol may have a receptor also, but we take more advantage of the hydrophobicity. But in exploring, as you said, in HCC, we can explore that possibility.
Sure, you have to optimize the lipophilicity and hydrophilicity balance.
Yes.
And then it could become.
Yeah.
Hi, great talk. So this the specificity is coming because of the presence of the GalNAc conjugate, right?
Yes.
In that one, the receptor is GalNAc mediated thing, which is overexpressed in liver, hepatic cells. Did you also consider targeting other organs by changing the sugar from GalNAc to mannose or glucose or glucosamine?
Mannose is a good ligand. Unfortunately, for the mannose, the balancing is not very well known or described. For this trivalent GalNAc has the receptor, the structural studies are known. But we are trying that in optimizing the hydrophobicity along that. Hopefully when next time Guru comes, he will give a talk on that mannose conjugated siRNAs. Yeah. So that's something which we are evaluating. Fucose is another possibility for oncology targeting that one. So that's also we are evaluating.
Yeah, of course, mannose synchronizes with your name, Nano too.
Oh.
Anything else? Anyone else?
So,
Yeah.
Just this thing. So your last slide, you had oxide conjugation. May I ask why not this 3+2 addition with azide alkyne? This thing does it not?
Oh, okay. That is the classical click.
Yes.
Yeah. The classical click, azide alkyne, involves obviously copper. Although it is a very nice research reagent, when we take it into therapeutics, copper metal ion becomes an issue. So people prefer to use copper-free click rather than copper-mediated click. That is one aspect. And Sharpless actually defined not only the alkyne azide as the click. He said everything possible which you can do like that is a click. So in that sense, the amino oxy is a good click because you can put two aldehydes, whether they are different functionalities or the same functionality, you can make that quick reactions. So that is another exploration we are interested in. And it doesn't require anything like a copper ion or anything like that. Good chemistry question. The GalNAc is, we are putting it into the sense strand. Not only that, it's once it goes, because it's a very quick turnover time. It's only 15 minutes from the time of sub-Q injection to reach to the receptor. It goes there and delivers this. Once it goes into the tissues, immediately glycosidic linkage breaks, so the sugar is gone. So it doesn't have any more recycling process and so on. To thinking that whether we could stabilize that glycosidase ring by other chemistries, we replaced the oxygen with sulfur, nitrogen, and carbon. And what we found is nature is the best. Nature's Galnac is the best. Even with chemical modifications, we couldn't improve anything further. Galnac goes, it delivers, it leaves. Yeah.
That should be the last question, okay? Yeah.
Hello, sir. It's a great talk. As you said, the oligonucleotides is unable to cross the BBP. So what could be the possible solution? A nucleic amino acid or maybe PNA or Janus PNAs. So there is any possibilities?
Yeah. One of the things people are exploring is going into the antibody constructs, thinking that antibody through the leaky brain curvature so they can transport. So those, and last year there was a paper appeared by taking this lipophilic conjugates, like they've taken the cholesterol conjugate from a Japanese worker, but it required a very high concentration, 50 milligrams per kilogram dose. So that work still needs to be done. We need to understand the blood-brain barrier penetration issues so that we can address and overcome this. But that would be an eventual great solution, because brain-related problems, whether it is simple schizophrenia, Alzheimer's, ALS, Parkinson's, Huntington, so many devastating problems need cure.
Thank you, sir.
Thank you very much,
Thank
everybody.
you. I'd
Great.
like to stop here, not because there are not because there are no more questions, but I think we are running behind schedule, and I need to give some time to the next speaker too. So may I request? Okay, sure. Okay. Thank you. Let's thank Dr. Manoharan for a lovely talk. Thank you. Thank you so much. May I request Professor Jyotirmay Dash to come to the podium, please? I think, again, I will not read what is written here, but she is one of India's foremost bioorganic chemists today, and with the great recognitions already achieved in her career. And we invite her to deliver this second talk, which is bioorthogonal synthesis using nucleic acid templates.
We will change the computer.
Pointer.
Yeah, good afternoon, everyone. Thank you, Professor Shivram, for the introduction. At the outset, I would like to thank the KNG Trust for giving me this opportunity. It's a great honor and recognition for me. And the last talk very nicely illustrated the life and the achievements of Professor Ganesh. And I would just like to say a single line that Professor Ganesh has been a source of inspiration for us, for our generation, for the new and the young generation. And not only that, not only as an institute builder or academician, he has been playing a pivotal role in advancing chemical biology research in our country. And I think I have been greatly inspired by his work since from a student and now. and I would like to thank him. And also on this occasion, I like to wish him a happy birthday, and also, congratulate him for for being honored the well-deserved honor of Padma Shri. And with this, I would like to start our research work, that is bioorthogonal synthesis using nucleic acid templates. So bioorthogonal reactions are a class of reactions that rapidly join molecules under physiological conditions. And as you know that the Nobel Prize in Chemistry in the year 2022, it says click and the molecules are joined together. And it was awarded to Bartolozzi for coining the term bioorthogonal chemistry, and the reaction that has been used, the classical reaction that was developed, independently developed by Meldal and Sharpless, that was copper-1 catalyzed alkyne and azide cycloaddition, giving only the 1,4 triazole compounds. So since I have kept my talk the title bioorthogonal synthesis, I would just like to share that these reactions occur under physiological conditions, and the reactants are inert towards biological processes, and they are very selective towards themselves. And the functional groups are thermodynamically stable and metabolically stable. And the reaction proceeds very rapidly, very fast, even at low concentration of reactants. And most importantly, the reaction provides a single stable product without any byproducts. And if we are talking about drug and delivery systems, then the product must be non-toxic. Is it on? That's not working. Is it on? You just check if it's on. It's not on. That's why. No, this light is not. Sorry. Now it's okay. Okay, maybe you can take this now. On the functional groups and the products must be non-toxic, the starting materials, and the products must be non-toxic to the living system. So this is the outline of my talk. This is what we have been working. At the beginning, we have just used copper-1 catalyzed alkyne and azide cycloaddition to design ligands for targeting DNA secondary structures. So as we all know that the year 1953 marked the beginning of the genetic revolution, and we celebrated the 60 years of DNA in the year 2013 at ISER Pune. And I had the privilege to attend the meeting. And it was an international conference, and many well-established researchers in nucleic acids and chemical biology, and I just started my career in the realm of DNA. So I'd like to thank Professor Ganesh for inviting me, and it was really a great platform to share knowledge and also scientific exchange. Again, we also celebrated 65 years of DNA at ISER Tirupati. And at that time, I delivered a talk. And today, we are achieving two different milestones: 70 years of DNA, and we are celebrating 70th birthday of Professor Ganesh, and who happens to be the pioneer of DNA research in our country, who have synthesized DNA and DNA-related work like PNAs, and also studied their intricate functions. And in our group, we are designing molecules for targeting these nucleic acid secondary structures like G quadruplexes and its complementary I motifs. So as I said, at first we have used this simple reaction, alkyne and azide cycloaddition in aqueous media, to synthesize a series of triazole compounds with the goal to target the G quadruplexes. as we have mentioned in the first line in this book chapter, that this is selectively targeting a particular DNA quadruplex. It's just like finding a needle in a haystack. So however, we have designed several compounds that can selectively bind and stabilize DNA quadruplexes over the double helical DNA structure. And those compounds are like derived from carbazoles, isoquinolines, even natural nucleoside guanosine derivatives. So then to selectively target a nucleic acid secondary structure, we thought to use nucleic acids themselves to synthesize their own ligands. So then we moved on to the nucleic acid directed cycloaddition, that is nucleic acid directed in situ cycloaddition, or we can say as in situ click chemistry, that was copper free. So I would first talk about this nucleic acid directed copper free in situ click chemistry, and then I would highlight its application in bioorthogonal chemistry in living systems. So the in situ click chemistry is a class of reaction that should occur without side reactions towards the endogenous functional groups of the biological target. Like the biological target should select its own alkyne and azide fragments and then should force it to react to give the product that can strongly bind to the target. So in general, there are two different target guided methods. One is dynamic combinatorial chemistry and the other is in situ click chemistry. So in this approach, simple fragments assemble through reversible bond formation and generate a combinatorial library of interconvertible building blocks or interconvertible products. In the presence of a target, the equilibrium of the system shifts towards those products that can strongly bind to the biological target. So today I'm not going to discuss about the DCC. I would just like to talk about the in situ click chemistry. However, both the approaches we have summarized in these recent review articles, and the review article only summarizes nucleic acid guided all the literature reports. There are many reviews on DCCE or in situ click chemistry, and they were like for the proteins, because this in situ click chemistry has been widely used for protein targets, and it was the first report by Sharpless that was also for the acetylcholinesterase to identify the inhibitors. But for nucleic acids, only a few reactions or few approaches are known, and which have been all the methods, all the literature since the beginning has been summarized in this article. So however, it seems like the synthesis is unscreening to one step and it's very simple. However, the process is quite complicated. So in the beginning, when we have started, like we have used a mixture of alkyne and azide fragments and incubated with the DNA quadruplex. Then we try to characterize, after some days, we try to characterize the products by HPLC and mass. However, we could not isolate any compound, and it was the identification of the products was quite difficult because the products were also overlapped with the starting materials. So thus we thought of immobilizing DNA targets on gold-coated magnetic nanoparticles. So we have taken thiol containing DNAs and we like quadruplexes and I motifs and the control templates like double helical DNA and a single stranded DNA. So first we have taken two different quadruplexes which are present in the promoter regions of CMYC and BCL2 oncogenes, and they are complementary I motifs. And both these genes like CMYC and BCL2 play key role in cancer progression. Therefore, regulation of these genes by targeting these quadruplexes and I motifs could be a potential anti-cancer strategy. So this is our approach. where we have taken a mixture of alkyne and azide fragments, incubated with the quadruplex or the DNA nanotemplates. After giving sufficient time, we have used an external magnet to isolate the DNA along with the newly formed products, and then we have heated them to identify, to isolate the products, which were then characterized by HPLC and mass. And here the process did not interfere with the native functional groups of the DNA, and we could isolate the DNA nanotemplate and again recycle. So now we can say that the target guided synthesis using DNA nanotemplates can be used to achieve the desired selectivity in complex chemical and biological systems. So this is the title I have taken from this paper by Bartoji, and this is the picture in this paper. So next we have used a mixture of alkynes and azides. The mixture could lead to 136 possible compounds, but I will just show you one example, and we have identified the maximum hit compounds, that is 6, for the c-MYC I motif. And these are the compounds, like without synthesizing and screening them, we could find like 6 different compounds for c-MYC I motif, and even we could get only one compound for the BCL2 quadruplex. So by comparing these compounds and the compounds that were obtained by the control duplex DNA, we could identify 4 selective leads for each of these DNA templates. So then, without doing any biophysical screening, we moved to the toxicity studies, and we have taken 2 different cancer cells and a normal cell line. So 3 of these ligands could selectively kill the cancer cells over the normal cells. Then we have also carried out biochemical assays like Q-RT-PCR and Western blot, which reveal that these ligands can downregulate the c-MYC expression in cancer cells. So I'm not going detail, and further studies like an exon-specific assay revealed that the compound can downregulate the c-MYC expression by directly stabilizing the DNA quadruplex structure. So next, our next question was, instead of using DNA quadruplex itself, can we use a biomimetic template of G quadruplex to promote the alkyne and azide cycloaddition? So this inspiration comes from the enantioselective reactions in which a small amino acid like a proline can catalyze the enantioselective transformation. So we thought if we can make a G quartet, which is the basic building block of G quadruplexes, then can it promote the same reaction? So for that, we have prepared a hydrogel system from guanosine and boronic acid. So this hydrogel contains entangled network of these G quartet fibers. So this G quartet is the guanosine quartet, which is the basic building block of these G quadruplexes, that is the coplanar arrangement of 4 guanines that are linked together by Hookstein type of hydrogen bonding. So our hypothesis was the products would bind to the gel network and strengthen the gel, and thus could drive the alkyne and azide cycloaddition. So thus we have taken a mixture of alkyne and azide fragments that we previously prepared for the in situ chemistry, and then we have incubated these mixtures with the gel network, and then we destroyed the gel and again characterized by using HPLC and mass. So quite remarkably, we could identify the compound that was formed was similar that was obtained by the c-MYC quadruplex. So thus, we can use cost-effective materials like a gel system than quadruplex itself, and since this is a copper-free synthesis, we can make these compounds in lab scale. And this study could also design other biomimetic synthesis or other biologically relevant DNA, RNA, or protein targets. So then from DNA, we just thought of using this method to RNA secondary structures. At first, we have taken this HIV 1 tag RNA. This is the structure of this HIV1 TAR RNA structure. This is TAR RNA which is present in the HIV1 genome. So it is a stable hairpin structure with a 3-base UCU bulge and a hexanucleotide loop. So the bulge region of this TAR RNA is the binding site of the TAR peptide, and CAP and TAR interactions promote viral replication. So TAR RNA thus has been targeted by small molecules to inhibit TAR-TAR interactions and thus viral replication. And ligands like antibiotics, aminoglycosides, neomycin D, and several heteroaromatic compounds are known as TAR binders or potential antiviral agents that inhibit the TAR-TAR interactions. So considering the folding topology of these RNA structures, we thought to use in situ cycloaddition to identify or selective ligands. So we have taken the TAR RNA as the target template. Then we have taken its corresponding TAR DNA and a TAR RNA without the bulge region as the control templates. So in this case, we have used a thiazole-based peptidomimetic. We have made 3 different alkynes and a series of simple azide fragments. So in this case, again the reaction is simple. Take the mixture of alkyne and azides in the presence of a biomolecule, and we have taken here biotintact TAR RNA, and which could be separated by streptavidin-based magnetic beads. And HPLC analysis revealed like the formation of 3 different compounds. This monomer, thiazole monomer, was obtained by the TAR RNA without the bulge region, and both these compounds, dimer and the trimer, were obtained by the TAR RNA, and only this primary compound was obtained by the TAR DNA. So, and also this in situ cycloaddition also provides insights into the binding mode of these ligands. Since this monomer was not working. Since this monomer, that is 3AA, was not formed by the TAR RNA or TAR DNA, and the other 2 compounds, they are also not formed by the TAR RNA without the bulge region. So it suggests that these 2 compounds can bind to the bulge region of the TAR RNA. Next slide. So we have conducted 2 complementary biophysical techniques like isothermal calorimetry and fluorescence binding titrations, and both these methods suggest like the compound which was this dimeric peptidomimetic, which was obtained as the major compound in the HPLC, binds to the TAR RNA with nanomolar binding affinity and 1:1 stoichiometry. Now, for the molecular modeling and a thread-based assay further revealed that this compound binds to the bulge region of the TAR RNA and can also displace TAR peptide from the TAR RNA. So thus this is a rapid method to identify a new class of peptidomimetic ligands for the TAR RNA by just using an in situ alkyne and azide cycloaddition. So I have shown that we can use alkyne and azide cycloaddition, in situ cycloaddition, to synthesize selective ligands for G quadruplex, for I motif, for TAR RNA, and we can also use a biomimetic template of G quadruplex to synthesize c-MYC selective ligands. So thus, for bioorthogonal reactions, we have shown that the reactions, I have shown you that we can do alkyne and azide cycloaddition in aqueous media and design ligands for targeting DNA quadruplexes and I motifs. Then I have shown the in situ cycloaddition in the presence of biological molecules without affecting the functional groups of the DNA, and then I would like to move on the reaction inside living systems without interfering the biological processes. So we thought of using this in situ cycloaddition to synthesize macrocycles. So most of the compounds known are linear aromatic or aliphatic by using the TGS. So we thought, can we use this quick method to synthesize macrocyclic compounds? And most of the classical G4 binders are macrocycles, for instance, telomestatin, oxazole, and porphyrin. However, it's difficult to synthesize telomestatin, and it involves multi-steps, and many people synthesize the analogs of telomestatin, and this porphyrin is non-selective for the G4. So we thought of synthesizing pyridine-based macrocycle. We have synthesized bis alkynes and bis azide fragments, and then again we used the same method of several DNA sequences, 6 different DNA sequences immobilized and DNA on the gold-coated magnetic nanoparticles, and the HPLC and mass analysis revealed that a macrocycle can be formed in situ. So actually the macrocyclization reaction is quite challenging, particularly the macrocyclization step. We need high dilution conditions, and it's difficult to control oligo and polymerization reaction. So this product was formed by both H-telo, that is human telomeric quadruplex, and c-MYC quadruplex. And the product formation was higher by the c-MYC than the H-telo quadruplex. Then we have used again copper-1 catalyzed transformation to synthesize structurally different, 4 different macrocycles, including the M1 that was obtained by the TGS. And this, we could crystallize the M4, that is the core molecule of this macrocycle, and its size is similar to that of G quartet. So therefore, the planar G quartets present in the DNA quadruplexes actually provide a size complementary reaction platform for the macrocyclization of the bis alkyne and bis azide fragments over the oligo and polymerization reactions. And then we have carried out 3 different studies like freight-based melting analysis, fluorescence binding titrations, and isothermal calorimetry, which revealed that the macrocycle M1 and another macrocycle containing one side chain binds both c-MYC and H-telo quadruplexes. So next, moving on to the cytotoxicity studies, all these macrocyclic compounds were toxic to the cancer cells over the normal cells, and again we have carried out this biochemical assays like qRT-PCR and Western blot, which shows that it can selectively reduce the c-MYC expression without affecting the GAF-DH in expression, that is the housekeeping gene. So that means the downregulation of c-MYC gene expression is specific for the c-MYC gene. So next we have carried out the immunocytochemistry using G4 specific BG4 antibody. So BG4 is quite specific for the G quadruplexes, and it produces red dots, so which shows that the existence of G quadruplex structures in the cellular system. And the macrocycle also localizes with the BG4, which indicates that it binds to the G quadruplex structure in the cellular system. And next, it also shows that it could also increase the BG4 foci to 2.6-fold, which suggests that it can bind and stabilize the endogenous G4 DNAs in HeLa cells. So, inspired by its synthesis by the target itself, by the G quadruplexes itself, and its toxicity in cellular toxicity and anti-cancer properties, we thought of using HeLa cell as the reaction vessel to carry out the in situ cycloaddition. So then what we have done is we have taken its alkyne, this bis alkyne and bis azide fragment, and incubated with the HeLa cells. And after 48 hours, the cell extracts reveal the formation of M1 along with the alkyne and azide fragments, which indicates the bioformation of the M1 in the cellular system. And further, the macrocyclization in cells was confirmed by immunocytochemistry using the BG4 antibody, which shows that when we have combined both the bis alkyne and bis azide fragment, it could increase the BG4 foci to 2-fold, which indicates that it is formed and binds and stabilizes the G quadruplex structures, and also the bioorthogonal synthesis of macrocycles in cells. So since the HeLa cells are abundant in the c-MYC and H-Tilo quadruplexes, and since this macrocycle binds to both the quadruplexes, so the cell could be used as the platform to synthesize the macrocycle in cells. So this is the first DNA templated macrocyclization using the non-covalent interactions. And second, and the macrocyclization like challenging reactions can occur in cells. This also demonstrates like the flat surfaces of any other biomolecules can be used to synthesize selective ligands. So this is, the cover page of the Angurenta, and this approach can be useful for the biomedical applications where we don't have to synthesize, the cell has to synthesize its molecule or the gel has to synthesize without the use of any reagents or chemicals. So thus it also shows that we don't have to need any chemicals, any reagents, but careful choice of the building blocks and fragments that can lead to important therapeutic agents. And so this is like our group research activities. Already I have discussed about the targeting nucleic acids. I have shown one example of the self-assembly of nucleobase, which can also be used for in situ click chemistry applications or to template reactions. And we also carry out, we also use DNA structures and nucleobases to design ion transportation systems. In addition, we also synthesize small molecules that is particularly with a focus on carbazoles so that we can modify the system for biological properties. So here I would like to also show one slide like PNA means in our country we know Professor Ganesh, and we also put forward a small effort and we have synthesized a PNA mimic, not a PNA, and where the nucleobase is replaced by a thiazole ring. This is a short PNA, so it is cell membrane permeable, and it could also modulate cellular function. And this is, I completed I think before time. So this is my group over the years, and I would like to thank CSIR, DBT, DST, IACS for the funding, and it's also my first visit to NCL Pune, and I'd also like to thank the organizers of NCL Pune for giving this opportunity, and thank you all for your kind attention.
Thank you, for that very lucid exposition. Any questions? She can answer.
Excellent talk, very nice. Just I'm concerned about what could be the mechanism when you're doing the click chemistry without the copper.
This is because your molecule, the products, bind to the nucleic acids, yes, DNA or RNA, and then they minimize the energy of the reactions.
So just you're saying they're lowering the energy gap and they're...
Yeah, so you are just like your biocatalytic transformation, like enzyme catalyzed transformation, you decrease, the energy, the transition state, the energy of the transition state. So you minimize the energy of the system and your product is formed.
So any selectivity...
But in this case, we are not using catalytic, we are using like stoichiometric, because we want to identify and then synthesize the product.
Okay, so there is any selectivity when you are using the I motif for the click chemistry and G quadruplex for the...
Definitely the compounds that are formed by G quadruplexes are not formed by the I motifs.
Okay, did you try any time with the duplex DNA they are doing the click
Duplex
chemistry?
DNA I've shown you, like we have got, so you have to take the sequence of your choice and you take a library and definitely you get something. But not always. If they binds, only they would form. So if you see here, you see for duplex DNA also we have many hits. So that's efficiency analysis, and you can see like 5 different compounds.
Okay, thank you.
very nice talk regarding click chemistry. So just a small query about gold nanoparticles. So along with that, you are using buffer which contains a salt. So is there any possibility of leaching of the metal nanoparticles?
That's very good question. And actually, gold nanoparticle, we just immobilize and carry out the transformation. We don't keep it for long and then use. It won't work. So we just freshly make them and use it. Because this is required. Anyway, you need a biotin DNA rather than we just thought of using gold nanoparticle, which is easier to make in the lab. And so here, only thing is, as I said, it would help you to identify the products. The HPLC would be more simpler rather than getting a mixture of compounds where you can't identify the products and starting materials. They, or all overlap and you can't really identify your product.
So excellent talk, Jyoti. So the last macrocyclic which bind to quadruplex. So can this molecule induce the quadruplex formation?
Yes, as you are seeing, they are increasing BG4 foci. So it indicates that all molecules do not increase the BG4 foci. Only they co-localize or localize, you can say, because we are not seeing like BG4 particular dots. You can see the BG4 foci, you can count, but our molecules is dispersed, yes, on all the nucleus, and but they increase the BG4 foci. So and in many cases, you will only find molecules, only they would be localized. So if it increases, then you can see that they're stabilizing.
So, but the BG4 is also a quadruplex inducer.
Yeah, that's a quadruplex inducer, but yet if you add your molecule, it is inducing even more.
Okay, okay, thank you.
You want to ask? You go first. Sorry.
I have a question. The nice structures which you are forming with the several nitrogens, have you looked at the pKa properties of those constructs?
Not really.
Because it seems like they will have some interesting cell permeation properties by themselves.
Yes. They are, most of them are cell membrane permeable.
Yeah, and it would be nice to know the pKa properties so that we can relate it.
Yes. They can use so many molecules, then we can also at least can do a structure activity relationship with respect to pKa.
Thank
you. Yeah. Nice talk. So you have azide component and the alkyne component. So can I consider like the azide component is binding independently to the quadruplexes and the alkyne independently, or with low affinity maybe, and maybe after the click reaction the binding becomes more?
There could be two different things. You can think of that in some cases, you see, one of the fragment can also bind, or it can be both. Both can bind and then promote the cycloaddition, because they would bind to the DNA and would come into close proximity. Then only they would react.
So after
sometimes if your weak binders, we may not notice in this in situ click chemistry.
But your final molecule is two components. So did you check independently the azide and alkyne? So both are binding, right?
No, we have checked. In some cases, we don't get even up to 50 micromolar, no binding.
Okay.
But in some cases, you see in this case particularly, you see here you are seeing little bit, only two-way, little bit showing blue fluorescence, little bit binding. But the azide, no. But when we combine both, you can see that it is
another quick question is like how
binding more strongly than the independent or individual components.
Yeah, thank you. So another question, so how selective is the different topologies of the quadruplexes? For example, parallel versus
exactly,
anti-parallel.
They play a key role, as you say in this case. You see we have taken like CMYC, 2 c-kit.
So I don't know the topology.
And BCL2 and Stilo, but you are seeing that we are not getting the product, the macrocycle with c-kit or BCL2. Only Stilo and CMYC, the product was obtained. Only by the CMYC and Stilo the product was formed. But it was not obtained by by the c-kit or BCL2.
So, but the
quadruplexes.
topology is same for all the quadruplexes? Or
No,
different?
no, topologies are different. You see, simic parallel and HTLO is, a mixed type of topology.
Okay, and suppose just one last question, I'm sorry. So if you have the same topology for the two quadruplexes, for example, you have one RNA quadruplexes and one DNA quadruplex with the same topology, it's basically parallel, right?
No, no, no,
So
topology, even if topology
do you think the
is-
molecule can differentiate between RNA quadruplex and
Yeah,
DNA
yeah,
quadruplex?
these molecules do not bind to the RNA quadruplexes.
Okay,
So even if topology, the loop structure and loop, that is also, play a key role. It's not that is the same topology, SIC also parallel, CMYC also parallel,
all
but the molecule doesn't bind
Okay,
to
fine.
both.
Very nice. Thank you so much. Yeah.
Thank you. I think we should close here. You are very organized. Can we close here or you want? Maybe one more. Last. Okay, last.
Yeah, Jyoti, that's a nice talk. So these are thiolated oligonucleotide. Are you going to, have you actually put them on gold or these are?
No, no, I put them on gold.
Okay, so you require a pre-organization, not just at the nucleotide level, but also on the surface of a nanoparticle. So
No, without the thiol, the DNA will also make the reaction, but the difficulty is in identification.
no, that I understand, but that is why I want to ask you this question. See, originally what you are trying to do, you are actually reproducing the chemistry done by Huisgen in 60s, the dipolar cycloaddition, okay, which is, now it is famed because Harplace has coined the term click chemistry, brought in copper and all this. And you got rid of copper and brought into nucleic acid. Now, what is the effective molarity of your fragments on the surface of the nucleic acid,
No,
which can actually explain a requirement of no Lewis acid?
No, Lewis acid is, no, not for that. That gold is not catalyzing the reaction. Because,
no, but do you use copper here?
No copper.
So copper is not required.
No copper.
So
You see here, for your example, it is there. If you see the RNA, we have just used a biotin. There is no gold, but the reaction takes place. So
Correct.
it's not the gold is doing. We have also carried out this reaction only in gold. No reaction takes place.
No, I am not.
You have to heat it. You have to give copper. If you do it independent of nucleic acids, but this reaction takes place because only they bind to the nucleic acids.
So now can you actually thermodynamically analyze this because of the effective molarity that you are enhancing, because of the pre-binding on the nucleic acid surface? Can you add these two things and explain this, what is the origin of this?
No, the origin is, this is already developed, I think. That,
Anyway, we'll discuss this later.
Yes. Okay.
Make you climb up a little bit. Surprise. I wasn't prepared for it. Professor Jawad Iqbal. Hey, how are you? Good to see you.
It's my pleasure.
Thank you.
Sure.
Thank you.
Thanks.
Thank you all. Before we break for lunch, there's a small announcement. So we have the representative from Vidyarthi Sahayak Samiti who was unable to join us earlier, who is just managed to join us now. So I would like to call him up on stage to kindly accept this contribution from us. And I'd like to call Dr. Nathu once again on stage to please come and hand it over to them. So Mr. Tushar Ranjankar is the managing trustee of the Physiology. We played the video, but thank you for accepting it on our behalf. Thank you. So we will now take a break for lunch. It is at the ISER Pune first floor dining hall, and transport has been arranged to get you from here to the dining hall and also to bring you all back later on. So if you have, so we meet reception area, please do take time out, because I think the slot at the around 4 o'clock that we have kept for that may not be sufficient, because there are a lot of things to see here. Thank you.
हॅलो येतोय हॅलो कैमरा चालू है ना नहीं टाइम लगता है थोड़ा सा उधर आने को हेलो हेलो नहीं थोड़ा टाइम लगता है उधर तक यूट्यूब में आवाज आने के लिए माइक टेस्ट 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 आ गया हाँ हाँ तो अगर नहीं अरे अभी कैसा मैं जाऊँ ना माइक लगाने को तो सांड के लिए जाणे मेरा काम थोडी आहे हो जाईल अरे नाही खरंच येत नव्हता कारण की ते हेडफोनच्या जॅकमध्ये तू आत्ता लावला ना म्हणून त्याला ऍक्च्युली पॅरल कनेक्शन पाहिजे हा
इकडेच टाक
येतोय ना आता
हॅलो हॅलो माइक चेक येतोय आवाज तो तिकडे आता तिथंच आहे ना आपलं आता ती सेटिंग नको हॅलो
आता ऍक्च्युली तिकडं पण ते फोनलाच टाकून
कुठं हां 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 ते पण बघ
हा ते पण बघ एका वेळेस दोन
एवढं जवळ नको घेऊ एवढं जवळ नको घेऊ
हॅलो
दोन आवाज आ रहे जावेद
हॅलो येतोय ओके चालेल बरं झालं चालेल
दोन आले
इथं इथं
हॅलो
हॅलो
पॅरलल कनेक्शन
येतोय
ओके दोन्ही आवाज येतील हां दोन्हीकडनं येत हा तू लांब जा आणि एकदम चिटकून नको तोंडाला
हॅलो हॅलो हॅलो
नाही नाही ठीक आहे
हॅलो
हॅलो माइक चेक
हॅलो हॅलो येतोय आवाज
येतो ना
त्यांना
अरे अरे आपल्याला नाही हां हां
हॅलो हॅलो
हां तिथं वरती लावा
हॅलो
कॉलर माईक येतो
यांनी काय
ब्लाऊज माईक नाही कॉलर माईक
नाही मग मला वाटलं कॉलरला लाव हॅलो हॅलो येतो आवाज ओके हां हां कॉलरला लावायला त्या लेडीज आलो कसं
चालू करून बघा एकदम. आता एकदा बघा. बंद केलंय, बंद केलंय. येतोय? मी बंद केलाय, बंद केलाय. हो सर, त्या आयडीची पिन काढली की आवाज बंद होतो.
येतोय आवाज? मी काहीच केलं नाही आतमध्ये.
हॅलो 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 माइक टेस्ट माइक टेस्ट माइक टेस्ट माइक टेस्ट तो आने को टाइम लगता है थोड़ा 5 सेकंड माइक अरे माइक हॅलो 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 त्याचा वाढव हाँ ये ऐसा आवाज़ आना चाहिए चांगला आवाज़ येतो क्लियर येतो ना हॅलो हॅलो एक मिनट हॅलो हॅलो हां हॅलो हां ओके हॅलो 21 नंबर संजू याचा हॅलो माईक टेस्ट हो एवढा हातातून धरून बोललं ना तर आवाज एकदम क्लिअर जायला पाहिजे त्यांना हॅलो हां ओके 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 थोडंसं कमी थोडंसं कमी थोडंसं कमी बस माईक टेस्ट माईक टेस्ट माईक टेस्ट माईक टेस्ट नहीं दोनों सेम आया अभी अंदर से कम किया था एक दोनों को अभी दोनों देख दोनों सेम आ रहा है हेलो हेलो माइक टेस्ट माइक टेस्ट माइक टेस्ट माइक टेस्ट माइक टेस्ट माइक माइक टेस्ट माइक टेस्ट ओके ओके
So good afternoon, everyone, and hope you have had a good lunch so that we have enough of energy to listen to the talks in the afternoon. Welcome to this afternoon session, and I welcome Professor Shantanu Bhattacharya to kindly chair this session. He is from IISc Bangalore and a former director of IICS Kolkata, and recently, very recently, he's been appointed as the new director of ISR Tirupati. Professor Bhattacharya, please.
Thank you, Manisha, for your kind words. So, we are celebrating Ganesh's birthday, but I don't see him here. Anyway, I think we have to go ahead with our academic lecture. And first speaker is Professor Pradeep Kumar, the professor of the Department of Chemistry of IIT Bombay. You all know he works on innovative way of making different type of RNA analogs, and some of them have therapeutic utility in the form of microRNA or siRNA and so on and so forth. And now I invite Pradeep Kumar to deliver his talk.
Thank you, Professor Bhattacharya. Am I audible? First of all, I'm deeply humbled by this invitation. I thank the organizers, Professor Natu, Manisha, Ramana, for inviting me to this important meeting, celebrating the birthday of Professor K.N. Ganesh and also celebrating the nucleic acid chemistry. So, first of all, I want to, maybe the light can be lit. So, I want to wish Professor Ganesh very happy birthday. He's not here. So, yeah, so this is a photo. You can see Mano is there, Professor Ganesh, and this was taken in Pacifica meeting in 2015 in Honolulu. So this is Dr. Mohan. He is he used to work for ISIS Pharmaceutical. He's from IIT Kanpur actually. So, so one of the important thing I noticed about Professor Ganesh is his passion for research and science, listening actually. So in every meeting, wherever we go, we attended many meetings together. So he sits through all the lectures and take notes and also ask very relevant questions actually. So he is a very sharp listener, and his observation skills are noteworthy. Apart from his other skills, everyone has pointed this out. So I wish him a very happy birthday. And before I go to today's topic, I want to briefly show some slides on the various activities we do at IIT Bombay centered around nucleic acids. So this is the area Professor Bhattacharya has mentioned, that we make nucleoside modifications, especially bifunctional nucleoside modifications, modifying the sugar and using them as therapeutic modification for siRNAs, and also recently we started using them for single gate RNAs for gene editing applications actually. So then we have a very, so in this direction, so we have reported some of the bifunctional modifications. You can see that there is a 2' fluoro and aminomethyl, and here 2' methoxy and aminomethyl. So depending upon the functional group, what you put at the 2' end, you can tune the conformation of the sugar, either as a DNA type sugar, if it is putting 2' methoxy, an RNA type sugar called C3', and if you put in fluoro modification. So they have multiple utilities, either in antisense, RNA therapeutics, etc. So we also work on extensively on quadruplexes. Jyoti had shown very nice applications in the morning of targeting quadruplexes using small molecule ligands. So here our interest is on discriminating the topologies of quadruplex structures. So for example, our interest has been to fine molecules which can bind to parallel topologies, preferentially bind to parallel topologies, and those molecules can't bind to anti-parallel or hybrid topologies formed by these telomeric sequences at the genome. So in this direction, we have reported a series of scaffolds. Some of them are listed here. So they have the discriminatory power to stabilize this parallel topology of promoter quadruplexes like CMYC, CKIT, etc. So, and today's topic is on damaged DNAs, and so which I'll be discussing in the next slide onwards. So we also work on catalytic nucleic acid, also called DNA enzymes. We try to find enzymes with DNA sequences which can do novel catalysis, and also DNA sequences or RNA sequences which can bind to targets called aptamers. So this, we use a method called in vitro selection or SELEX. So coming to this topic, today's topics, so I'm going to talk about polymerizers. So since the audience is slightly broad, so I'll just give some very basics about polymerizers. So this is, so we all know that DNA is double helical structure, and they are copied by DNA polymerase. And DNA polymerase, if you look at the structure of DNA polymerase is, the structure, three-dimensional structure is like our right hand. It has various domains. So it has got a thumb domain, finger domain, palm domain, and a proofreading domain called exonuclease domain. So, and these, when everything comes together, the template, the primer, incoming nucleotide, everything comes together, the hand closes or the enzyme closes, then the productive catalysis happens. So if you look at the reaction, so this is very important for the productive catalysis to happen. The base pairing has to be perfectly okay actually. So A pairs with T, G pairs with C, and if there is a mismatch base pair, the mismatch base clashes with the floor of the enzymes, that will affect the catalysis. And also we know that this is one of the most error-free process in biology. So, there is very few errors it makes, or various mechanisms by which errors can also be reduced. So the chemical reaction catalyzed by this enzyme is basically, so this is the template DNA, and there is a primer. It takes the 3' OH of the primer and attacks on the alpha phosphate of an incoming nucleotide and pyrophosphate leaves. The enzyme catalytic site is stabilized by 2 aspartate residues, which is coordinated with the magnesium ion actually. So, and it is like a molecular motor. So once one nucleotide is added, the enzyme move around the primer template complex, go to next, go to next, go to next, like that. But it is a very, very fast process. So coming to the DNA damages, we know that DNA is under attack by various exogenous and endogenous agents. So some of the agents are mentioned here. So the focus of today's lecture is about chemical exposure. So chemical damages DNA and what are the implications of that, and if these damages stays back, it can either lead to mutation. If it is mutation happening in genes associated to cancer, it can lead to cancer, and structural changes, basically it can also lead to cell death, which can lead to premature aging. So these are some of the chemically induced DNA damages. For example, this is a very simple DNA damage. So this is caused by ethanol, okay. Ethanol, excessive alcohol intake, when it goes to the body, so the ethanol will be oxidized to acetaldehyde. So once you have acetaldehyde, the guanine NH2 can react with acetaldehyde and form an imine, and then imine undergo a reductive amination reaction, form an amine. So the ethyl group can hang on to the DNA. That is a mutagenic DNA adduct. So that is one of the reasons people who take excess of alcohol get cancer, liver cancer. Okay, not the sole reason, it's one of the reasons. So these are other DNA damages caused by various chemicals. Benzyl bromide can form these adducts. Similarly, pyrene forms pyrenyl adducts. So the derivative of this is also found in cigarette smoke actually. So, and furfuryl alcohol form these kind of adducts, and this is an adduct which we have studied, so which is from a natural product, which is a dyeing agent that form this kind of adducts. So though we have DNA is under attack and so many adducts are formed, so most of these damages are taken care by various DNA repair mechanisms. So we have this is a textbook repair mechanism, nucleotide excision repair, base excision repair, etc., etc. So, but there are cases when at the time of DNA replication, when the DNA polymerase try to replicate, at the time of replication, if these damages happens, for example, chemical exposure happens, so those damages cannot be repaired by any of these mechanisms. So the system has two options. One is because the polymerase cannot read through these damages, polymerase has to shut down. So that will lead to cell death. That is the last option for the system to do. So what it does is that it will opt for other mechanisms, whether it can be tolerated, this damage can be tolerated so that it can be bypassed so that the replication fork can continue replication. So this is called DNA damage tolerance or translesion synthesis. Across the lesion, you are doing DNA synthesis. Here, the important thing is it's not a repair mechanism, it's a tolerance mechanism. So this is the most important slide for my lecture. So here I explain one of the mechanisms by which this happens. So this is a template where the damage is present. So this is the primer, this is the incoming nucleotide. This is the normal DNA replicative polymerase. So when this polymerase encounters this template, damage template, what happens is that it cannot read through by the active site clashes. So then there is a polymerase switching mechanism comes into play. So what happens is that this polymerase will be removed, a new low fidelity polymerase will come in, which has a wider active site. I will show you the structure, and that will come in, add few nucleotides so that the damage is tolerated. This process can be error free or error prone. Either it adds the correct nucleotide or it can add to the wrong nucleotide. Some of the cancers are associated with adding wrong nucleotide by this kind of polymerases. So then once this happens, the processivity of this enzyme is very low. There is again switch back to the normal polymerase, that replication fork is rescued. So this process is called translesion synthesis, and polymerase involved in this process called translesion polymerases. So these polymerases are present in all domains of life. So you can see that in eukaryotic and prokaryotic. So in eukaryotic systems like humans, two prominent polymerases are DNA polymerase kappa and DNA polymerase iota, polymerase eta. Kappa and eta are the two prominent polymerases, which I am going to discuss also. So coming to the structure of these replicative versus TLS polymerases, you can see that this is the normal replicative polymerases. There are couple of major differences. First of all, you can see that the active site, this is the active site where the reaction happens. This active site is wider. So this wider means it can accommodate the DNA damage. So, and another important thing is that the proofreading domain, the exonuclease domain present in the normal polymerase is completely absent in the TLS polymerase. So that means TLS can be error free or error prone. And also it has an additional domain called little finger domain or called pad domain. This helps to stabilize this minor groove adducts like what I have shown, the N2 guanine adducts. So coming to the research problem which we have addressed. So this is methyl eugenol. Methyl eugenol is this blue, this compound, methyl eugenol is this compound, and estradiol. These are two ingredients present in lot of natural products found in many plants, fruits, herbs, spices, etc. Okay? So, and they are known to form DNA adducts. What kind of DNA adducts they can form? Can form N2 adducts and also this is N6, this is a typo here. So N6 adenosine adducts actually. So this is a N6 adduct and N2 adducts both can be formed, and these are found to be reported to be carcinogenic. And because of this, this is a NPR news article which I picked. In 2018, FDA bans 7 synthetic food additives, which includes this methyl-eugenol, actually. You can see that methyl-eugenol is banned by FDA. And also they have been marked as possible human carcinogen by International Association of Research on Cancer, and reasonably anticipated to be a human carcinogen by National Toxicological Program. So this is worrisome because lot of spices, food items what we intake has these compounds actually, okay. So and how these adducts are formed. So these are metabolically activated. If you take methyl-eugenol, it will get hydroxylated by an enzyme and then sulfated, and then you can see that this sulfur group leaves and forms a stabilized carbocation that will react with the N2 of guanine, forms these N2 adducts. So the questions we asked, can these adducts be mutagenic in humans? And or you can frame a question in a different way, is translation synthesis. We know that these adducts are recognized by translation polymerases. Is translation synthesis across these adducts are error free or error prone? So if it is error prone, definitely it will be mutagenetic. So how to study this in the lab? So you need to get adduct bearing DNA. So these DNAs, the naturally occurring DNAs are in microgram quantities. You cannot isolate them. Of course, you can detect them with a high resolution mass spec, but to study you don't have that option. So you need to synthesize them. For that you have to make oligonucleotide bearing these adduct. And you need the polymerases. You have to express this polymerase and purify them. And once you have this, you can do some of the assays like primer extension studies, steady state kinetic studies, and also you can either use crystallography or molecular modeling and dynamics to look at the structural aspects. So to make these adducts, so we have used a simple straightforward chemistry. So we started with the aldehyde. Through a series of straightforward transformation, we made the corresponding amine, and then we do a Buchholz-Hartwig coupling with a protected 2-bromo-guanosine nucleoside and forms the adducted nucleoside. Then you deprotect these acetyl groups and then you do the dimethoxytritylation to form the corresponding DMD nucleoside, protected DMD nucleoside. Then you do phosphorylation to get phosphoramidite building block. These are the building blocks required to do solid phase DNA synthesis. So using these blocks, we have made this template DNA where X is present. Either X can be methyl-eugenol or estradiol. Both these adducts we have prepared. So then we went to my colleagues' lab. My student went to my colleagues in the bioscience department. Kiran Kondabagil is a virologist, where he expressed and purified this protein, polymerase kappa and polymerase eta. With the relatively good purity, we got both the proteins, and then we started doing the assay. So the assays are very simple. So you have a template where the adduct is present, then you have a labeled primer, it is a FAM labeled primer, and then you mix with the individual DNTPs. Okay, you want to check whether this polymerase causes error or not, error free or error prone. And then you add metal ions and you just run the gels actually. So what you can see here, so you will see only the DCTP get incorporated because the modification is on guanine and the corresponding complementary base is cytosine. So that's why you see that DCTP incorporation is happening, but no other incorporation is seen in the gels actually. So this shows that this polymerase kappa is faithful and it's replicating error-free manner across these adduct. So the same thing we have done with the polymerase eta is another less efficient polymerases, TLS polymerases. You can again see that polymerase eta also performs the error-free TLS synthesis. So then we looked at the kinetics, steady state kinetics. This is Michaelis-Menten kinetics. You can see that for in the case of polymerase kappa, so you can see the Vmax values for both these adducts, okay, the enzyme Vmax for these bypass across these adducts, where they are very similar, but the Km is different. But if you look at the polymerase eta, you'll see the difference both in Vmax and Km, depending upon 1-methoxy versus 2-methoxy. So that means 1-methoxy can have an effect. So if presence of an additional methoxy makes makes the enzyme less efficient. This you can see here in this kcat by km plots. You can see that in the case of EG adducts estradiol, you can see that it is bypassing more efficiently than an even unmodified template actually. So in the case of methyl-eugenol, it is still bypassing efficiently by Pol kappa, but it is less compared to estradiol. But in the case of polymerase eta, this is polymerase eta results. In the case of EG, which is similar with unmodified template, but in the case of MEG, the efficiency has come down dramatically. So this tells you that 1-methoxy versus 2-methoxy group present in these adducts has an influence on the catalytic efficiency of this enzyme. Nevertheless, though this process is error-free. So we also looked at the full length extension. These enzymes are not very efficient, but we can see that Pol kappa is able to produce the full length product actually. If it is XDG, this is the 27 mer, so basically it reads through and comes up to here. That is the 27 mer. And in the case of Pol kappa with both the adducts present in the template, you can see the full length product. But Pol eta is known to be less efficient. You see that the Pol eta is not able to read through this adducts and make the full length product. So it's less efficient. So then we look at the structural aspects. So this is the X-ray structure reported by our group in collaboration with Hong groups in Canada. So this is for a different leucidine adducts. So there is the Pol kappa structure is available with the N2 adducts present. This is a 2.9 angstrom structures. We took these structures, we had these structures. So we took this structure and we modified this DNA, the nucleotide at this position, and ran long MD simulations actually. So you can see that, so this is here you can see that the MEG adduct is present, and this adduct reoriented itself in such a way that incoming nucleotide can form strong base pairing with the template DNA actually. So also you need to know that for efficient catalysis by a polymerases, couple of parameters need to be fulfilled. One is called, one is basically the Watson-Crick base pairing, perfect Watson-Crick base pairing between the template DNA and the incoming nucleotide. And the reaction distance, distance is basically the O3 prime of the primer and the alpha, P alpha distance, that is the reaction distance, and the C1 prime distance between the two sugars, the first atom of the two sugars need to have a value around 10.8 angstrom, and the attack angle is O3 prime, P alpha, and this O beta position. So this angle should be in the 150 to 180 degree. So if these matches together, and then if the metal ion orientations are proper, and then the productive catalysis, the polymerase will be able to bypass. So we looked at these parameters. This is from the 500 nanosecond MD simulations. So this you can see that C1 prime distance plotted against the simulation times. You can see that these both the curves, the red is for the modified complex. This is a ternary complex. You have a primer, you have a template, you have an incoming nucleotide, and you have the whole protein around it. So you can see that they perfectly match, and there is a slight variation in the case of reaction distance and attack angle, but this is within the acceptable range for productive catalysis. That's why all the parameters are matching. The enzyme, the adduct is not distorting the structure of the enzyme so that it doesn't allow it to catalyze this process. So this you can see, I don't know whether it works. No, can you enter just? Yeah. So you can see that this active site is stabilized by both hydrophobic and some of the hydrophilic residues. For example, the serine 128 basically forms hydrogen bond with the oxygen of this adduct, and there is a proline, there is an arginine, and also there is a phenylalanine makes very critical contacts, hydrophobic contacts with this active site of this enzyme. So what you can say that these adducts, polymerase kappa and polymerase eta can bypass these adducts in error-free manner, H-pol kappa is more efficient in bypassing this adduct compared to H-pol eta. So what we are saying is that both EG and MEG cannot be carcinogenic in humans if TLS by these two enzymes. There are other TLS polymerases are present, so we have not studied them. If these two are used to bypass, then you will have error free and it cannot be mutagenic. That is the conclusion of this study. So how many minutes I have? Okay, I'll try to conclude. So we come to another enzyme called Primpol. So this is a newly discovered enzyme. 10 years ago, this enzyme has been discovered. So this is a DNA primase. Same enzyme has multiple functions. So it has a primase activity. It can synthesize primer, and it can also act as a translation synthesis polymerase. So the principal role is reinitiating DNA synthesis at the stalled replication fork because of adducts. And this structure, it's a partial structure reported by Anil Agarwal group from Mount Siani Medical Center. So this structure is available. So I have been contacted by Alina's group from Russian Academy of Sciences. They have this enzyme. So they wanted to study the TLS across these adducts we have synthesized. So we had a nice collaboration. So we have screened different adducts by whether these adducts can be bypassed by this human Primpol or not. That is one of the objectives. So you can see that it increases the steric bulkness of these adducts from going from ethyl to pyrenyl adducts. You can see here, this is where the marked area, you can see that only the ethyl adduct is bypassed by this enzyme. The second one is magnesium cofactor and another is manganese cofactor. Manganese shows bypass, but that is not real bypass actually. It is like a template switching mechanism which we have explained in the paper. It is not a real TLS activity. So what you can see only the TLS activity with ethyl group. So then we looked at the structure. So we took that crystal structure and modified with the adducts and ran dynamics. You can see that in the case of ethyl, the hydrogen bonds between incoming nucleotide and the template DNA is perfect. So, and when you increase the steric bulkness going from isobutyryl to benzyl, you see that the hydrogen bond efficiency goes down and benzyl is drastically affected. So then we looked at these parameters which I mentioned in the previous project. So the three parameters, one is reaction distance, another is the distance between the two sugars and attack angle. You can see that this black one is for the unmodified and red one is for N2 ethyl. And black and red try to overlay, and there is a huge difference between the green one, green curve and also the blue curve. So they are like going away. So that means this clearly shows that the enzyme cannot accommodate this adduct perfectly in the active site. That is the reason why this human Primpol is not able to do TLS activity across these bulky adducts. So we also looked at the surrounding amino acids residues. So looked at some key distances R288 which stabilizes these hydrogen bond interactions. You can see that again you will see that the black and red line matches the population. This is basically the population matches, but that is not the case with the other adducts. The same with the total number of hydrogen bonds around the active site. So there are roughly on average 50 hydrogen bonds around the active site, but there are only few hydrogen bonds for the other two adducts. So the active site is severely disturbed by this bulky modification which we have seen. Also experimentally they are not able to, Primpol is not able to bypass these adducts. So to summarize this part, so it is a human Primpol can only bypass N2 ethyl, less bulky N2 ethyl adduct. So successful replication condition not satisfied by the bulky adducts, and there is a protein network get affected, and recently we have published this work. So this I will skip. So finally I would like to thank people who have done this work. So the students who were involved in both the projects are highlighted. So the most of the work I showed in the first part done by Priyanka Deshmukh. She is going to defend our friend Govindaraju is going to be our examiner. So and all the MD simulation is done by Shruti and helped by Vipin. And also my collaborators, initially I collaborated with Dr. Deepak Nayar's group to get the first crystal structure, and other collaborators and all funding agencies. Thank you for giving me an opportunity to talk here.
Thank you, Pradeep Kumar, for very interesting lecture. Let us now make the floor open. Any questions, queries? People are just having post-lunch inactivity.
I will ask a very simple chemistry question. Yeah, sure. For your N2 modification, you bring the bromo first. That by diazotization, or how do you install the bromo?
Yeah, that is by diazotization.
Okay.
Yeah.
And then you do this. Have you tried that directly displacement of fluoro?
Fluoro, yes. We tried. It was in our hand. It worked for small adducts. Ethyl and all you can make. So when we increased the bulkiness, it was not working. Yeah. So a lot of people use that actually. So we spend a lot of time. That is a post-synthetic modification. Yeah. So we tried actually. Yeah. Thank you.
Yeah. Yeah. No, you first do that.
Okay.
Sure.
Yeah. So you just very small question. So you talked about one primase here. You said that primase makes primers. So I'm just wondering how the primase makes it. It's like an RNA polymerase or DNA polymerase?
It's a human prim code can synthesize primer. So it's like a primase. We have the primase, no?
But it has to be, it is DNA polymerase or RNA? Because DNA polymerase cannot start ab initio from
Yeah. It can synthesize both RNA and DNA actually. Primer. So it has many functions.
Okay.
Yeah.
Okay. Because usually we know that the polymerase basically.
Yeah, right. So you need a primer. RNA primer need to be made.
It extends the
polymerase
only extends.
Yeah.
Yeah.
Hello, Pradeep. Very nice talk and very beautiful works actually. So when it's your dimethoxy, this olefin is there. Did you observe any time is a fluorescence, anything kind of?
We not checked actually. So it is. So we not checked the emission of the oligos actually.
Okay.
So that's I should say actually. Could be, but I don't know actually.
Yeah, just I was thinking if in nitrogen lone pair and then your conjugation will be there. So I don't know.
So there is a possibility.
And second question was there, when you synthesize this DNA primer, so that our dimethoxy was still intact after purification?
Yeah.
This is stable.
Yeah. So we have all characterization data.
Okay.
Yeah.
Okay. Yeah.
Well, I have one query. You have mentioned methyluginol as a potential carcinogen. This is a pheromone. And this pheromone is used to, disrupt the sex of pests, which are fruit flies. So it's a big market. So, but you have actually used these to covalently make the adduct and study this. But I wonder whether the critical concentration of molecules that are present in the vapor will have any effect, which are not covalent, but whether it can eventually make some kind of.
So it has been isolated from the liver cells.
Okay.
Yeah. So that's why these people were scary and they went to FDA and complained. Activists went and filed complaint to FDA. They should not be used actually. But of course, it's the concentration matters at the end.
Yeah. It can be used very heavily in agriculture, but that's a completely different concept.
Right. It's used as herbicide actually.
All right. Not herbicides, but this essentially the female species of Bactrocera and male species, their mating it disrupts. That's all it does. And it can do wonders.
Yeah, eventually it happens.
No, I think you need only a few molecules. They are very low concentration. Okay. If there are no further questions, I think let's thank Professor Pradeep Kumar for a very beautiful lecture. And I think don't disappear. I think this organizers have.
have the next man who is collaborating with Pradeep Kumar with the student's thesis you just heard. And Govindaraju is another stalwart, of this gang of Ganesh. And of course, all of you know that he is in New Chemistry Unit and chair of the Educational Technology Unit at Jain CSR. And he has, of course, moved a little bit away from classical nucleic acid chemistry, and now he is more interested with old people, Alzheimer and amyloidosis and all these related disorders. Let's hear from Govindaraju. Floor is yours. Time starts counting.
I can't argue on that.
Pointer.
नीचे से It's okay, otherwise it's fine. No, this side. ठीक है, otherwise it's fine. I think it will come. Okay. ठीक है, I think it will. Thank you very much, sir, for that kind introduction. Let me, at the outset, let me thank Dr. Nathu, Dr. Manisha, Dr. Ramana, and everybody behind this special event. First of all, for organizing this event and also inviting me for this special event. Thank you very much. When I got a call from Nathu sir asked me specifically to talk about Alzheimer's disease. So that's how the title. So that's how the talk is. But having said that, before I move on to talk, so of course I'm truly, since my guru is not here, I can tell you my true feelings that I'm truly emotional for the event, for the day, because we are celebrating our guru and in our alma mater. That is something NCL. NCL, it's very close to our heart, so I say this. So thank you. Let me dedicate this talk to Professor Ganesh, Professor K.N. Ganesh, on his 70th birthday, and also for his, every achievement and contribution to Indian science. Of course, we are all part of that journey. The theme of this today's event is DNA advances in DNA RNA therapeutics, and I'm going to talk about Alzheimer's disease. Then I decided, let me make a bridge course, small. Usually we all know teaching is very important. When we do take a course, advanced courses, we do some, like advanced courses. You see Professor Shantanu Bhattacharya and Professor Javed Iqbal, they all know. So we give a bridge courses to take along, and I'm not telling that way here, but just to bridge that switch over to my talk. So let me take probably a minute or so that to tell you something about DNA, so my link, what we are doing very briefly, and then I'll move on to actual talk. So that is, so far we talked about DNA RNA therapeutics. So let me quickly touch upon DNA RNA diagnostic that we have done recently, of course, because of COVID pandemic. Everybody knows, we talk about in this gathering, I don't have to talk about too much about DNA. Nevertheless, the DNA can form a duplex. Usually you can call that as a canonical form. When it forms other structure like 3-stranded structure, 4-stranded structure, and their variations, we call them non-canonical DNA structures. And targeting these
either canonical or non-canonical DNA structures. So there is a myriad of opportunities. Meaning he can actually do lot of biophysics, bioimaging, diagnostic, therapeutics, and even theranostics. So there is a huge opportunity here, particularly with respect to health, and if you are interested in health and well-being, even for materials related research. So that is something, the opportunity with DNA and RNA. And as we all know, all of us went through difficult times, last few years, and every one of us want to do something, for this COVID-19, whether it is the isolation ward or, how to set up, isolating a patient, or thinking about, how to, develop a diagnostic or a therapeutic, management, in whatever, in many different ways, every one of us, particularly in CSIR labs and all the higher, universities, IITs, ISR, everybody was trying to do something. And we also decided, let us do something for that. And it is actually not in the first wave, toward the end of first wave, we thought let us, the gold standard for detection of COVID-19 is RT-PCR. Of course, there are a few other things, other detection methods as well available. We wanted to develop a technique which is very simple and cost effective, probably much simpler than RT-PCR. What can be? So what we did was we did some bioinformatic analysis, and that is the, you can see the genome, SARS-CoV-2 genome, and we realized that there are sequences in this COVID, RNA, this is RNA virus, in the RNA sequence genome. So there are sequences which have, which can form a G quadruplexes. Unfortunately, they are not stable enough, because the sequences which you see, only you can see diodes, okay, not more than that. And in fact, we have developed a technique here. So the ways to stabilize these G quadruplex structures, and we also developed a novel small molecules, fluorescent small molecules, switch-on fluorescent small molecules. Just take the samples and then amplify and then put this small molecule, you can make the readout. And that is precisely what is called GQ targeted reliable conformational polymorphism, which is a very simple alternative detection technique for COVID-19. Well, this was wonderful. We have demonstrated this in real samples. Unfortunately, by the time we did this, we are already kind of passing third wave. So not much of use for this technique as far as SARS-CoV-2 is considered, or COVID-19 is considered. However, this is a very modular platform. That is advantage. So now we can extend the same technique for any DNA RNA based pathogen for detection, and we also believe one can also develop a therapeutics using this target. So here we are targeting the positive strand, and of course next we realize that targeting a complementary, these unusual the DNA sequences in the complementary DNA strand is also very viable for the technique. And very recently we have demonstrated that indeed we can also do that, targeting these unusual DNA structure, unusual nucleic acid structures from the complementary DNA as well. And in fact, so this has become a very important project which came out of COVID-19. We never applied for a project, but now in fact we have 2 BIRAC projects so running, and possibly some diagnostic kit potential for a field utilization will come out very soon. Not for COVID-19, but few other diseases. So that's where that is a bridge. Now I will switch over to the actual topic of today, that is Alzheimer disease. Let me just go talk about little bit of basic. We all know the proteins are synthesized as polypeptides on ribosomes, and these polypeptides are subjected to protein folding so that they acquire 3-dimensional structure and become functional. But sometimes these proteins, these functional proteins are their processed product. When I say processed product, means peptides, undergo misfolding, and obviously they become toxic. Well, cells have a repair mechanism. Either they can put back this protein into a folding, or can be pushed for a degradation through proteasomal degradation, or for instance for even autophagy and so on and so forth. But under some adverse physiological conditions, these proteins escape this both proofreading or a refolding or a repairing mechanism and degradation and become amyloidogenic, meaning they undergo ordered aggregation to form a highly thermodynamically stable products that we called amyloid aggregates. And these amyloid aggregates in the brain are actually responsible for at least more than 25 different neurodegenerative diseases. And in fact, Alzheimer is one such disease, in fact a major disease, as you would possibly I can tell you that this is almost more than 60 to 80% of all dementia cases are linked to Alzheimer disease. Unfortunately, although the disease, when I talked about protein aggregates, as you can see here, there are two kinds of protein aggregates. I'll come to that in a while. One is called FLAX, that is from amyloid beta peptide, and tangles, that is from a tau protein. I will allude to that. I'll come to that in a while. Although the disease has been identified more than 100 years ago, of course, by the doctor named Alois Alzheimer in Germany while treating the patient Augustus Dieter. She was actually in her 50s. It was more than 100 years ago. Still, there are no fully approved detection or a treatment for this particular disease. And if you talk about any other major diseases, so-called HIV, stroke, and all that, so the number of cases, deaths have coming down. Meaning, I'm not telling the disease is not coming down, but biomedical interventions are available. But in case of Alzheimer disease, the number of cases have risen. Actually, this is a world data of 2013. Now they're risen by almost more than 100% increase in Alzheimer disease cases. Therefore, we need to do, this is going to impact public health and economy, so we need to do something for this, not just, and that's also, for example, India. So we are doing this research, there should be Indian relevance as well. So earlier, 10 or 20 years ago, in fact, when I started my career, some of my contemporary peers indeed asked this question. And they asked me, what are you working on? When I said Alzheimer disease, why are you working on Alzheimer disease? It's a Western disease. But now nobody asks this question because everybody is aware. Even in India, as you can see here, but particularly in Asia and India, the number of cases are going up very rapidly. And of course, the reason and all, it's not time to discuss, but definitely is going to be a major health problem. So this is the bigger picture. So that is our so we are taking a kind of a holistic approach in dealing with Alzheimer disease. So that is our plan. So we are trying to understand disease mechanism. We are trying to develop diagnostic. We are trying to develop therapeutics, and of course that also will have a theranostic implications. In fact, we have been quite successful in developing molecular tools or a platform for detection, which is not I will not be talking exclusively today because of the time limitation, but I do talk about therapeutic aspects today, some of the aspects, and little bit touch upon very few mechanistic aspect, but I'm going to miss out on many of the major the research outcomes that is right now happening in our lab in terms of understanding disease mechanism. So, well, we know certain things, but it is also enigma, the complexity, as my title itself was alluding to it. It's a it's a very complex disease, so still many things are known, but we do know so far there are many other things. We do know many things, but there are also many other facets of this disease which are unknown. So precisely that's what we are also doing, working on. Many of them, in fact, either unpublished or even communicated. Probably I can talk about this aspect on some other platform. So this is the complexity of Alzheimer's disease. So looking at this slide, you may say that this guy is not professional at all, in preparing the slide. But believe me, this is how the Alzheimer's disease is. So it's so complex. In fact, not everything is included here. I talked about protein aggregation. There is an extracellular amyloid beta which is generated from APP, which was alluded to in this morning, and there is a tau, which is a microtubule binding protein, which collapse and also form aggregation. And in addition to that, there are metals, which it forms an inclusion complex with these protein aggregates, and then become a factory for generating reactive oxygen species. There is oxidative stress, mitochondrial damage, membrane toxicity, and microglia activation and neuroinflammation. Neuroinflammation is again very, very important. It was important in COVID-19 as well. So here also it's very, very important, and just to name a few. So there are many other facets here which are not included, but just to give you an idea that indeed Alzheimer is a very involved complex, the disease mechanisms. And if you really want to know more about this Alzheimer's disease, possibly you may refer to this a comprehensive book which covers, if not all, at least most of the aspects of Alzheimer's disease. Now, coming to this toxicity of these proteins, I talked about protein aggregation in the brain and also becoming toxic. How can we see that? Can we see this toxicity, how this toxicity is arising, how the toxicity arises? Well, we can do a simple experiment. So, for example, cell viability, all of us know, how to measure a cell viability, meaning to look at the cell and see whether they are happy or not happy. If they are happy, how much they are happy, whether 100%, 80%. So, can we do that? Yes. For example, what you see here, these are the cells. You have to trust me, they are healthy cells. Okay? We have a very nice setup, bio-AFM, confocal integrated with AFM, and that is a picture taken from our lab. This is a neuronal cells. You have to trust me, it's healthy. Okay? Now you incubate these cells with amyloid beta, which undergo aggregation and interact with the membrane, and you can see that there is a unhealthy thing is it's coming in, meaning there is a formation of stress fibers. Well, stress fibers formation is not uncommon. It does form, but it is a dynamic and reversible. But here, the stress fiber formation is so extensive, though it won't be, it will not revert back, rather it will lead to neurodegeneration. Meaning, so the cellular loss, neuronal loss, and that leads to disruption in neuronal circuit or network, or even what you call wiring, neuronal wiring and all, and that subsequently results in the so-called cognitive decline, deficiency in learning, memory, and all that. So what we have to do is, we have to identify this condition and then develop as tools, molecular tools, and see whether we can reverse that state to this state. That's what we have to do, both the detection and also rescuing cells from that state to this state. And the other aspect is reactive oxygen species. See, these are all tiny species. Probably these are the tiniest molecular species which we can come across, but they are very important physiologically. But trust me, you can call them friends in a healthy conditions, but if they go behind a threshold condition, they become a dreaded villain, which we never seen in a movie, we can never even imagine. So toxic, so impurious these ROS can be. So just to quickly, oxygen, so it goes to water through 4-electron system, and intermediate is hydrogen peroxide. And hydrogen peroxide is converted into hypochlorous acid, and in the presence of myeloperoxidase, and this is very important. It has a life-saving effect. For example, if you talk about immunity, again, I don't know, I'm coming back to, again and again, COVID-19. Everybody was talking about, immunity, improving immunity and all, right? So in fact, this particular species plays a very important role. I don't know if some of you might remember, there was a controversy involving Trump, bleach controversy. I don't know how many of you remember. So he suddenly said that, in the early stage of coronavirus, even the bleach can be useful in dealing with coronavirus. But everybody criticized him. Let me tell you, he was not wrong completely. It's just that his advisor possibly didn't, probably explain him properly to put it in a right way. This hypochlorous acid, if it produces in the neutrophils or a phagosome, it is very good, it kills viruses and bacteria. But if it is produces outside of these neutrophils or phagosome, or if you have to take externally, it's very, very toxic. When I say, when he was referring to bleach is actually hypochlorous acid, sodium or a calcium salt of hypochlorous acid. But just because it's, internally produced and kills virus, you cannot drink it. So that was a mistake. So it indeed has a life-saving effect. However, under Alzheimer condition, there is an overexpression of myeloperoxidase, and that leads to production of HOCl, and there is a loop mechanism, and become everything become a toxic in the cell, and that is leading to life-threatening effects, causing oxidative stress, inflammation, biomolecular damage. We also saw in the Pradeep's talk, and DNA, RNA, lipid, biomolecular damage that aggravates Alzheimer condition, that is protein aggregation, which I mentioned earlier. Well, how do we see that? Indeed, in our lab, we developed molecular tools we have established a double transgenic mice model, very unique mice model, Alzheimer phenotypic mice model in our lab at JNCSR, very unique facility we have, and that is a brain. We have also developed a molecular tools which are very, very specific. In fact, rarely we come across small molecules which are as good as antibody in terms of selectivity and sensitivity. Here I can proudly say we have a very small molecule, 400 Dalton molecular weight molecule, which exhibits selectivity and sensitivity as good as antibody. So what you see is that is the protein aggregates in the mice brain, AD phenotypic mice brain, and same thing you can see in the brain tissue from a human. It of course is a postpartum tissue. This is actually from a Alzheimer disease patient. What you see is that is so that is amyloid aggregates, and this is tau tangles, and this is in fact this is the technology. I'm not going to in detail. This is a platform which not only detect Alzheimer but also differentiate Alzheimer from other neurodegenerative diseases. There is something very important. If somebody has a dementia, more often it can be a mixed dementia, meaning Alzheimer can coexist with Parkinson's disease or few other diseases. That makes the situation even more worse. Unless you know what is what, how can you even think about treatment? So here we have a platform which is not just detection, it can also differentiate from other neurodegenerative diseases. And of course, we have developed several such platforms, and I'm not going to I'm skipping a lot of it. And this image, particularly what you see on the right side, it is not just an image, but it is an image actually show you the complexity of the diseases, which I showed you one complex slide. Precise, that is what it is actually. So using a few molecular probes and more than one molecular probes and more than one and targeting more than one biomarker, we could actually image that multifaceted or a complex disease in the mice brain. Yeah, probably I'll skip it. So with that, I hope I gave a quick introduction into the complexity of the Alzheimer's disease. Of course, many other things I'm not covering today here because still the ongoing or unpublished work. Probably I can talk about later on completely new mechanism. Now I will come to therapeutic aspects. Coming to Alzheimer's treatment, as I already mentioned, there are no fully approved drugs for Alzheimer's disease. But nevertheless, in terms of whatever available or under development can be classified into two classes. One is small molecules, other one is of course biologics. There are some drugs available in the market under small molecules category. Of course, I am hesitant to call them drugs, but nevertheless, they are these acetylcholine esterase or a NMDA antagonist molecules. All they can do is they can modulate the levels of brain chemicals for a very, very short time. So it's an indirect way. And last year, there was a big news. There are two biologics, particularly antibody-based drugs were approved by FDA, two of them. And maybe Dr. Manos may recall this, and some of you will also know probably the most controversial drugs ever approved by FDA so far, because these particular biologics or antibodies, indeed they kind of dissolves the protein aggregates, but that is not translating into gain in cognitive functions. But however, possibly we can also have to look into a positive side. Since there are no drugs for Alzheimer's disease, possibly FDA went ahead and then approved it so that probably more and more players come into picture and then new effective drugs can come into effect. So having said that, so I can safely say that there are no fully approved drugs that target core pathology of Alzheimer's disease as of now. So now the question is, why so? See, I mean, there are many research groups, both in academia and industry. There is a lot of effort, but why there are no success so far? In my opinion, both in academia and industry, we are targeting individual pathways. But unfortunately, Alzheimer's disease is not a linear disease, rather a complex disease, as hopefully I have convinced you. So targeting individual disease mechanism or a target will possibly, we may not be going to be successful. And if at all, if you have to be successful, we may have to go for multifunctional modulators which target multiple etiologies of Alzheimer's disease. Yeah, so of course, in the beginning, that is the peptide I talked about, amyloid beta 42. and that is a core recognition motif. In fact, there are several groups, even material scientists, you play around this particular peptide and thought of developing its own medicine. So for example, you take a fragment of this peptide and modify it or as such or modify, and then it should go and then clump it and then prevent aggregation. Yes, there was success, but it is not successful enough to think about taking them for therapeutics. So just to understand, so at some point we also developed a peptide mimetic. As you can see that is a fragment, and we inserted this cyclic dipeptide. This is another major area that we are working on in our lab. So I can give a separate, we can talk on that. But nevertheless, I introduced at a predetermined position, and we have made a library of these small, not library, small set of peptidomimetics. Out of them, one of these particular mimic indeed prevents aggregations. What you see is a test tube experiment. See, that is the protein aggregates, and if you add this, it prevents aggregation. And what you see is an immunohistochemistry study, and it prevents actually whatever you see that red, it is interacting with the membrane. These aggregates are interacting with the membrane. Then there is associated toxicity, but this particular mimic inhibit this protein aggregation and protect cells. And what is more important, when we talk about toxicity to the cells, what we mostly look at is physiological functions when it comes to cytotoxicity, whatever. But I also would like to mention here that it is not just physiological functions, even mechanochemically, mechanobiology, also very, very, very important. And just looking at these mechanical aspects, you can actually make out whether the cells are happy or sad. So for example here, so you see that's what I told you. You use a very interesting technique, post-peak QNM, quantitative nanomechanic AFM. That is we have a very unique advanced AFM technique machine, and so that is healthy cells. And when you interact with A beta, and you can see that their size decreases drastically, become rigid. Okay, and then stiffness, normally the cells will have around 29 kilopascal stiffness, that increases to 400 because of the rigidity. So that is clearly tells us that these cells are not happy. So there is something is happening, there is a collapse of cytoskeleton. And of course, one can also look into adhesive properties also. So this is something very important, probably not necessarily fully therapeutic in angle, but there is a lot of things one can understand in the cells under this kind of toxicity. Okay, so now early on we worked on several small molecules, just like targeting individual pathways. It's not just all of a sudden it occurred to us that Alzheimer is complex, we have to multifunctional. No, we started, I'm not presenting most of it. We started looking at individual targets, and one after the other, including autophagy. So we did targeted autophagy. There was success, but we realized at some point that targeting individual routes will not be very useful. So at that time, we wanted to develop some multifunctional molecules, and we come across, for example, this peptide. Very simple, GHK. This is a tripeptide which is there in all of us. In fact, I can call it as a natural product from humans, and also I can say it as a Sanjeevani. Believe me, this tripeptide does wonders in our body. It is antioxidant, anti-inflammatory, regulates genes, wound healing, you name it, it does. So many functions it does, and it does all these functions by complexing with copper. Okay, so this was very interesting. And then also another aspect, I think I'm running out of time. As we age, the levels comes down. So there is also link to aging and all. Okay, so we decided to make a conjugate of this with another hybrid peptide that we have developed for earlier work. And this particular conjugate indeed become multifunctional inhibitor. However, there were two problems. It works as an inhibitor at a 100 micromolar concentration. It's not therapeutically viable to take it forward. And then also it is a multifunctional only in presence of copper. But let me tell you, iron is also plays a very important role in case of Alzheimer's disease. To make it much more true multifunctional inhibitor, of course, ours is an organic chemistry group. So we did, modified the structure of this GHK to develop this peptidomimetics. We can call it as peptidomimetics, or one can also call it as small molecules. And indeed, you can see that they complexes with copper and also complexes with iron. And one particular molecule, this particular molecule, I'm not going to to the details. Sir, how much time I have? 2 minutes, maybe 1 more minute extra for me. Okay, it's a multifunctional, so I'll skip it. And we have also showed it is very effective in NRF2 pathway. So I'm skipping a lot of the data here, but and this is another concept that we have developed, hybrid multifunctional inhibitor. Probably this is one step ahead of this drug repurposing. So to develop a specific drugs, by utilizing the knowledge from the failed molecules or a natural product. This is another concept that we have introduced. Again, paucity of time, I will skip that. Here is another. And this is a natural product. We say from a berberine we have modified and made it a multifunctional molecule. Indeed, it protects cells, as you can see from the apoptosis marker. By now, we are able to, so far we have talked about small molecule, natural molecules, peptides and their analogs, and we learned a lot from this work, and we are ready to design our own synthetic molecules. And this is one such synthetic molecules we developed. Indeed, works as a very beautifully and a very interesting bio-AFM data. This is very, very interesting. I can talk to you later on if you are interested. And this is a combinatorial therapy. See, there is a combination coming, developing a synthetic chaperone to protect the proteins from crumpling down and aggregation. And also lithium is a well-known. Lithium is known for used for depression and so on and so forth, and also being considered for Alzheimer. So we have developed this particular synthetic chaperone. Will be very effective for combinatorial therapy. The other one is liquid-liquid phase separation. This is another concept, very, very important, emerging, and particularly with respect to tau, we have developed these particular synthetic molecules and which can effectively modulate this protein understanding as well as modulating this particular. So last molecule, this is the one molecules which is purely by design, completely a synthetic molecule. We have designed a set of molecules and screened them, and it was found to be very effective in the in vitro experiments, and of course in cells as well. And with that encouraging results, we went ahead and tested in our mice model. So as I told you, this is kind of almost 10 to 11 years of work. And what you see on the right side, so that is healthy brain mice, and the middle one, that is Alzheimer mice. You see a lot of aggregates. Believe me, Alzheimer patient, human patient brain looks exactly like this. What you see, the red thing, that is the the protein aggregates. And now if you treat these mice with our molecule, that is TGR63, there is a significant decrease in the amyloid burden, or a reduction in the amyloid burden by 75 to 85%. But that was very, very encouraging. But if you remember, I mentioned about biologic antibodies. So they also may not be this extent, they also clear aggregates, but they are not successful in providing, giving the, helping gaining cognitive functions. So just clearing protein aggregates is not good enough. So we went ahead and we did a standard neuro behavioral experiments, at least 3 different. These are the very, very tough experiment, open field novel object and also maze experiment. I'll give you, sir, a 40-second video, if you permit me. So I will not go into the detail. I will show you a small video. This is a behavioral, experimental video. Hopefully that is very convincing. Okay, perfect. So what we do, this is called Morris water maze experiment. So we take this is a water bath or a water tub, and you can, hypothetically you can divide that into a 4 quadrant. And in one of the quadrant, we place a small platform which is hidden just under the surface of the water. Okay, so it is not visible, but at least it's also not something which if cognitive functions are normal, one can see it. So now what we do, we take these animal models, both wild type and Alzheimer, treated, untreated, and then we do, we leave them, to swim around, and we do a test around, training, and leave them in the water bath, and then swim around, and then finally they will also, kind of try to find some platform to rest, okay, and see what happens, whether actually they can find a platform. That is learning. And next what we do is repeat the experiment and remove this platform and see whether these mice remember the location of this platform, that is memory. So we are testing learning and memory. Yeah, you see that these are wild type, treated, untreated, and AD mice, treated, untreated, and see this. See, they have already found the platform, meaning the drug also has no negative effect. And this is the AD case. See, the treated mice could actually, that guy could find the platform. But this person, this guy, actually will be roaming around. This mice has no clue, anything. It is aggressive, okay? It's anxiety, and it will keep on, this is the quantitative data. Now we remove that platform and just see whether how many times these mice comes to this location. See, these guys comes several times, meaning they remember there was a platform. Although it is not there, but they remember. And in this case as well. But again, this guy has no clue, absolutely no clue. See? So I hope, so I'm almost done. I hope seeing is believing. So it is indeed, in the mice AD phenotypic mice, we have shown that our compound not only modulate these protein aggregates, but also improve learning and deficiency in learning and memory. So, and this particular compound, so in fact, it's a very unique compound, and we have done toxicity and many other studies, and it's been licensed to a US biopharma, and now they will be taking it forward for clinical trials. In fact, they are doing advanced toxicity studies already. With that, I hope I convinced you the complexity of Alzheimer's disease, and then of course we need to go for a reinvent ourselves to develop multifunctional molecules to target these multiple toxicities. And of course, this particular drug, as I told you, the drug discovery takes almost 12 to 15 years, and this is where we have reached. It took almost 10 to 11 years, maybe close to 12 years, and we are just cross fingers. Hopefully, you can be successful. Of course, you know how difficult it is. With that, I would like to thank my students, funding agency, and on the collaborations and everything. And thank you for your kind attention. And again, sir was not there. I would like to wish you a very happy birthday. And you're going to be with us and then guide us, in every aspect, science, education, policy. And thank you very much, sir. And thank you, sir. My apology that I took some time extra.
Thank you, Govindaraju, for a fascinating talk. I think floor is open for a couple of questions.
Hello.
Yeah.
Hello. Hello.
Those kind of approaches where you're actually trying to use hydroperoxide-based lipids in the presence of iron, which is the trend now. Secondly, actually, I have done some work with my colleague where we have actually directly gone to the patient's Alzheimer and use those samples to use NGS to look into upregulation, downregulation of genes. I think from here, what to possibly do, you see, your double knock transgenic mice obviously is based on the fact that you do not have the so-called protein, right? Which gets into beta amyloid formation or tau protein. I think the best thing would be, it's a very nice, I'm not trying to sort of criticize this, but I think it might help you to really reach out to doctors. And if you don't have here, I can connect you to some doctors in Hyderabad where they can give you the samples. And actually you can use some of your small molecules to look into this, whether they alter. There are a lot of these genes which actually you can see using NGS, which can be done, So single molecule, I don't know really, but I think first of all, the peptide that you have, how does that help in terms of, let's say, refolding the protein properly? Because that's what the case is.
Yes.
But does the large peptide that you showed, does it really do that, is the question.
Yes, sir. So thank you very much for all our important questions. I will answer the last one. The peptide part, they're actually not is more of an understanding. So we were also understanding, and in fact, I don't think I will take it forward. So they are only understanding purpose. They're not for any, I'm not even thinking about advanced studies in the animal model, not let alone the clinical trials or anything. So they are more of understanding, but we have learned a lot from that. And coming to small molecules, okay, the peroptosis, in fact, I have a 5-slide where he just communicated the work, and we have very, very interesting work. And since it was also live everywhere, Manisha called me whether it's okay to which one, at that I have not started, but 3 things we are doing. So we are doing the antisense, we heard about it. So we have some early stage very interesting data. And the peroptosis, we have very interesting data we have communicated. In fact, I have a slide I can show you a little while after some time. In fact, we have identified first time GP export activator, and we have established the link between amyloid beta, peroptosis, and Alzheimer's disease. And people usually take molecules only targeting antioxidant reactive oxygen species, but we have identified a small molecules which bind to GP export and enhances its levels and activity. I have the data right here. Yeah, and the other thing is we have just identified non-coding RNAs. Just 2 non-coding RNAs we have identified. We believe this is completely new, and we believe it will serve as both as a biomarker for a detection and also a therapeutic target. And coming to small molecules, no offense to you, because I'm also interested in antisense and even antibody and whatever. For example, antibody, the drugs, whatever he's talking, with all due respect, it is approved, but there is a lot of negative, this one is coming out of that because there is a bleeding, inflammation, and there is a shrinkage after antibody treatment. And I strongly believe this is, there will always be improvement. I cannot say that it will stop, the area will stop, but as of now, with these 2 and the antibody, things are not going very well for Alzheimer's disease. And I believe small molecules still play a very, very important role because they will be less expensive, and if we can develop a very potential molecules and we can address every the full society. For example, this antibody, it costs around 40 lakhs. Now a little bit maybe reduced in the US. In India it has not come. If it comes to India, per patient per year it will be around 40 lakh. And with all the negatives, so I don't think it's a big thing. It's the way to go. But definitely small molecules, I still believe, I'm not a small molecule guy, used to be, but I believe small molecules still play an important role. In addition to that, small molecules not just targeting these protein aggregates. For example, the molecule I showed you, I only showed the protein aggregation, but it is also acting on a memory pathway. So it's a multifunctional. So we need to take care of, because protein aggregation alone cannot do the job. Protein aggregation is a core, but it's one of many pathology, many disease mechanisms. So we need to be very, very careful while designing such molecules. And of course, the RNA, non-coding RNAs or antisense also possibly code. Of course, we have to be very careful again, not disturbing the other physiological routes. I hope I have answered. Yeah. So other data I will be happy to show you. In fact, I was determined to show here on this platform, but I avoided. Yeah. Maybe I can flash it. For example, you can see that. So we have identified a GP export. So I had a slide. So yeah.
Yeah, Mano.
Yeah. I will have a much shorter question than Professor Iqbal.
Okay. Hope I also make it short answer.
It's really amazing, remarkable. Congratulations for doing this kind of work, and it makes this country so proud. I'm so proud of your work.
Thank you.
So my question is, when you are making these small molecules, how do you deliver, and what is the advantage compared to other mechanisms So are you eventually going for oral delivery? So you are achieving blood-brain barrier penetration.
Yeah.
That is one question. The second question, I'm equally interested in understanding your non-coding RNA approach. Are you thinking about microRNAs or what are?
We are thinking of micro and many of them. So maybe I will talk to you. So probably offline we can talk about that. This I will talk to you separately. And it comes to blood-brain barrier again. People talk. This is an important problem. And as I told you, I have only gave you glimpses of few molecules, but I have developed so many molecules. Not all of them crosses blood-brain barrier. So we were lucky. Particularly this last molecule which I showed you, it crosses blood-brain barrier in the animal animal model. So that we have shown it crosses. Of course, the mechanism, full detailed mechanism, we also don't know, but it crosses. So it is just that we got lucky. Of course, we have also in the series many other molecules they don't. This particular molecule crosses. So that is it is a, you can say, call it serendipity. But in this case we are lucky. But in other cases, for example, we are also working on antisense. So there also we are developing some delivery agents where we will use possibly some of the targeting molecules, some you have mentioned morning, or something similar of that type. I can talk to you because your inputs also will be very valuable for us. But we do have a concrete plan for that.
Well, I'm sure all of you will agree that Govindaraju has actually stunned us with his outstanding piece of work, and it portends really well for our country and the humankind. And please join in thanking Govindaraju for such a stupendous work.
Thank you.
Yes, it works on dementia.
In mice. Yes.
Mice I know, but you can start injecting. But have you actually, till date, to the extent I know, only with the autopsy one can detect this Alzheimer's.
Okay, sir. Sir, actually the detection part I didn't talk about it. So we have a small molecule which crosses blood-brain barrier,
Some
and we are,
biosensors.
it's a small molecule, and we are also already that is part of our company. So we are taking it forward.
Kindly present it on our behalf. Thank you.
One thing everybody should remember, Professor Bhattacharya was a student of Professor Govind Khurana.
Thank you all. And so that brings us to the end of this session. And we again will have a short break for tea. I don't think any of you had a chance to fall asleep with these two amazing lectures that we had. But anyway, there is tea and coffee outside. Maybe we can just take a short break and then we can assemble back again at 4:30 for the big event of the day, the felicitation session for Professor Ganesh. Please.
Hello. Hello.
Hello friends, and welcome back to this session that we are looking forward to now. And I request Professor Hota who is going to anchor this session on our behalf as representative both from NCL as well as ISER Pune.
Thank you very much for considering me still to be an NCL guy.
Also ISER Tirupati.
Okay, thank you very much, Manisha, and the so-called Professor K.N. Ganesh Endowment Trust. Why I say is a very nice thing. That's called, the more colloquial and appropriate word is gangs of Ganesh. Okay, that is much better than calling it like endowment and then gangs of Ganesh. I mean, that's better. I mean, everybody will be familiar with that one. Okay. Let this, with, yeah, very appropriate. I mean, it becomes too formal. I mean, why we have to be so formal about that? Okay, so before we start, I think I request Professor Ganesh and Mrs. Ganesh to occupy the dais. Yeah, because I mean, otherwise any other person who is also born yesterday can also join there. and yeah.
After marriage, we never sought isolated,
That's one of the advantages of aging, Though I think some people may say that whatever HKN peptide or which peptide, the tripeptide, where is?
Because I won't be able to give this actually.
Okay. Which peptide will be that one? HKN? GHK, yeah. Along with that GHK, these are the perks that you get. Yeah. Yeah, and K also is there, but yeah. Anyway, so with that one, let's start. And to start with, I think we got a very nice presentation which has been made by, I'll not tell you that one right away, but wait for some time. And this presentation summarizes what is Ganesh over last 70 years since 1953. See, he studied in that Mahajana higher secondary school. Yeah. Oh, okay. I thought in the beginning. I think for those of you who don't know, he is a Morley Chassis. Of course, he is. Oh, we have seen this picture earlier. Yeah, on the lighter note, both Mano and Ganesh had similar kind of hairstyles at that point of time and now also. They are continuing their friendship. Wow, this one is again yet another nice species. presentation. I think, I think by now you must have 100 questions in your mind about how could he do all this one, including this presentation. By the way, this presentation was made by him only. Okay, but anyway, let me tell you the answer to that one. I'll try to tell because of my interaction with him over the last 15, 20 years, and that will be the last sentence that I will say. How he could build two institutions of excellence in this country with all the bureaucracy that is existing. So that I will tell only towards the end of it. So bear with me till the end. Okay, that's one way to, and keep thinking why he could do it. And then if you have any sense of how one can do that one, I think you can also tell that one at that point of time when I call you upon. Okay, so by now you also must have understood that he has a game-changing thing that happened in 1981. There are a couple of things that have happened in 1981 in his life as I see it from that presentation. The first one is he returned and joined IISc Bangalore as to start his work in peptides with Professor Balram. And the second one is he got married too in 81, I suppose. That's what is written there. So now there is a message, though his father-in-law and mother-in-law wanted to join, but they could not join here today due to some health issues. And so they have sent us a nice video clip of their feelings about Professor Ganesh. Let's look at what they are going to say.
Good morning.
So he is, I think everybody knows that.
I am going to talk about Professor Ganesh as a builder of institutions and a person committed to spreading the message of importance of science. And he has contributed immensely for this. And ISIR Pune, ISIR and ISIR Tirupati are standing examples of his commitment. He has built at both places, he has built exceptional facilities and up-to-date modern labs, and he has contributed immensely towards the spreading of the message of science. And as a human being, he is a very caring, inclusive person. He has been always open to suggestions from his colleagues, has never maintained a distance, and for the students, he has been a father figure, promoting their personal, solving their personal problems on a day-to-day basis. I wish him all luck and wish him a very happy birthday on 25th of this month. Thank you.
Oh, that's a beautiful message from his. So one among us is a person who worked with him while and by the way, Professor Ganesh has got 2 PhDs. I don't know whether he mentioned that in his presentation or not, but that there was no mention. He got a PhD from University of Delhi too. And while he was doing PhD, he overlapped with Professor Iqbal, who is here. So could you please pass on the mic so that he will share his experience or his interaction with Ganesh?
Thank you very much, actually. Maybe I can stand there.
No, that's okay. I mean, yeah.
This way, right? Now, let me at the outset thank the organizer, Dr. Natu, Monisha, and others. Okay. You know, I've stopped traveling these days, but I think for the sake of Ganesh, I decided I must make this journey. Where do I start from? In fact, I think I'm the only one in this room who has known him for more than 50 years. Let me go back to 1972. I was already a PhD student with a gentleman, very well-known professor at that point of time, Professor G.V. Subramaniam. And I had a very distinguished colleague, one many of you know is Professor V.S. Chauhan. And we were all local in the sense that we could speak the local language. There comes one day a very thin and almost a school kid-like person walks into the lab, introduced by our professor that this is Ganesh. So we jokingly said that, is he going to do your master's or something? Because Ganesh at that point of time was underage. He was not allowed to register as a PhD student. And he had to wait for a year, in fact. Till, I mean, Delhi University has its own rules, of course, every university has. Now, in the meantime, he came in the company of two very notorious people called Javed Iqbal and V.S. Chauhan, because according to our professor, we were not really ones who should be doing PhD, at least me. Because we were most time found in coffee house. Those of you in new chemistry department of Delhi University, the coffee house was not very far from where we used to work. In fact, it was just about a few minutes away from our lab, old lab. Ganesh grew up under our tutelage, in fact. I can claim that, and he knew no Hindi. That was a very difficult person to have around him, because if you don't know Hindi in Delhi, then what do you know? Lot many people, of course, only know either Punjabi or Hindi. I'm talking about early 70s. So he learned one word, which I think today he was narrating to somebody. If you ask him in Hindi, he said, "Pata nahi." So one day he was standing at the bus stop. Somebody asked, "Ye bus kahan jayega?" "Pata nahi." Phir usne pucha, "Aap kahan ja rahe hain?" "Pata nahi." So this is how his training started under us. And then he used to live in a hostel next to mine. I was in Goyer Hall, he was in Jubilee Hall. And V.S. Chauhan used to live with his parents. So three of us actually started knowing each other very well. And I think Professor Sheshadri used to be there at that point of time, who would around 6 o'clock in the evening come down from his office and visit almost every lab. And this is where most of the people used to frequent coffee house would make sure that they are around, because even though we were not working with him, he would make sure that he would visit each and every lab, irrespective of whether people are working with him or not. So that was one time when we would all quickly walk into our lab, stand as if we are doing anything. And then, of course, so slowly and slowly, Ganesh started learning Hindi. And then, of course, he came under the tutelage of V.S. Chauhan and Javed Iqbal. And of course, I don't want to talk about many other things, which is not good at this forum to talk about, because I'm sure many of you have guessed it. Actually, let me now go on the serious side of Ganesh. Ganesh and I, of course, worked together in the same lab. There used to be this V.S. Chauhan who was runner, who actually ran for, I think, Delhi State or even for some India or something. He was a big runner. So there was a running ground next to our lab where he would go and run in the evening, and he would ask Ganesh and us to run his column. Those days, column will go for almost a month, because we were working on something called lac resin, where we used to isolate a lot of terpenes. And they were very notorious terpenes, in fact. And so, anyway, so, Ganesh was one person who actually, among three of us, was the most studious, liked by our professor. Many years later, when I was in Cambridge, he came, and Ganesh and I actually were in Cambridge together, and actually we shared a room for some time. And I can tell you that while I'll come back after drinking in a pub, I'll go to sleep and he will still work till very late at night. He would work on something, This is my personal experience. Anyway, so I asked the professor when he came to Cambridge, and I asked him, who you think, in your opinion, the best student you ever had? I knew he will never talk about me. He, of course, said Ganesh. In fact, he said Ganesh is the student who has really. Now, look at the person who has traveled a lot today. Actually, many things I did not know about him, because I actually moved out into industry, IIT, and then industry, and therefore my interaction with Ganesh was not as much. But we used to somehow, but the kind of journey he has traveled is amazing. look at the kind of recognitions, traveling, members of academies, member of publications, papers, journals. I think it's amazing, and also the students he has created is remarkable. Some of them are, today I met a couple of them who are now running their own companies. And this is something that really is something that very unique in terms of what he was doing and how his students have moved into something which is commercially very sort of attractive in terms of what they do. And of course, running or building two institutions over a period of 15 years is no joke. And I have some friends who also built institutions. I was with them some time ago. They look almost 80 years old, and when I saw him today, I said, look, you still look the same Ganesh. Of course, not the Ganesh that came in 72, he looks like a schoolboy. But then, hasn't really changed much except that, like me, has lost hair, and of course, he's also grown a beard like me, of course. But I think, it's amazing how, Although Dr. Natu said that I should speak for 2 minutes, first he said 5 minutes, then 2 minutes. I hope that I have done an average of 4 minutes, and I think I should stop here. Indeed, a pleasure. I could have gone and told many things, but maybe not now. But those of you who are interested, I can talk to you later on as to what we did. And wish Ganesh many more years of building institutions. And I'm very proud that I have a friend, a colleague who has been endowed with such recognitions. And in fact, those of us, let me just finish. A lot of my colleagues and friends with whom I grew up as academics, in fact, many of them become directors. My IIT Kanpur colleagues, if you can go on naming them, half of the department became directors. And it is and more than that, that actually you get to build an institution, which is even more exciting. Because many people go to an institution and run it. I also ran a CSIR lab but then, building an institute itself has its own joys. You have your own stamp on it, and I think he has done a remarkable thing. And I wish all of them, the family, Mrs. Ganesh, daughter, everyone, and thank you very much for listening and putting up with me for this while. Thank you very much.
Thank you very much. Thank you very much, Professor Iqbal, for that great setting up the tone. But again, the question remains to be the same. How did he do that? How could he do that twice? And how did he keep that particular enthusiasm up for maybe more than 30 years or so? So let's wait till the last minute. Okay. In the middle, if anybody has any kind of idea about how that could happen, you can always say that whenever I interfere. Okay. So now, not only that, as he built two institutions, so he has to pass on the baton twice also. Okay. So the first time he transferred that one to Professor Jayant Udgankar, the picture was shown. But the second one, he passed on the baton to Professor Santanu Bhattacharya. So I request the current director of IICR Tirupati to come and then say a few words and then about Ganesh.
Thank you. Thanks to Dr. Natu, Dr. Manisha, Ramana, and the entire team for putting together such a memorable event in honor of Professor K.N. Ganesh. I have seen him first time in 1991. One of the bioorganic chemistry meeting that was organized in BARC. And I was introduced to him. I think Dr. John Barnabas introduced him to me, and then we started our friendship. And then subsequently in 1992, there was a meeting in Indian Institute of Science that was entitled Chemistry Today and Tomorrow. So Professor D. Balu actually took both of us together, and then he said, both of you belong to the tomorrow type of chemistry. So that's how I was introduced. And in practical life, when we were looking for DNA samples, I think that time nothing was available in India. And there was a company called Bangalore Genii, which was manufacturing, but that is
more than 60% was either salt or other impurity of incomplete coupling products. But that time, I think Professor Ganesh demonstrated how you can make oligonucleotides of high purity for the first time from our country. We all know his legendary contribution of institution building. He has not only built the best ISAR of the country, that is ISAR Pune, and also one of the youngest ISAR for last 5 years. Whatever he has built, the dedication, the diligence, apart from his academic brilliance, I find it very, very, surprising that how much humility he has. This is a quality, is something we must emulate. And this is something, not only his ethics, his equity, his academic brilliance, his honesty, all these factors we all know about. But this humility is something one must actually remember. And I think I have only requested him to guide me because this is something in an era which is not known to me of building institution, a new campus, and he has actually assured me that he will be with me. But his connection with me comes from another angle because both of us belong to the same family because we are coming from the DNA family, Hargobind Khorana. I am his student and he was also connected with Michael Gate who worked with Hargobind Khorana. So genealogically we are also connected. But you can also see 1953, that's the year we came to know two DNA structures, first by Pauling and Corey, published in PNAS, and 2 months later in Nature, Watson and Crick. And the following month, May 25th, Ganesh was born. And if you look at May 25th, how many famous people have been born? There are many names. I don't want to talk about some of the 9th or 10th century emperors of China and Japan. One name all of you know, Zeeman effect, the magnetic field, the Zeeman effect, who got Nobel Prize in 1902. He shares the birthday with our friend Ken Ganesh. And if you look at Krishna and Ganesh, to me, it should be a peptide. It's a 13-mer because KRISHNA is a hexapeptide starting with lysine. It's connected with N, which is an asparagine, and Ganesh is also a hexapeptide, starting with glycine to histidine. So it's only natural he should move from DNA to amino acids, and that's how it came what we know.
Again, he could not join us today, but he sent us some kind of a message that I request him to play now.
It's a great pleasure for me to be able to say a few words on the occasion of Dr. Ganesh's 70th birthday. When I think about Ganesh turning 70, I realize how old all of us have become. I first met Ganesh sometime towards the end of 1980 or the beginning of 1981. When he arrived in Bangalore, I think he was fresh from postdoctoral work at Cambridge, and he came to Bangalore looking for a research position. For a little while, a very short while, he was in my laboratory, and I remember that Ganesh and I were at that time trying to do some fluorescence work on my cells. But very quickly, Ganesh moved to the Center for Cellular and Molecular Biology in Hyderabad. And of course, everyone knows from there onwards, he moved to other places. Ganesh had, I think, a wonderful career, and I think he was one of the first people in India to recognize that the peptide nucleic acids were a very important and growing field in the 1980s. And he established excellent laboratories for doing work in many areas of chemical biology, later in collagen research. And I think he has contributed enormously to the growth of science in the interface areas of chemistry and biology in India. Ganesh, of course, has many other qualities. He has been an extremely successful administrator, and I think his stewardship of IISER Pune, where this function, the city in which this function is being held, is really testimony to the amount of time and effort that he has spent in building a new institution. He's also had the unique opportunity to build a second institution at Tirupati. I think on his 70th birthday, what we can look back is at a stellar career that Ganesh has had, and also to be thankful for the many contributions that he's made to our science and to our scientific institutions. Thank you.
Okay. NCL. So he interacted with a lot of people, including great teachers at University of Pune too. One such Professor Wadia is with us right now, so I request Professor Wadia to just share his experience or his advice or his blessings to Professor Ganesh.
Dr. Ganesh, family members, and many, many young friends present here. It's indeed very gratifying to see so many nice words about Dr. Ganesh. Fully deserved and richly deserved. We shared many friendships in different ways, sometimes as teacher and student, sometimes as very good friends. And in all this, what comes across is that he is a true gentleman. In these days, true gentlemen are very few indeed. There is one problem when you ask people like me to say few words. Instead of hearing few words, we hear few hours. But before anybody wants to interrupt, yes, there's no point of going on saying many things more. Thank you very much for all that you have done for Indian science. Thank you very much.
Okay, thank you very much, Professor Wadia, for that great, short and sweet message for him. Okay, now I request, there is one more request for me. So what happened was Ganesh has given the baton once to Jayant, and then Jayant passed on that baton to Professor Sunil Bhagwat, who is also here. So I request the current director of ISER Pune to say a few words about his interactions with Professor Ganesh.
Ganesh, I can't really claim the kind of length of association that all these people are claiming, but I can say one thing. In the morning I talked about the institution I now am in because of you, but institution building is not just the campus and the buildings. And one thing I have to thank you for is the excellent set of people that you have hired in that institution during your period. It is very difficult to be able to identify right kind of people. It is even more difficult to nurture them, because as we all know, academicians rarely ever agree on something, and getting them ahead with your vision requires exceptional skills. So thank you, Dr. Ganesh, for handing me the right kind of people to work with. Thank you.
One photograph, few of my successes will be very nice. Come on. Sorry, I'm interrupting.
No, don't worry, that's fantastic.
It's a great opportunity to have, to No, you were saying that it is a Kal, Aaj aur Kal photograph.
Okay, any clues about how could he do that? Not yet? Yeah, I saw that. Yeah, 1 week NCL. Yeah. So you just keep it, the background to be just white. Okay, that's fine. Okay. So while he was at CCMB, we tried to find someone who can actually talk about how he was and then all that. But we tried to get, I think organizers have spent a lot of time to find one such kind of person who can give that kind of a, perspective, or what he was during that period of time in CCMB. So unfortunately he could not join us again, but he sent his message to this particular occasion. Can you roll Professor Deepankar Chatterjee? Yeah.
I am indeed very happy to note that on 26th May, ISER Pune along with NCL celebrating the 70th birthday of Professor K.N. Ganesh. I am so sorry that I cannot attend this function because of prior engagement, but I was associated with such function before and delivered the endowment lecture for the first time about a decade back. Achievements of Professor Ganesh is enormous. A scientist with distinction, mentorship, and students will vouch for that. He was a remarkable scientist and a remarkable institute builder, which is a very rare opportunity for an individual. Two achievements are very rare. And I have never seen a single person, never met a single person who would criticize Professor Ganesh or even show you dislike for him. He was liked by everybody, including people at Delhi, I think. So he was given position at different places. Now he will start his second innings during this occasion. I hope he will be much more active and I will be able to collaborate with him again. I collaborated with him at the beginning of his career and my career together and remained friends for a very long time. I wish him and Sujitla a very long and fruitful academic and family life. Thank you very much.
Okay, thank you, Professor Dipankar, for that great thing. And then there was one who started his career in CCMB and then assisted or worked with Professor Ganesh for a long time while setting up at IISER Pune. And his name is Professor Shashidhara. Again, he also was supposed to be here today, but he could not join us today. So, but lately he sent his video of his thoughts about our remembrances of Professor Ganesh interactions.
Ganesh, best wishes on your 70th birthday. I should have been there. It was my duty to be there. Fortunately, I couldn't due to some urgent matter that have come up here at HCBS. So even if I'm not in person, I've been remembering every day that we spent together building IISER Pune, and working with you as an assistant has really enriched my life, both personally and professionally. And I have learned so much from you. Ganesh, I don't know from the audience how many of you know, I met Ganesh on 1st May recently, just 25 days ago at IISER Tirupati. He's building another campus, a large campus, which is almost ready. And he was showing me around the new buildings that he's building, the design principles of the labs and lecture halls and so forth. And we are moving from one building to another building. And Ganesh was 25 days younger to 70 years, was moving so swiftly in hot summer day. It was 40 plus on that day temperature. And I was feeling very tired, but I couldn't see any kind of tiredness in Ganesh. And he was showing this building with the kind of enthusiasm he was showing into 10 years ago when he was building IISER Pune. It's not about buildings alone. At every time we were discussing, he was talking about the curriculum, syllabi, how to conduct laboratory courses, how to conduct field work for ecologists and evolutionary biologists. And he was talking about the bigger picture of IISER Tirupati and the context of science education for the whole world. I think, Ganesh is a complete scientist compared to anybody else in those, in the world I have come across. There may be a fantastic scientist, Nobel laureates, all kinds of accomplished, researchers. There could be a fantastic educationist, institutional builders, but Ganesh is all in one. And he's an institution himself. And there's so much to learn. Ganesh, I think you should build many more institutions, many more journals you should edit, many more young scientists should get mentored by you, the way we all have benefited. And today is an occasion where we celebrate all that you have accomplished. It's also an occasion to wishing you the best health in the next couple of decades. I'm sure many more hundreds of scientists will get benefited by interacting with you. And I'm sure 2 more new institutions will benefit by your leadership. And I wish you all the best. And I would like to be your assistant again and hope to work with you again very soon. Bye.
Thank you, Shashi, for that great thing. And it's not that everybody is talking about Ganesh and then about nucleic acids alone, but he also worked on various aspects of nanoscience too. And in that connection, I request Dr. B.L.V. Prasad to say a few words. He is now director of an institution in Bangalore.
Thank you, Dr. Hota and all the organizers for, again, having me here. My association, again, with Professor Ganesh. He has been a mentor, great mentor to all of us when we joined NCL along with Dr. Hota, and Dr. Hota joined around the same time in 2003 at NCL. And I did not like to take more time, but one fortunate thing happened is the institution that right now I am at, Center for Nano and Soft Matter Sciences. When I joined, after some time, I received a letter from DST saying that your governing council has been reconstituted. And I was really, with shivering hands, I opened the letter and I found that it is Professor Ganesh who is the governing council chairman. And like, I'm very, very happy. Right now, I can find comfort. So if I can explain one word about Professor Ganesh, it is when you go to him, he just says, "Maihuna," right? Like, no, don't worry, right? Like, no. So that is one bharosa that you get whenever you go to Professor Ganesh. With one anecdote, I will explain. I again happen to be very fortunate to conduct a JSPS Indo-Japan academic seminar at NCL, which was a huge gathering. A lot of people, about 20 scientists from Japan, many other similar number of scientists from India. And like, we were running really short of money, right? I don't know, there are no accounts people, audit people here, I hope. Right? And then like, at some point of time, Professor Ganesh said, accommodation was becoming really a big problem. He said, okay, he just took over as the director of ISER. He said, let them come and see what is ISER. And for that, he said, I will take care of the accommodation of all the people. So it was really that kind of a bharosa that you can get when you are really running short of all these things. And when you go to him, as it was mentioned, one of the things that you notice in Professor Ganesh is that attention to detail. What are we doing it for what for? For example, if we are building a lecture hall, what it should be, where it should be, including if you conduct a seminar or whatever it is, he will probably even tell you from where people should enter and from where people should kind of exit. So that kind of an attention to detail, very fortunate, Professor Ganesh, to have worked with you in many conferences, organizations, and of course in science, a lot of things to learn from you. On behalf of Center for Nano and Soft Matter Sciences, I have come alone. I did not bring big contingent, but
yeah, when nano things are big, It's not only in terms of the size, but when I joined, actually nanoscience, the particles are small, but actually papers used to come in big impact journals. So that's what it is. Yeah, thank you, Prasad, for that.
On behalf of Sense to Professor Ganesh and Professor Ganesh's family, so our best wishes for all the things to come, and we are very happy now that he is going to be in Bangalore, right? Thank you very much for coming to Bangalore also. Thank you.
Okay, so we all have heard about his academic accomplishments, and academics does not mean that just working in the lab or publishing a paper, but it's also about managing the publications too. And in that, he has actually collaborated, or he has guided ACS to roll out the ACS Omega, where he worked closely with editorial assistant or some senior person like that. And again, she could not join us today, so she has sent us her message to us. And then can you roll Miss Diksha Gupta?
Happy birthday to an incredible researcher, academician, visionary, and a great human being. Your research contributions and unwavering dedication have been inspirational. Not only you made significant contribution to science and education, but you have also left a lasting legacy as an institute builder. Your leadership and guidance have fostered countless careers and paved the way for future generations of scientists. I have been fortunate to work with you closely for many years. I have fond memories of our interactions and discussions at ACS, and you were always a peacemaker and consensus builder when we needed it the most. May this special day be filled with joy, celebration, and the knowledge that your impact on science is immeasurable. Here is to many more years of your inspirational leadership. Happy birthday.
Thank you once again for that great. And now I request Professor Nirmalya Balla from ICER Pune's Chemistry Department to come and share on behalf of the Department of Chemistry. Either you can stand here and talk or you can go there. Yeah.
Thank you, Bataji. Yeah, so
Yeah, I think you don't require, your voice is quite good.
Yeah, so on behalf of the colleagues of Chemistry Department at ICER Pune, we express our heartiest congratulations to Professor Ganesh for the birthday and the monumental achievement, which are nothing but inspiration for generations to come. So, Professor Ganesh is an extraordinary human being. So, Professor Ganesh is an extraordinary human being with a very special kind of energy. So Professor Ganesh, as you know, he has handpicked the chemistry faculty at ICER Pune except the youngest one. And seeing is believing. So quality, no compromise on the quality. That is, so we are very thankful to Professor Ganesh. And at the end, we would like to say that we are blessed to have such a visionary person with us. We are really blessed. So thank you, Professor Ganesh, once again, and we wish you all the best along with your family to continue the journey. Thank you.
Thank you, Nirmalya, for that kind note. Okay, so I was just reminded by our friend Gauri Shankar that he has a very, very, very rare distinction of hoisting the Indian flag for 32 times, it looks like. Okay, which no president or no prime minister of India could do so many times. I mean, thanks for that nice statistics. Okay. So one of his friends, he has spent quite a lot of time with him, and he led this particular institution, CSIR-NCL Pune, and then went on to become director of ICR Kolkata, and again he was busy in some other meeting, so he could not join us here. So, but he sent his some video recorded message for Ganesh. Could you please play that one?
I know Professor K. Ganesh since the time he joined NCL Pune in the 1980s. He was in the organic chemistry division. His easy personality helped me to become good friends. And today, it's unfortunate that I'm not being able to be present physically. In fact, I am in Port-au-Prince, Trinidad, attending a Commonwealth Chemistry Congress. Ganesh and I, as I said, were very good friends. In fact, we used to chat on several issues wherever we used to meet. In fact, that, I must say, is the first out of the 3 chapters of our interactions. And as I saw Ganesh excel research in organic chemistry, we became great friends. And in fact, eventually, of course, he became the head of the organic chemistry division. I also became the head of the physical and materials chemistry division little later. And that also helped us talk on a lot of official matters of NCL. One of the great things about Ganesh that I realized that time, he was already a great teacher. Not only he does research, he was taking a lot of responsibility in teaching. Our second phase started when Ganesh became the director of ICER Pune in 2006. As you know, in 2006, ICER Pune and Kolkata were established. And we actually took a lot of interest. In fact, ICERs were established under the initiative of Professor C.N.R. Rao. And with the support of Dr. Mashelkar and Dr. Sivaram, Ganesh and I contributed to the ICER Pune's syllabus, and in fact common ICER syllabus, and also building of ICER. And then this phase I saw Ganesh as a great institution builder. In fact, later on when I became director of NCL, we used to have a lot of official meetings because NCL and ICER were so close to each other. Our third phase of interaction started actually in 2017 when I became the director of ICER Kolkata. At that time, after 11 years of directorship of ICER Pune, Ganesh moved to ICER Tirupati. And I got a lot of help and support from Ganesh in terms of ICER system, in terms of how ICER works, and that really helped me in my 5 years of directorship in ICER Kolkata. In fact, I must say that all along I saw Ganesh, not only at NCL but also at ICER, a person who is so easily accessible, who has provided inspiration and mentorship to all the young faculties. And I think that is one of the great things that I must talk about him. In fact, he became a very, very successful director. And today, as he steps down from the directorship, moves to the next phase of his career, I will remember him.
Oh, let's thank Saurav for that one. And then one of one of us in the audience has also worked with Ganesh, and then now he's an assistant professor. May I request Ashwini Sharma to come and share his experience of working with Ganesh for 2 different terms.
Yeah, thank you so much, all the organizers, for giving me this opportunity. So I wanted to say so many things, but I'm completely, because most of the things have already been said. Okay, so I'll be just saying in new generation's language, like whatever everything has said, +1. Okay, so count that in also. And usually in research, we say the PhD students are here, so we say you have a researcher, we have 2 fathers, right? One is the biological father, right? And the second one is the PI. Because no matter where you go, so everyone, for your personal things, what's your name, what's your father's name? And for a professional thing, wherever you go, what's your name, who is your supervisor? So these 2 names will always be attached with your name, you like it or not, right? Okay, so my father usually says me, I learned very good lessons for both of them, for both of my fathers, my biological father, my professional father. So my father usually say one thing, which I have always, a lesson from my father is, in Punjabi, I'm from Punjab, so he usually say, man vich ho sach, to chat pe jaake nach. Simple thing is, if you're true from heart, you don't need to be afraid from anyone in the life or anything in the life. And I'm not afraid of anything, anyone in the life. Okay, and the professional lessons which I learned from Professor Ganesh is my second father. You can say my professional father is the number 1 thing which I really try to follow. I'll try my best to follow it. Number 1 is the respect everyone. Okay, that's a very, very good thing. You'll see like anyone, go to his office. I have seen that personally during my PhD life. Now I'm a faculty at Tirupati, whether a janitor goes into his office, or a professor goes to his office, or even I'll say PM or whatever, the people, high delegates he meets, the way he treats the people is, it's the same way. I think it's very difficult to maintain that things, right? But he always talks very politely and respect everyone. That's one lesson. I hope I can learn that from him, and I'm trying that. The second thing is the patience he has. I have never seen him angry. I think all of you agree with that, right? So I've never seen him he's getting angry on anyone. Okay, so the third thing is, which is the enthusiasm about science he has. Like Professor Manos' lecture we have seen, he has shown a picture where he's delivering some lecture, right? And I'm sure all of you have noticed, the time he starts discussing, his body language changes, his tone changes, and the face becomes like, the thing is like, even if he's talking alone to you, it's not in a conference, but that enthusiasm, I think these 3 things I really want to follow in life, and that's a lessons of life I learned from him. So with this, I wish you a very happy birthday, 70th birthday, and many more birthdays to come. And thank you so much for giving me the opportunity. Thanks.
Okay, thank you, Ashwini, for that kind note. And everybody is so serious. I think let's have some jokes. Okay. So what is the thing is that sometimes when you go to his office, it's that you will have some other thing to do also, right? So as he said, he has got so much of patience to listen you. Ultimately, the other side of the story becomes like you will become a patient. For a long time, I mean, he'll be keep on discussing. I mean, and especially that is true when he comes back, especially from any kind of a conference. And even today also, morning when Mano was giving a talk, he made notes of it. And for those of you who are still working in ISER Tirupati, I think he'll be coming. So you will get a lecture, very free lecture on today's lecture 2. And then he shares that particular notes with everybody with great enthusiasm. And then his patience is so much. Sometimes we admire that one and sometimes we suffer too, because there is somebody else who is waiting there. I mean, on the lighter note, don't worry about that. I hope that many of you have faced, but nobody would like to tell that one. That's definitely true with most of the directors. Not only him, but he is twice D square, right? Okay. So how he could do that? Can anybody tell? Can anybody? Yeah, that I will tell. My assessment. But anybody has anything to say that?
I will make it yes.
Okay, tell me.
This is from my personal experience.
That's true. I am going to share my personal experience at the end of it. He did not tell me that one. Yeah.
From people who are coming from Madurai,
Yeah.
we believe behind every successful man, there is a woman.
Yeah.
Shakti. Shakti keeps it going. That's my answer.
Okay. I mean, how does that Shakti come? For that one, I'll tell you. Yeah. Anyway, you keep waiting, keep waiting, keep waiting. Yeah. Okay. So. Huh? Huh? Yeah, I have my answer ready. I am just trying to, see that anybody tells that particular answer or not. Okay, let's not worry about that. Okay, somehow it could be very trivial thing, but there will be message in that one, my analysis. So some of his students have contributed to the success of a variety of institutions, including some in US and some in India. And one such example is Dr. Vipul Rana, who is right now among us. So I request Vipul to share a few words. He is right now in Innova Synth at Pune.
Well, it's a great pleasure to be here, and this occasion comes once in 5 years for us. We hope to have many more of these 5-year occasions of getting that energy. And this year's occasion is pretty special, considering that he's got one of the proudest moment for what we call ourselves as the KNG family. A proud moment of having father figure receiving Padma Shri. So we as a family of KNG students, we feel very proud, and we feel that this is something that we carry along, not only as a recognition of his scientific contribution, but also having his contribution in the institute building in the sector of education. When we talk about this kind of recognition and accolade, we try to build so much of enthusiasm in showing the praise. This year's recognition is extra special, being a Padma Shri awardee, and we feel very proud to be here. 2 points I have here. See, we see the number of students that have passed through along the way. I have an opportunity to speak with so many people who are working in industries, who are working in institutes, who are working in different research universities or in academic institutes. Everywhere you see the legacy of Professor Ganesh. Everywhere you see any field of oligonucleotide therapeutics, somewhere or the other, as they say that the genealogy of the Hargobind Khurana, the similar genealogy of Professor K.N. Ganesh, anywhere you go, oh, you are from you are connected with Professor Ganesh. So it's like, there is a recognition that is given. So it is kind of like, you go to have the H.C. Brown lab student, H.C. Brown student or a K.C. Nicolo student. Like that, anybody who's working in the field of oligonucleotide related any chemistry or biology, if you are connected with Professor K.N. Ganesh, you have got some recognition. That's the kind of legacy that we feel that being associated with Professor Ganesh. But another amazing thing is the kind of students that we have, across the board. If I give you an example, for example, we have, my seniors, say Prakash, who's working at IONIS, or from earlier ICs, to Rajiv, to Ramesh, to Meena, to Vasant, to Rajendra Joshi, to Gopal. But if you see the range of students, they are not only the oligonucleotide chemistry or the nucleic acid chemistry, but they have also done a lot of work in a lot of other areas, and now many of them are, the respected names in the field of excellence, that they are, considered as expert in their field. That is something which is really amazing. It's, if you consider, for example, Vasant Jadhav, he is considered now, one of the foremost person in RNAi, even at Al-Nailam. He decides a lot of strategies on how the siRNA biology should be done. If you consider Krishna Kumar, for example, he deals with multiple siRNA programs at Novartis. If you see Prakash, he's having so many chemistries, as Mano explained, so many areas of chemistries that they have developed. If you see Rajiv, earlier when he was working with Mano, he's started with Galnac. Now he's at his new place called RNA Therapeutics. He's building another technology platform, and he's being very well respected in the areas that they are working. So what I'm trying to say is not only the legacy that Dr. Ganesh has given, but also the platform that he has given to all the people. And it requires kind of a learning along the way. When we were in the lab, when we were working in the lab, in Dr. Ganesh's lab, we had people like Gopal, who was doing molecular biology, Vasant, who was doing molecular biology, Rajendra Joshi, who was doing bioinformatics. So you see the kind of range of areas that people are working, and they have passed through Dr. Ganesh's lab, and they have published all the papers for which Dr. Ganesh has, gone into the, deep of those areas where they are working on, writing the publications, working with them to create their thesis, and providing them that platform that they can go forward and work in their career going forward in the life. So not only from that point of view, that they are, whether they are doing a chemistry, nucleic acid chemistry, which is very well abundant and very well established in the lab, but also doing all the areas that you are working and providing that platform to all the students, all the people that who are working, or even not even just collaborating. They get that kind of platform. People like Govind Raju, you see the kind of excellent chemistry that they are doing. Fantastic. Mahesh, who is now going into the entrepreneurial journey. Meena, who is now a very well known person for the pharmacokinetics for these oligotherapeutics. So Gopal, who is now a very well known professor for the kind of biology that he is doing. So these are kind of just the example that I'm trying to give. We have got the legacy to carry forward, and then they are going to have the word that they are going to spread, the kind of chemistry that they are going to spread, the kind of science that they are going to do. So we come here to get the energy, we come here to get that kind of enthusiasm. And as he says, one thing that I always remember, if you like something, what you are doing, it's not work. So I think that is the motto that keeps him going. And we should follow the same way, that we should enjoy the work that we are doing, so that it becomes kind of a career builder for us. We hope that we get this kind of inspiration, we get this kind of energy for us to carry forward in our life and in our career. Thank you so much. Wish you all the very best. Thank you.
Okay, as Vipul said that one, and at the same time, everybody talked about academics. And I think I know one thing, that he also has some connections with the entrepreneurship club in ISIR and NCL at that point of time, and then later on he contributed to the innovation center that is now led by Dr. Premnath. May I request Premnath to say a few words, please, about his interests and what he thinks about entrepreneurship, or whatever that interaction that you had.
Only about Dr. Ganesh.
Okay, that's fine. I mean, his views.
Well, first of all, very happy birthday. And I think, my association with Dr. Ganesh is from a very different point of view. Roughly speaking, the Venture Center in the NCL Innovation Park and ISIR started around the same time. We shared the campus. Perhaps we were the single-digit people in that campus at the very beginning, right? And I'm very happy that had the benefit of having Dr. Ganesh there, because it was, I can tell you, that was an old abandoned part of NCL, which basically we had to resurrect and bring it back to life. So it was good to have his company at that time and his support. And it was very important that ICER also got started around that same time. So we could at least synergize some of those things, and Venture Center benefited greatly from ICER getting started around the same time. I'm not a chemist, so I'm not going to even talk about any of his scientific achievements. But I just wanted to tell you, but I do follow technology very closely. And in this era, when we are going to, when nucleic acid therapeutics and its role in diagnostics is going to play a very important role, I think Dr. Ganesh and his group set up the foundations in India. And I'm sure many of his students and colleagues will go on to hopefully shape in the decades to come the whole sector. And I hope really that we will see many of those things happening. Of course, occasionally he has introduced me to some of his students who have become entrepreneurs as well. So that's very good to see, and that is happening. But beyond that, I want to tell you that it was in February 2010 that Professor Ganesh joined our board of the Venture Center. And we have been very honored to have him as a member of the board of the Venture Center. I specifically requested him. There were times when he also wanted to leave the board because of his other commitments, and I requested it to stay on, especially because, I don't know any other person who has built institutions the way he has, right? And it is important as, we will come and go as individuals, but institutions will stay to contribute. And all of us have benefited enormously from the institutions that have been built by our predecessors. So I think in that context, it's important to celebrate people who build these institutions, who make it possible for so many thousands of students, so many faculty members to all at least try and reach out to their potential, right? And that, I think, is a legacy which Dr. Ganesh is going to leave here. But of course, I'm hoping that he will stay connected, keep doing many more things, and I wish him all the best as well. And I would like to, on behalf of the board of Venture Center, wish him all the best. But at the same time, we are hoping that he has recently, because of his move to Bangalore, requested that he leave our board, which we have to reluctantly agree to. But we are hoping that in the next board meeting, we will have a chance to honor you and also, thank him for all his contributions. On the board, of course, he has been the voice of supporting education, students, and at the same time, I think it was important for us for him to point out anything that we might do wrong, right? And fortunately, there weren't too many occasions for that. So thank you very much, Dr. Ganesh. Thanks a lot, and all the best to you.
Professor Ganesh's 70th birthday. He is entering the septuagenarian journey, and then not only that, there were some students who have also joined in US, and then they have recorded their message because they could not come here, and they were watching this on live. I think this is available on YouTube. So could you roll that one, please? So there are 3 students who have...
Dr. Ganesh Basant here, wishing you a wonderful 70th birthday. Hope you all had a fantastic day with Feast of Science that I know you relish. Sorry that I couldn't be there in person, but I know Mano is there representing Alnylam and broader oligonucleotide therapeutic society that have brought now very differentiating medicines including mRNA vaccines. Thanks for introducing us, so many of us, to the interface of chemistry and biology by or via the oligonucleotide research. And it was at this wonderful organization, National Chemical Laboratory, where many of us got this training. This has helped shape scientific careers of mine and many of my friends and contribute in a meaningful way. We are proud of your huge legacy, which is still ongoing, the work at NCL. Looking forward to many more scientific contributions from you for the good of the society. Thanks to Dr. Nathul, your always smiling co-conspirator for many such endeavors, including this one. And my friends, Manisha, Dinesh, Vipul, and also Dr. Ramana and Dr. Lele from NCL for organizing this wonderful event. I'm sure there are many other folks that were involved to make this successful. So please keep the Ganesh Krupa, and I hope you all have a great time in the rest of the event. Bye for now.
Hello, Dr. Ganesh. Ramesh and I are here with our children Maya and Kabir. We want to wish you
by the way,
very
both
happy
of them
birthday.
did PhD with
And
Professor Ganesh.
Ramesh and I also want to let you know that you have a very special place in our hearts. Not just because you shaped our lives by being our PhD advisor, but also who you are as a person. I still remember the day when you invited my mother to your home for a meal back in Pune. I will never forget the respect you had given to my mother. So thank you for everything you have done. And again, thank you very much for what you have done and what you're still doing for the scientific community. So again, we are very happy to join you on your birthday, and I hope you have a wonderful day. Thank you. Bye-bye. Happy birthday.
Happy birthday.
Okay, so one more student from his group who gave a fantastic talk in the afternoon. Before this session, I request Govindarajulu to say a few words. I mean, of course, he talked so many things, but now you get another opportunity to continue your talk. I think Santanu will definitely, he has no role in this one, so now you can do that. So I have to give the time now, so yeah.
Well, I don't attempt to say anything about KNG except to say that if each one of us, KNG alumni, doing good, I think the credit goes to our guru. That's all. And I'm from Mysore region, and more than me, Professor Ganesh is from Mysore region, and we have a special tradition of felicitating with a Mysore Peta. And so I request Professor Shantanu Bhattacharya from the angle of association with IISc to come and do the honors.
Yeah, the Mysore Peta and Agra Peta are two different things. That is, one is Peta and the other one is Peta, but these two are not the same, of course. I mean, yeah. But that fits very nicely for especially Ganesh with the transformed the hair attire that he has. That has been given to him by God.
Today I got a third degree. This is the kind of scarf we give to our students at ISA Tirupati and Kalukesh. ISA Tirupati Yoga. So it's a third degree for me.
May I request our KNG alumni to come quickly. I know Professor Rota will not be happy with me. Please do come up on the stage quickly so we'll have a photo and
Telling in a light-hearted manner, again a wedding photograph.
Are you sure? If everybody is front, who will be at the back? Abhi, now you also go and hide yourself. You can join that gang
Thank you
of
for giving me
Ganesh.
this honor.
Why you say only after I came here?
Because no, that now the gang is full. ये दोनों और उसके बाद प्रकाश और ये अजीब बस और एनसीएल का आशीष ले लेकर किया नहीं आएगा किधर है वो अभी आया है हाँ ठीक है
Sir, I wish you a very happy birthday and a long and beautiful life with Madam and family and friends. Thank you.
Thank you, Govind, for that nice, what is that, peta or feta, I mean, whatever. Peta, yeah. Not the Agra peta, but Mysore peta. Yeah. Yeah. Yeah, I know. So moving on, I think there were two other persons who have sent their video recorded messages for Ganesh. Let's look at what they're going to say, and first with Prakash. So this Thala P. Prakash is from Ionis.
Good evening, everyone. Today we are here to celebrate 70th birthday and congratulate Professor Padma Shri K.N. Ganesh for his hard work and dedication to advance science in India. I extend my heartfelt congratulations to Professor K.N. Ganesh, who earned Padma Shri from Indian government for his dedication and hard work. We are proud of your achievements and your achievements are inspirational for all of us. Your mentorship and guidance helped all of us to perform very well in a highly competitive and international arena. Wish you a very happy and healthy life.
Let's thank Prakash for that nice message. And then now, can you roll Rajiv? He's yet another student
Good
who
afternoon
worked
to
with.
you all. Thank you for giving me the opportunity to share this video clip. Sorry that I couldn't be there in person. Dear Professor Padmasri Ganesh, congratulations on your 70th birthday and on your amazing achievements. Your astonishing legacy as a mentor, scientist, and a visionary will be long-lived. You have been a true inspiration to many of us. You keep reminding us that learning is a never-ending process, not to be shy of or not to be hesitant to exploring new avenues and innovation. You were instrumental in instilling a mindset in me to think outside the box. Your urge to continue research after the retirement is well anticipated. May your dream of going back to the workbench and testing your own ideas all by yourself comes true. Looking forward to seeing more mind-blowing research from you. My family and I wish you and your family the very best. Cheers.
Thank you very much for that kind note. So moving on, I think for a long time we listened people who worked with him or who worked with him for PhD or who worked with him as faculty members or scientists or all that. But there is one who just joined us who has been one above Professor Ganesh and who has mentored him to some extent. And I think it's time for us to know about Ganesh from Dr. Sivaram, please. So I've been waiting for you for long. I mean, so I just noticed that you have come, so I think.
Was I on the schedule?
Yeah. Don't worry. Otherwise, we'll change the schedule. What is that? We'll rewrite.
No, I never. Hello, Suchitra. So no, I thought I'll come to here, not to speak. So thank you, Hota. What can I say? I really have nothing much to say because it's been, as I said, 40 years that probably I have known Ganesh closely since '89 when I came to National Chemical Laboratory. And we have been partners in many crimes. So I hope that those crimes have all turned out to be good crimes. So, it's impossible to say in words what a valuable colleague can be in your professional life. And through various stages in our journey in National Chemical Laboratory first, and then I will come to ISA later, I think we have worked very closely together in trying to create a better environment to do science in National Chemical Laboratory, to encourage young people to excel and to promote in general scientific excellence. And I think one thing that I noticed very right in the beginning is that there is a convergence of thoughts and ideas because Ganesh had extraordinarily high standards that he set not only for himself but for others. And therefore, I remember, when we started hiring people at some point of time, the first person I will go to and ask whether this person is worth hiring is Dr. Ganesh, because I knew that I will normally send the people to him to first interact, and then I will ask him, is he worth hiring in this institution? The reason is he is brutally frank, but I know that he is extraordinarily focused on that sense of excellence, and which I think we all started seeing in him when we were all much younger, but that I think has now proven to be true in the last 30, 40 years of his journey. This apart, I don't want to speak about his science. I think there are many more illustrious people here who can speak in much better way about the quality of science that he did, the trailblazing areas that he created, and I can see the kind of students that he has created from this morning lecture that I heard from Manoharan. And I think that's a very important thing, as a mentor, to be able to take students who are coming to you with absolutely no background and then shaping them over a period of 4 to 5 to 6 years into professionals who will go on to do the kind of great things that I heard this morning from Manoharan. But I think what made me an admirer of Ganesh was A, his simplicity, 2, his quiet persistence in doing what he did. Not that everything that he did or anybody did was without any barriers, but I think he never made any complaint of a system which is deficient, that is kind of holding him back. And I think that's, the mark of greatness, because system is always foreboding, system is always pulls you back, but the question, the hallmark of a great person is how do you overcome the system and progress. And I think that was his contribution to the National Chemical Laboratory. And then he, in fact, I remember way back in 2001, I said, are we in the National Chemical Laboratory, are we training our students the best manner? And the first thing I called is, I asked him that, I said, maybe there is something deficient in the manner in which we are training, not necessarily an individual mentor or a student, but across the board. I said, are we training the best students in the National Chemical Laboratory? And then I remember we had extensive discussions on that, and then many things changed thereafter, where I think he took on the leadership to make those transitions and transformations within the laboratory. Later on, and as I said, an opportunity came our way, and we had to set up the first ICER in Pune. And who else can I fall back upon, So I called Ganesh, and I said, there is an opportunity for us to create something, and would that be of interest to you? And of course, I never said he will say no, but I did ask him. And then started this great journey of almost 3 years, starting from 2004, in order to create ICER from scratch, And without, I can be honest here, without Dr. Ganesh, this would have been impossible, Of course, Ganesh will say without Dr. Shivaram, this would have been impossible, but that's okay. But I know I didn't do much. I only had a little bit of influence in influencing others, but I think Ganesh did all the heavy lifting in trying to build this institution from scratch. And very honestly, none of us knew how to do it, okay? So, I don't think he knew how to do it. Maybe that's the best thing, sometimes you have to make people do things when they don't know how to do it, because they probably will do a better job, So, so we organized everything, the infrastructure, the support, the curriculum, the student admissions, everything happened with a small group of 15 people, 12 people working with Dr. Ganesh. And that, of course, as I said, that's history now. But he not only went on to build ICER Pune, he went on to build another ICER at Tirupati. So I think Lord Balaji must be very happy with you, ICER, that he took you there, finally. And for all the good deeds that you have done, he took you for the last 5 years to the abode of Lord Balaji. And under his watchful eyes, he built another ICER, Tirupati. Of course, that too is a phenomenal place that he has built. I have gone and seen the campus as it was developing. Now Shantanu has told me I should come and see the campus when it is fully developed. I think in all this journey, we were professionals, and one good thing that the National Chemical Laboratory taught me was that as professionals, we could have individual interests, we could have individual aspirations, we could have individual ambitions, but we could all work together, because there's a larger goal. And once the larger goal we see, irrespective of who we are, or where we come from, we could all work together. And that also required the people to respond in a similar way. And I think most of us in this audience, or especially from the National Chemical Laboratory, will know that Dr. Ganesh was an extraordinarily institutional man, and the other good thing about him was that we used to have lots of debates, lots of discussions, lots of things that we could do, we could not do. But at the end of the day, when the decision was taken, Dr. Ganesh, whether the decision was in line with his thought or not, but once the decision was taken, that was an absolutely unanimous decision, and all his support and weight was behind in the execution of the decision. And I think we have gone through many such processes in our lives. I'm extremely happy that I came to know you, Dr. Ganesh, very well. Of course, I also came to know his family very well. We were not all living in the campus, very close to each other. And so we all got to know the family very well. And so it was a great pleasure. And as I said in this morning, I'm so happy, and Ashish, that you decided to do this for Ganesh here. I thought ICER will hijack him for this, okay? So not that I have that ICER is also equally mine as Dr. Ganesh, but I thought, as I said, that this was, in my opinion, the Janma Bhoomi for Ganesh, okay? So this is where he was born, this is where he became known, this is where his science became known, and this is where he grew up as a professional, as a leader. And what he did in ICER was his karma bhumi, So he just had to go and deliver his duty to ICER. So I'm glad that NCL Pune took on this leadership, thanks to Monisha, thanks to Arvind Nathu and all his former students, that you chose to celebrate this event at the National Chemical Laboratory. And I think it's a very fitting tribute to Dr. Ganesh. As I said, this morning, 70 is like a new 30, okay, for you. So all over again, you can start, and I'm sure, and life is long, I'm sure we hope to see many more accomplishments from Dr. Ganesh in the future, because somebody said the most creative parts of one life in science is actually above 80. So I'm still waiting for that, okay? So there are many scientists who are 80 who are extremely creative, So I think age is not a bar, age is just a number. You are young, you are still healthy, and I'm sure you will live a long life and happy life to both of you and to your children. Karthik, his son, has written a beautiful book. I don't know how many of you have seen that book, okay? Amazing. I was planning to send him a mail, but I forgot, So it's a book on what? It's a medical book. I saw the volume. My God, it's amazing. And for a doctor, practicing doctor, to write a book that tells you what scholarship that he has, I think is a chip of the old block. So I can see that their ability to write, curiosity, even in medicine, which is practicing. In fact, actually it's Rama who showed me that book, because I think she sees Facebook, I think, So then I went and I did some looking into the book, So I think wonderful children, 2 children who are so wonderful. And so all the best, and we hope to see you more often, now that you are freer. And I hope that you have a great, healthy and happy life together. for all times to come. Thank you.
Okay. Sivram. Thank you, Dr. Sriram, for that great visionary statements about Ganesh. And now I think I got the answer from Dr. Sriram to some extent, but still I did not get the answer because as we are trained to evaluate manuscripts of students, I mean, unless and until what is there in our mind comes into the answer sheets, we don't really give full marks for that one. Still, any guesses about how he could do he could do that D square thing? D square is director of 2 institutions and then building 2 institutions. Any guesses again? Okay, keep continuing. I'll tell after some time. Okay. Okay. I'll not elaborate that one because that is where the answer is. So now I request Mano, who has got a special gift for Ganesh. So I request Mano to release the book or the gift that he has got from US to be given to Professor Ganesh. Though it's a top secret thing, but I know what is there in that secret thing, but
Sorry
I
for
won't.
taking the time over and over again.
No, don't.
You know, somebody mentioned what you do in this life for you goes away with you. But what you do for others stays forever. And that's what Ganesh has done. And that's what we heard over and over again. Somebody mentioned about, people go from here, we publish how many patents we got, how much we did for the company, how rich we became and all that. Ganesh, he didn't think about all that. I was just searching for, as he protected any patents from all the techniques what he has developed. I couldn't find any. He was keen on just propagating science and making great students and publishing science and keeping the science going. And more recently, on 2 occasions, I sat and had dinner with him. One occasion with the family dinner in Boston, he was explaining all his things happened in the past, what is the future plans and so on. The other one with Vaidyanathan, we were talking in Chicago, his experience after coming back from the Middle East, he's thinking about future institutions which he has been asked to construct by the king of these places, in bringing the ISER type institutions, which he was so excited about in explaining. So he's going to continue his contributions for the next 100 years. And in this occasion, I wanted to give him only one small gift. This work was done along with my colleague Dhruv Datta and finally completed by Vipul. What it is taking his publications, looking just the scientific abstracts from the SciFinder, putting them all together, starting from his very earliest publication to the most recent one, so that he can just look back what he has done and what can be done. That is one thing for him. But in putting just this together, so many ideas came to our heads. Oh, only the time is not there. Otherwise, we can make every molecule what he has described into a new therapeutics. How many PNA type molecules we will be able to make by looking at this. We have also assembled some pictures at the end of the book. So this is available as a PDF. So please let us know if you want to have a copy of that. And Vipul has made an offer that if you write to him, he will make the bind copy of these books available to everybody. So with that, I want to give this to Ganesh.
Oh, maybe Vipul and Drupjyoti can also join on stage. I mean, yeah, Vipul. Go, 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 go.
Just like the way we started the symposium, the title of the book is Growing with the Double Helix. So, if I had to apply it for a job again, this one
Okay, so we are pretty much towards the closing of this particular felicitation section of this conference, and so what remains is oftentimes it has been echoed several times, and then Ganesh used to say one thing, that you may walk out of NCL, but NCL does not walk out from you. I mean, NCL and individuals are coupled with each other. The moment you become a member of that NCL family, it will be there in your blood forever. So that is rightly echoed in this particular thing by organizing this conference here, and for all of us to assemble here. And let's thank with a big round of applause for NCL Pune, or CSIR-NCL Pune. And now I request Ashish Lele to, if you don't mind, I'll just make up le lijiyega. So he only said that one. So Lele is Lele. Lele ko le lijiyega. I knew you. Okay.
Thank you so much for giving this last couple of minutes to me.
We have been waiting for you.
No,
Not the last.
I know. The last whole week has been a physically draining week on many of us because of the big event happening there. And whenever I go there, I get trapped into a few things. So I apologize if I'm late here. But Dr. Shivaram, thank you so much for saying this, that we took the leadership in organizing this event. It couldn't have been done any other way. Dr. Ganesh spent 19 years of your life, right, at NCL. Once an NCLite, always an NCLite. As Hota said very truly, it doesn't get out of your blood, it's always there. So it couldn't have been any other place but here. I remember when I joined NCL in '93, I have never had the occasion to work very closely with KNG, as we call him. But when I joined, I was told that there is this person, very big scientist, heading the, I think '94 was when he started heading the organic chemistry division. Does very esoteric science and uses chemistry to make biological molecules, right? It looked very, very esoteric at that point of time. And then Bhatnagar Award and so many other recognitions came his way. So he always had that aura for young, people like me who had just joined NCL, and we always looked up to KNG as the scientist that we should aspire to become. And then getting to know him a little bit more by the time he left, and then started building ICER, and we have watched that journey very, very closely. Still remember those few floors in side chambers when the first labs and the first classrooms got started, guest house being built on campus here in ICER. And then over the years, we started getting a little jealous of that place because it was coming up so beautiful, right? And the campus was so beautiful. That vision that KNG had, transformed that place, which was a dry piece of land and some very tasteless things happening on that piece of land that we were aware of. We used to stay close to that land in the new scientist department, so we know very, very well what was going on there. And that complete transformation of that land into such a wonderful campus was so nice to see. And for people like us, it was a learning experience of seeing how do you build an institution. And one more important thing we learned was that it's not just brick and mortar. The soul of the institute is the people who actually are there and who create that institution. So we have seen, great scientists that you probably picked very carefully and built all the division and the departments in ISER. So it's been a great learning experience for us also. I think equally well you have made such enormous contributions to Venture Center. You've been on the board for such a long time. I'm sure Premnath, somewhere in the audience, there you are, has spoken about all of that. So, as Director NCL, I also wear the other hat now of being on the board of Venture Center. So we are very, very grateful to you, KNG, for helping build that wonderful Venture Center. It's that great leadership of building institutions, physically, and the soul of it is what you have taught us. And I don't know how it is possible, but building one institution was not enough. You went and built another one. So it's just phenomenal contribution. So we have known you as a great scientist. We have known you as a great institution builder. Third innings, we are looking forward to seeing what else is coming from that phenomenal brain that you have. JN CSR, I think, gives you a superb platform. And we will look forward to really superb, the best of the science still yet to come out, and we look forward to that. So thank you for accepting our invite. Thank you for giving us this opportunity to celebrate and to felicitate you. And again, very, very happy birthday. 70 years is just a number, as Dr. Shivram said. So we look forward to many more such years to come. Thank you so much.
Thank you.
Okay, we are almost at the penultimate thing, and still anybody has the answer for the d-square thing. Still no answer. Okay, now I request another person who has spent quite a significant of time, and then he is also the chairman of this KNG Endowment Trust. For me, it's as somebody said, it is a gang of Ganesh. Okay, so I request Nathu to take the stage and then tell a few things and then do that. And quite recently, the word Nathu has become very powerful because, the thing is, whether that Nathu was powerful or this Nathu is powerful, it is up to you to decide, okay? This kind of Nathus, yeah.
I received several messages of congratulations at that time.
So the similar thing happened when Grubbs got the Nobel Prize, and then when he got the Nobel Prize, one of the recipients is Chauvin. So the Swedish guy pronounced it Chavan. Okay? And Chauvin is the guy who gave the mechanism for the metathesis reaction. He's a French guy, and the guy has, then people started talking about some Chavan got the Nobel Prize.
So, I know Ganesh for last 36 years. perhaps in chronology only next to Javed Iqbal and Suchitra. They are the senior most. And I had spoken about Ganesh so many times. So many times I feel what I knew, I think. But this gentleman does something new all the time, maybe experiment, maybe something, and that gives me some inspiration to use some new analogies. So this time, I'm going to talk mainly about the burning problem in the evening, that is the cricket. Yeah. Ganesh started his gully cricket in perhaps Mahajan school in Bangalore, and then he joined the NCA. NCA is not National Cricket Academy, it is National Chemistry Academy. He joined the National Chemistry Academy of Delhi and Cambridge, got graduated from that, and after playing few matches, first class matches in CCMB Hyderabad or IIC Bangalore, he has proved to be a great leader in all the three formats of the game. Test cricket, that is NCL, 50-50 in terms of ISR Pune, and short stint of T20 at ISR Tirupati. So he has proved to be a great leader in all the three formats. He's rock steady, wall like Dravid, captain like Pataudi or Ganguly, who had the mammoth task of building the teams, which I'm talking of him, everything applies to him. You just don't, instead of cricket, if you just imagine ISR or NCL, that is good enough. Task of building the team, like Wadekar, who had many senior players into the side, and like Dhoni, wherein it was a mix of galaxy, stars and youngsters. So he was fit for all the three. Every time he assured Tumhara Vidhi Nayega to youngsters when they failed and exemplified by his own example to take him further. Nowadays, he has left the job to Rohit and Virat in the format of Sunil Bhagwat and Santanu Bhattacharya. He plays many roles at a time. This is his specialty. He plays many roles at a time on the field and changes his roles in short time, thanks to the airlines. Sometimes he is in the first slip, very closely watching the student, that is the batsman. Next moment, he is at long off interviewing a candidate for ISR. He keeps a mini binocular in his eyes and judges a bowler many times from the boundary line, where he says that he is interested in, he is busy in the role of the institution builder, but mostly he is looking through the stadiums with big goggles. In the next ball, he is at silly point to have more keen observation of the wicket and to direct the bowlers. Many times, he goes to the deep mid wicket at omega angle to go through the manuscripts. Teammates feel that he is away, but next moment you see him discussing with the MOE, Minister of Education umpires, on some financial or administrative issues. He discusses, disagrees, asks for DRS, uses his computer and contact skills in the DRS, and gets the decision in his favor. I had never seen him only in the role of umpire. But one doesn't know, during the lunchtime, he must be dashing to Delhi to act as an umpire in some other match and come back in the evening. That is also possible in this case. Immediately he realized that he has to take a class. And again, he has to take a class. He goes to the pavilion, has a cheerful glass of drink, that is water perhaps, runs his class and again appears in the last over to bowl and take few wickets and win the game in all the formats. But in this running between the wickets, he is not a great catcher, mainly because he was injured while taking catches when he played for NCL. So, as a remedy, he appoints some good catchers, as ultimately he knows the catches win the matches. Generally, a government servant retires very early in some other department, even in the field, even in the first few years. But disproving that, he's entering the fourth role of the career, like C.K. Naidu and D.V. Devdhar, who are scoring centuries in the areas even after the age of 55 years. If live TV would have been there in the beginning of his career, like many cricketers, Suchitra has to sit all the time cheering him in the pavilion because he moves a lot and plays cricket. But wise lady she is, she has taken perfect care of his cricket kit bag and clothes, updated even for his journey in the midnight. All 24 hours she has kept the fort intact and maintained and nurtured the entire family. Ganesh visits the family also occasionally, only if there is a match in that town, whether it is in Bangalore, Hyderabad, Pune, Delhi, whatever it is. In Tirupati, he has attempted to create a cricket ambience, but there is no match luck. Many times he feels he is playing too much of cricket and neglecting the family. Then he takes the family abroad for a holiday, but sows the seeds of the next conference or meeting for the next year. But in all these games, he has never overstepped the pitch, not used any abusive language or words for everybody, including umpires, never dramatized even his small failures. Nowadays, in front of TV, if there is a small failure, in order to go, and generally fielders are dramatizing. He never did that. No delay tactics, as practiced by Dhoni day before yesterday. He always encourages his colleagues to do better with tips, but not with the shortcuts, respected the age, planned his strategies in case of emergencies like rain. He knows the Perth Lewis rules better than GFR and Ministry of Education guidelines. And see the match is through and thinks of the next day and night match with minute details and perfection. No wonder all these qualities have brought him many laurels, such as man of the match, like Bhatnagar Award, man of the series, like directorship, man of the year, like academies, wisdom player list, like several international delegations he has said, and ultimately a lifetime achievement award in the form of Padma Shri. Fortunately, I was a spectator in the stadium clapping for his success for last 36 years. His team associate and all the time a well-wisher. He is run hungry like Tendulkar, and therefore I wish him a healthy century and all the best is in next cricket innings, Ganesh 4.0. Thank you very much. But that will be.
So I think all the Ganesh gang of Ganesh will be led by the endowment trust chairman And so please all of you join on stage so that they have brought
On
something.
behalf of the Ganesh Well-Wishers and Ganesh Endowment Trust, as a mark of respect, we would like to give a small present to Professor Ganesh and Suchitra. What I have just now. Now you need 5 people. On behalf of all I would like to present this lamp to Ganesh. I hope you will be able to use it in your Thank you. Thank you.
So those of you who are still working, be careful when you go to his office. Okay, so he can use it as a weapon too, or otherwise he can ask you to weigh. Okay, so either one is going to be difficult.
Ganesh 4.0.
Yeah.
And similarly, I would like to give a small present to Suchitra. Thank you.
Please close, please.
Oh, we will
Excuse me, sir. Yes. Ready? One more. Ready? Smile.
Just before that one, I think I request. So there are two things that are remaining for me to do. One is that I have been asked by some of the people who have assembled here that why Dr. Mrs. Ganesh is not speaking. So we request you to speak. So a few words. No, that's fine. Okay. Okay, so she said that, not one word but 3 words. Okay.
You said 4 words.
Yeah. Okay, so now any clue about what is my question is? Still I am not getting the answer, partially answered, but I think 2 out of or 3 out of 10 marks only for the answer given by Dr. Sivaram. But still not 10 out of 10. So let's see whether Dr. Ganesh answers that question or not. Though the question is framed on him, but let's see whether he answers and how many points he can get through that. Okay, let's, and at the same time what Mano also has a request that he said that, what is that, he said so many people have said so many things. Would you agree or not? That's what he said. I mean, what? No, otherwise it has become very serious affair, otherwise.
Yeah, hello everyone. Yeah.
I'm really very overwhelmed, and I really do not know what to speak now, let alone answer your question. Before I say something, let me answer the question first. I think all the people who are in ISA systems know what is called a repeat exam, right? So when does the repeat exam happen? When you get an F grade. So I got an F grade in ISA Pune, so I had to go and do repeat ISA Tirupati. So that was as simple as that. Yeah. Yeah. So I'm really overwhelmed, and the kind of affection and all the words that were spoken about me, right from my teacher, Professor Vadia, and all the others. And I was just thinking, I wish I were all that what you said. I was getting doubt within myself. Have I really done that? Do I really fit into that thing, etc.? But thank you very much for all the things which you discovered about me, which I myself did not know. Thank you. Let me start. I know after all these things, you've been sitting here for 3 hours or more than that, and I'm sure all of us need a drink now, after hearing so much. And I'll be very brief and maybe say something which, myself, I feel very, emotional about, about my whole journey. I started in, Central College, I will not go very far, and all this was only because of I had great mentors, like Javed mentioned about GBV Subramanian, and the kind of person he was, both in terms of subject, personality, etc. Consciously or unconsciously, we all develop certain things from our supervisors. We know all of us do get frustrated during PhD. Some things, we like, some things we don't like, but at the end of the day, the PhD times are the most, enjoyable times. I'm sure each one of you would believe that, the people whom you meet at that time, etc. And it was very wonderful, having Javed, Chauhan, etc. Although he did not want to say what are the various things they taught to me. But it was very, very wonderful. And I think if our supervisor were alive today, I would have been very proud of Javed, Chauhan, and maybe me also, because we just happened that we are all done, probably done well. And because I had a very good teacher in Central College who prompted me to go to Delhi University, I went. All my batchmates, they joined University of Science, Bangalore, organic chemist, but I'm the only one who left Bangalore and went to Delhi University. And impressed or influenced by my supervisor G.B. Subramaniam, then I landed up in Cambridge. And so lots and lots of things we did as students. One of the things which I clearly say that we worked as a team even at that time. As Javed mentioned, we used to work on what called lac resin. So Vadia would remember all the terpene components. In fact, we were competitors at NCL Pune, Sukhdev's group and T.R. Shishadri's group. But they were much greater, but we were contributing in a tiny way. So looking at that, I was amazed when I came back to NCL and even I got a job in the laboratory, which was, kind of a competition or whatever, in real sense, science things. So when I went to Cambridge again, it was a kind of, game changer. I chose to do a PhD degree, and staying in a college and doing a degree was entirely different. Doing postdoc abroad is different, but in Oxford and Cambridge, if you get registered for a degree, to be with all those students, undergraduates, college system, it gave me lots and lots of experience, which later on I started practicing. I think it just became a part of my life. Came back, and for a very brief period, you heard Mr. Balram. I was there. When I came back from India, I think my supervisor Jeremy Sanders, he was an NMR person. In fact, Turi NMR came when I was just doing PhD. And he said, if you go back to India and want to continue with NMR, you must work with Balram. He was the first one to do what is called as the NOE experiment in a small molecule. He worked with Botner B. And later on, he did his postdoc with Woodward, working on the NMR spectrum of vitamin B12, etc. So when I came back to India, and I came back, I came back with any preparation. I went around looking for jobs in different places. And one of the places was IPCL Baroda, when Dr. Shivram was heading the R&D. So they had offered a job to me. But then I came back to Bangalore, and several things influenced, and I started working in Balram's lab just for a brief time. Then I went and joined CCMB. That's the time at CCMB was being built by Dr. Bhargava. So I started my career in looking at how institutions are built. I think that had a great influence, the kind of thinking which Dr. P.M. Bhargava was doing while building the institutions, what you look for, and to have that grand picture of something, even if you cannot achieve it, that was very, very instrumental. And I was an organic chemist and was wondering what I do. I knew that the future of chemistry is in biology, but I was not sure what I should do. And within about 3 or 4 years, I realized that many of our biology colleagues, they wanted DNA fragments, for restriction fragments, for cloning, etc. But oligonucleotides were not available. In fact, even abroad, it was not very easy. Only in selected labs it was being done. So somewhere after 3 years, I went and told Dr. Bhargava, as an organic chemist, I would like to get into DNA synthesis. Although I did my PhD in Cambridge, I never got a experience in the DNA. So then he allowed me. Then I went to Michael Gates' lab just for 3 months. And when I went to Michael Gates' lab, he said, okay, you come to learn oligonucleotide synthesis. We want 20 milligrams of a 23-mer. That was the problem definition. At that time, micrograms was a challenge, and going from microgram to milligram was a big scale-up, at that too of a 23-mer. No machines, nothing was available, and we had to do what is called coupling by phosphotriester coupling, using pyridine as a solvent. And once you start, it is a half-an-hour coupling period, and to make a 23-mer, you have to do 11.5 hours, simultaneously. You cannot leave in between. So you come, keep on adding. Somewhere after the 10th coupling, the reaction fades. You just close it up, come back. So Chitra would remember, that I would come back the day. When I come back early, she knows synthesis has not succeeded, come back. And when I come back very late, maybe it has worked. So at that point, even a scale-up, which I learned the scale-up problems after joining NCL, but DNA was a very big, which kind of resin you had to use, a control per glass, or organic resin, whichever. So by repeatedly, we could make about 20 milligrams, close to that, one strand by one method, another by strand by another method. But I'm sure Mano would remember, there were no methods of purification of oligonucleotides. HPLC, the only method available was you had to run column at 60 degrees using formamide as a solvent. And how do you recover oligonucleotides from evaporation of formamide? That's a serious problem. So, but anyway, made all of it. I showed by analytical HPLC it is pure, but I didn't have time. All this was done in 3 months. I came back to CCMB, and MRC was so gracious. They gave me a small synthesizer with a 1 cycle automated with a small wall system. I came back and built up this DNA synthesizer at CCMB. And later on, people joined, including Sudhir Agarwal, one of the big names in oligonucleotide. He joined Michael Gates. They completed. Finally, oligonucleotide was made, and it was given to Kurt Utrecht because he wanted to do NMR. Doing NMR of a 23-mer oligonucleotide was not, again, an easy problem. And Utrecht had developed all these 2D techniques. And later on, he did that NMR, and we got a paper, as was shown by thing. Years later, when Kurt Utrecht visited NCL for one of the science congress thing which was organized, I met him, and I told him, I was the one who was made it. He said, that's fantastic. Even if I give $1 million, nobody can give me this oligonucleotide at that time because it's just not, was possible. So I came back to India and set up this small synthesizer, started working on DNA, but then I realized I already was thinking of how to modify DNA. And I wanted organic chemists to join my lab, students for the PhD. CCMB was a great place. Dr. Bhargava, as a director, he would sit in the PhD interview of every student, in the interviews for the PhD admission. He said, I will only select potential Nobel laureates. We used to say it's easy to get a scientist position in CCMB, but not get a PhD, as a research scholar. But we could never get a chemist because chemists would not be able to answer so much of biological questions. And finally, I got a student from Usmania University, would be my first student, an organic chemist. He comes and he says, I want to isolate a protein. I was looking for oligonucleotide chemistry. He said, I want to clone a protein, etc. But whatever. So, but through that, I learned quite a lot of biology. And that's where Dipankar pitched in, and we have an excellent paper on how to study repressor protein interactions by chemically mutating the DNA. Because I could make sequence, I could mutate every base, made a lot of oligonucleotides. And normally it is done by genetics means, but here we could do that, and we mapped out what are the bases which are required for the particular DNA-protein interaction. So it's a great learning process. There's a time when I met Professor John Barnabas, one of the fantastic teachers. I think Professor Wadia would know about it. And Professor Barnabas, working in Ahmednagar College, he published a paper in Nature, about hemoglobin. That's amazing, working in a small college here. And then he became the head of biochemistry at NCL. And he used to come to CCMB very often. He was a person who was fascinated about synthesis, biosynthesis, etc. Although he was an evolutionary or a biochemist. And then he told me, Ganesh, you must come to NCL. You must do biology in a chemistry lab rather than doing chemistry in a biology lab. So I remember it was in 1987. I spent 6 years in CCMB. 1987, it was in January, I still remember, he organized what we call as a job talk to come to NCL. And I still remember, NCL auditorium was my seminar. He was so enthusiastic, John Barnabas had invited. Right in the front, we had L.K. Doraiswamy, R.B. Mitra, Mashelkar. They were all sitting right in the front, 1987. And I was talking about solid phase peptide synthesis, but I'm not sure, anybody would have heard of that. But anyway, I don't know whether it is right or wrong. They gave me a job. So I joined in May 1987. And these days, when you join, you get startup grants, right? And I think the best startup grant I got from Dr. L.K. Doraiswamy and Dr. R.B. Mitra was Dr. Nathu. He said, we'll give you 3 people, Dr. Nathu, Dr. Vaijayanti, and Anita. These are 3 people who will work with you. And they gave 40,000 rupees as a startup grant. And the DNA synthesis chemicals were extremely expensive. I know we had this first lab there, it was actually, it belonged to Professor Venkatraman. When I went to the lab, I could see a lot of dyes, these cotton fabrics, very colorful dyes, asbestos top, we knew that we should not work on. That was a thing. But nevertheless, I was quite excited. I joined. But one of the things of Dr. Bhargava was, when I left, I must share this thing. He never expected that I would leave CCMB, we were the first one to move to the new CCMB labs. He never expected. But my problem was getting chemistry students. 2 weeks before I left, he assigned a chemistry student to me, fully knowing I might leave. He thought that I would still, stick on. And when I went and told him, Dr. Bhargava told me, yes, you have made your decision. But any day, if you feel, you get disappointed there, come back. This job is for you. In fact, he had done for Deepankar Chatterjee, who joined CCMB, went to Banaras Hindu University. He was not happy. After 1 year, he came back and he got the full things. What a magnanimous, person, Dr. Bhargava was in that respect. He also told me, if you don't come back within 1 year, someday you will become director of NCL. I still remember that, But, anyway, maybe that would have happened, that did not happen, whatever it is. But the most important thing was I had, at that point, 1987, 5 lakhs worth of DNA chemicals. The lab was very rich. I had bought it. He said, take all these chemicals. So I got 40,000 rupees startup grant, but I got 5 lakhs worth of chemicals, DNA synthesis. It's amazing. 40,000 I used to, replace all the asbestos stuff with, the Formica tops, etc. And I had Dr. Nathu and Dr. Vaijayanti, who had been trained very well in chemistry, although they did not know anything much about the DNA, etc. So I started charting my own path, DNA synthesis, RNA, and then I could get some grants and I could buy all the analytical equipment, etc. And of course, I got, I wanted chemistry-driven students, and that's the time Vidya was my first student, and then Manisha joined, and a lot of, Prakash, Rajiv, they all joined. We pursued entirely something. Many times there was a disconnect between the kind of chemistry, what we were doing with the other, big things happening in NCL. People synthesis of drugs, insecticides, pesticides, everything. And Dr. R.B. Mitra was head of the division, and after giving these 2 very nice startup grants, I never met him for about 6 months. And then he had to leave. He became the director of CLRI. And 1 or 2 weeks before, he called me and said, I think, Ganesh, we have something in common. I also work on these insecticides, pesticides, they all have phosphorus. DNA also has phosphorus. I think we should collaborate. Phosphate, we should collaborate, I still remember. And of course, after this left and Dr. Rajappa took over, And then this so-called OCS, organic chemistry synthesis, came. And the culture of PhD, I mean, it was slightly different at NCL. In fact, internally people were graduating, etc. They were coming. There were various of outside PhD students. So we took a route with Dr. Rajappa and all that started, students, induction, etc. It all happened. But I was always feeling this, and when Dr. Rajappa left, Dr. Mashelkar called me and said, I think you should become the organic chemistry synthesis head. I was not very sure because I didn't have that kind of per se, synthetic chemistry expertise to be head of organic chemistry synthesis. So I told him, I think, there are other people here who deserve it and I should not. He said, no, this is a decision of the management. You must stay. And that's the time I talked to Nathu. Look, I want to learn industrial synthesis chemistry. And we visited almost lots of industries around Bombay, looking at the reactor, scale up, all the kinds of things. Nathu had good chemistry connections. And I did all that, but I cannot do any of things of my own, but, just to get some projects for that things, etc. It's an amazing thing, and I'm very happy that all the students who did at that time, although they were not, industry oriented, I think all the products, the students who came, now they're all working in industry, R&D, somewhere else, and some of them have made, they're in academics. So in a way, we are a little bit ahead of time, now DNA involves a lot of, other kind of scale-up problems. And somewhere it was 1990, I don't remember, 96, 97, when was that IRT meeting was held at ISIS that was my first exposure coming to US, and that's where I met Mano for the first time. I could really see, the DNA, it has to be scaled up. Yogesh Sanghvi, I met, Prakash was there at the time. That is a great open. I think he was the one who first gave me a, the site of what is oligonucleotides, how it is coming up. In fact, the title of the symposium was How to Make Oligonucleotides as Drugs. That was the title of the symposium. If they become drugs, what are the other kind of things? Nobody knew, not even FDA, how to approve them as drugs, etc. So why I was telling you that I grew up with the area. That is most important for me. When I started my career in DNA, no machines were there. So I grew up with the area, and it gave me a lot of advantage and gave me lots of breaks. And somehow I've always believed that, working scientists should at least attend one international conference. That was my motto, at least one international conference. Because when you go to an international conference and come back to India, you feel totally different. Even if you're presenting a poster, it doesn't matter. You should go, meet people, discuss. When you come back, you're so excited. So, we developed. So, and then Dr. Mashelkar, I think during his time, I learned so much. He gave me quite a bit of responsibilities, managing things. And to see people like Dr. Mashelkar and Dr. Shivram working, and you really get the, what is about leadership? What is about management? You have to just watch them, And that was a great thing for me, and that's why I could do something at ISIR Pune. It was only because of observing Dr. Mashelkar and Dr. Shivram, working at it. And quite a few things we did about leadership, attending workshops, patent awareness. It brought a different kind of dimension to my own thinking. And the best thing happened was, as Dr. Shivram already mentioned, this ISIR Pune break. And I also must mention, I was most impressed in when I came to Pune initially was the kind of cordiality which happens between NCL and the University of Pune. Professor Wadia, Professor Narsiman, Dr. Dawle, and so many people, it was a routine thing coming, going, and students. It was a fantastic thing, the university NCL partnership. And I also got opportunity to teach in Pune University, and I had to teach NMR to biotechnology people who don't even use it. And that is the other challenge. If you want to teach something, and then you start simplifying, and you try to understand the subject much better. So Pune University really gave me a very good platform to teaching, understanding the problem of teaching, curriculum, etc., and the simplicity of all the professors. I think I imbibe a lot to my contact with Pune University, particularly Professor Wadia and others, who sort of really told me, what is it to be like a teacher, just teaching without expecting anything else. So, ISIR Pune was a different thing, and again, Dr. Shivram, he himself mentioned, he just asked me, are you willing to take over this thing? And he said that none of us, we knew, how to start institutions, but Shivram was, like, the way in which he, made the first fight and golden rules for starting the institute, and particularly how NCL can help in establishing it, and then getting this Mirado, which was a building which had to be dilapidated, but that has to be done. Similarly, a hostel giving for the first, the 25 or 35 students who joined. I think on August 16th, 2006, I think, when the first batch of BSMS students came in, a lot of NCL colleagues, the engineering people, the other scientists, they all helped so much in setting up that hostel in every way. So we had a very wonderful time, and, I think I showed the picture, the first flag hoisting, what we had in the NCL hostel here. And after that, dealing with undergraduates is something, I learned a ball game. All of us do PhD students, we teach them, or even postgraduate, but handling undergraduate student is a tough, thing. And there were so many episodes, RGB joined around the time, RG Bhatt and Srambika, they all joined. So, and then, I don't want to go, but things just started happening, myself, I do not know. I think I will only been lucky at all stages in my life just to have good people around me. I think that is another thing, which if I had done something, I don't take the credit. It's because of the good people around me. They used to say in the first 3-4 years, why IISER Pune is doing all these things, I used to tell them, we have Professor Shashidhara, you don't have. That's the difference. And Shashidhara, he spoke, amazing person. And the way in which he managed, he was, we couldn't attract professors in, we had chemistry, biology, mathematics, physics. We could not get professors till about 2011 or 12, 6 years to head either physics or mathematics department. I was heading the physics department. Shashidhara was heading the mathematics department. That helped us understand the culture of, each discipline. That is the greatest lesson I learned in IISER Pune was we cannot apply the same principles. Chemistry, we very well know. Somebody joins, how to support them, etc. But supporting people, biology faculty is so different from physics faculty, different from mathematics faculty. Each subject has its own culture. You have to respect that culture. You have to, support those faculty in those things. And even, for example, ecology or the field work, they require a different kind of things. The same rules will not apply. So that is the biggest, the diversity of the discipline, the culture of each discipline, I learned quite a lot. So, IISER Pune, first term, 5 years, and again, shifting, we got this thing, then we shifted to the, near that, Cosmos Bank, we call it Balance Building. We went there. After 5, 6 years, we again came back here. We made one by one. None of them were conceived, it just happened. We call it Mendeleev block, then we call H-cross physics, and then we just had Mendeleev block, and we thought that other building would come up. It didn't come, so we made one for physics, one for biology, and they've all, even today they're in existence. They were put up in a. It just happened actually. I still don't know how it all happened, and the government funding was coming in piecemeal. Many times contractors would raise the bill. We don't have money to pay. Some would happen, and by about 10 years or so, we took, or even less than maybe 8 years, that the whole campus was set up, greenery and everything. And so as I again said, when I finished 11 years, and when again they called, I was eligible for a third term as a director. So when I went for the interview, and the minister, he asked, what would you, it was so happened that, they were interviewing for 5 IISERs. Everybody's first preference was Pune, to become the director of IISER Pune. Everybody wanted IISER Pune. So then the minister asked, why you want to go to IISER Pune? Oh, it's very well established. If it is very well established, what is the challenge? Why do you want to go? You must go to another institution and take up the challenge. That is the minister's thing. He said the same thing to me also. Why do you want a third term there? You should go and build up. I was already mentoring for 2 years in IISER Tirupati. It went. And a lot of things which I learned from IISER Pune building things, I could, every time I always would check with Shivram, cross-check, ideas are right, whether it is in any capacity, I think he has been a great, mentor, friend, companion in the whole growth of, the IISERs. I think one should give for IISER Pune, Shivram, the main credit. Of course, Dr. Mashelkar made it, he brought it to the NCL land, bringing the IISER. And I'm sure NCL will also feel the same way, that the IISERs came. And Dr. Lele was a member of our Senate. I very much wanted the engineering discipline to be introduced. And at some point, I approached Ashish, can you come over here as a professor? We want to start engineering. He said, no, just now I have accepted a job in Reliance. And I would like to, we just missed out. I'm glad that he's now back into, into NCL. So it's a great story. 19 years at NCL, 11 years in ICER, and again another close to 7 years, 5 years officially, but 7 years unofficially, because I was mentoring both the institutions at some point at ICER Tirupati. So I've survived as a director for 17 years. I have run out of ideas now. I don't know what else I can do. And I thought ICER Tirupati, the last ICER I'm going to build, not any more ICERs. Till this king from RAC, he said, I have heard so much about ICER. Can you make one ICER in Ras Al Khaimah? It's not often. So, but I know they don't require an ICER. We are advising something different for them. So that's what the ICER story. And then, and the ICERs were brand new institutions. Students don't know what ICER would be. Faculty do not know. Parents don't know. First 5 years, it was very, very difficult for us to convince. I think I know some of our chemistry colleagues are here who are there. Every time when the admission happens, we have to keep on teaching. Nathu took so much of tutorials for parents, telling them, we are all false, you can do this, you can do that. But in the heart of it, we know, in science degree will not, you can do, wherever you go, you can do well, but we can't promise a so-called pay package, So we went through that. And Nathu was a fantastic counselor, and he headed 2 of the JAC admission thing. And I don't know how much of time he has spent on talking to the parents to see that they don't go for litigation about any seats. It's amazing, it happened. So it just happened. And I knew that faculty is the key to building an institution. So we went to, once you appoint good faculty and give them any resources, rest of it will automatically follow. That was our philosophy. In fact, today I cannot say that. At some point, to say startup grant is a blank check. Because when we select a candidate, we know what he wants, what is the area, what kind of equipment we want. When you select him, you have to give him. Otherwise, if he is busy in writing DST, DBT grant for the first 2 years, if he doesn't do anything, then, it's too late. So you give him, think what he wants, and after that, he will bring you a lot more grants if he succeeds. We could do that. But progressively, as the years passed, the funds got dwindling. Initial stages are very good because there is nothing there. You have to create, so faculty would get, a lot of money. Same thing, to some extent, we have done in ICER Tirupati. But there, the funding policy is slightly different. Till now, to be honest, we had no problem at all about the funding. We learned quite a lot about the building. And with the other ICER directors, I used to keep on sharing, because somehow ICER Pune director was the member of board of directors of all the other ICERs. It was decided that one ICER director should be there in another one, but when they made the statutes or whatever, typographically they cut and pasted. So ICER Pune director was there in all the things. So the first 5 years, I was a member of the board of all the ICERs. Each ICER will have 3 board meetings, 3 multiplied by 15 board meetings you have to go and attend. Even today, director of IAC is a board member of all the ICERs, statutory, they are there to do that. But that first 5 years, I have seen every ICER, the first brief, how it was laid, how it developed, had a great lesson. I think I've been uniquely blessed that it's difficult to get to be a founder of one, institute, but to be the founder director of 2 institutions has been a, great this thing for me. So I had to thank God and all the good people whomsoever gave me this chance, etc. So that has been, I can go on and on. I will not, tell very much. But NCL has played, as Dr. Shuram said, it is Janma Bhumi. But actually, CCMB was a place where conception was, before born. CCMB conception happened. Then I was born in NCL. And then, the Karma Bhumi. Yeah, it was conceived in, I think. There it was, the initial courtship was happening in, I think. So that is how, now I think many people refer to as institution builder. But to be honest, I've become only a builder now. I've become a builder. Now I go, I know the contractor, what kind of tiles, how much of plinth area, what kind of light fittings. I can give consultancy on that thing now. I've only become a builder. I must say that, I think, yeah. I think it's been a great journey. I can go on. I will not do that. I just want to mention 2 people. One is Mano, who is here. I'm so happy and very moved that he could come here. He has been here for 2 of my earlier occasions. But till yesterday, we almost thought that we would miss him here. And, against all personal odds, he actually arrived, yesterday, today early morning at 2 o'clock at Delhi and took a flight, came to Pune around 7 o'clock and he's still awake, gave that wonderful talk. He has been so active, not just for that, but for all the things, which he has taken our students, he has mentored them. And I think today in the DNA area, I don't think anybody knows more about the medicinal chemistry of oligonucleotides than Mano. Very highly respected medicinal chemist, but more than anything else, how he's influenced by, those Tamil, these things, and he really implements in all those things. A great human being, and luckily we get chance every year once in a ACS national meeting, and we go for a lunch or a dinner, and every time as in US, he invites me to Al-Nailam. But my main problem is every time I visit him, I have to give a talk on a different subject or something new chemistry. That's my challenge. So unless I have something new chemistry, I won't accept, I'll give some excuse and not even visit, Al-Nailam. And visiting Al-Nailam, and talking to him has given me a very, very great exploration. And through him, I've been able to meet a lot of nucleoside chemists. You saw some photographs in that, when he organizes talk in the nucleic acid chemistry, introduces. That has been, I think, many times we say, there are a lot of things happen in scientists' life. That's the reason meeting your peer people, going abroad is very, very important. I do know that not enough money is given. CPDA nowadays, you can't even get one ticket. But there's something people don't, we may fund people to any crores, but if we don't give, a lot of things, as you know, in abroad happens only through networking, conferences, meeting people, getting to know things before. But unfortunately in India, many of us who work, we don't have the same luxury. Once in a while people come, you hear them. But somehow I made it a point to visit, that's a bit showing off all those slides, mostly having dinners, I think you saw all the slides. But dinners are the ones where you actually meet people, talk to them, several things about chemistry, etc. And I've been very fortunate, again, to get all that exposure, thanks to Mano in the nucleoside chemistry. And the kind of IUPAC, I was in another thing, IUPAC was another great experience. Although, many people who go to IUPAC, we know IUPAC for the nomenclature, etc., but there are a lot of other things to learn in IUPAC, how chemistry is done in the third world, Africa, against even more odds than what we do in India. And somehow I got a chance to be even the president of, one of the division, things, met lots of people. And I will not say much except to share one more experience that is about my journal leadership, ACS Omega. It just happened somewhere in 2015 or 16, ACS was about to launch a new journal. At that time, this open access was just a buzzword. It was coming up. And 2016, they constituted a committee to select the editor of the journal. And somehow, I don't know what was it, I was put as one of the selection committee members. There were no Zoom, nothing. There was only international telephone calls were there. We would fix up and talk. Kirk was the one who was leading. So they had, I think somewhere around October, November of 2015, we had several discussions, and I was really exchanging my views about, journal, publications, how difficult, etc. And in the 2016 January, usually all the ACS has about 50 journals. The chief editors of these journals, only the editors-in-chief, they meet in one of the places to discuss about a lot of policy issues. So after that, they called me, you must come and give a plenary talk for the ACS chief editors in one of the places in San Diego. I don't know what courage I had. I accepted it. That is the first talk, plenary, even before I meet people. The only editors-in-chiefs, who are notorious for rejecting our papers, they're all there. And I had to address them. So I had spoken to several of my Indian colleagues, what are the problems of having publications in ACS? Everybody is saying, RSA journals, YLA, they give a lot of opportunity. ACS, somehow when you submit, the moment they say .ac.in, software itself rejects your paper. That was the thing, they didn't even go for reviewing. So I went and presented statistics. Today, I cannot give such a talk what I gave on that day in an ACS forum. I was just simply, against my own personality, was brutal, telling, ACS, that this is it, and all that. So I thought after that nobody would speak to me. But during the I was the only person, a non-editor, in the whole thing. They all came and told me, and they said that, we liked your thing, share your slides, we will look into that. But then they told me one thing which appealed to me. Indians, you all do good work, very good science, but you don't know how to write papers, how to give titles. Editors would look for abstract, what is new, scientific, titles, and if the editors themselves don't convince, it would not. And they mentioned they knew a lot of people, in the Indian scenario. That struck a great chord with me. I think something we have to educate is, we all do good science, but you have to, know how to present it. And I'm sure many of our colleagues, we know, we've all learned it hard way. Writing papers, writing proposals are not part of our PhD training. And so after that, when I came back to India, then, I thought that something we should do it, I have some. Well, it so happened that January it happened. In February, they called me and said, would you like to be one of the chief editors of this journal? So that was a great surprise. I thought they would just banish me, but they said that. I said, let me see, and then I accepted. So if that is so, now you cannot be in the selection committee member. You cannot be a member of the selection committee. So in March, they launched this new journal. There was a great experience. You go to San Diego Convention Center, ACS National Meeting. ACS does everything in style, launching this new journal. So many TV displays, everywhere our photo is, coming up, editor-in-chief. That gave me a lot more publicity, people would give a second look, okay, is this the same person who is, displayed? And that was, starting an institution is one thing, starting a new journal. How do you attract readers to the journal? That to an open access journal where you have to pay and publish. So I fought very hard. Indians cannot pay so much of money, they decrease the funding, etc. But even then, it's always, difficult. But anyway, that took a different philosophy. But it so happened that, we were about 4 editors. After 1 year, we were only 2 editors, myself and someone from China. Now we have 18 associate editors. And I've learned quite a lot about publishing. When you read a journal, what is that editor would look for? What does the reviewer look for? Looking at the publication from the other side. And many times when I go, I share this thing with my colleagues, etc. Writing papers. Doing good science is one, but writing papers is entirely a different ballgame. And luckily, although we don't worry about impact factors, the journal made very steady fast. And last year, the journal published about 3,500 manuscripts, including the paying, etc. But the impact factor went up from 2.5 in the third year, impact factor starts to 3.2 and 4.2. And the study, even ACS had not seen this kind of steady impact factor, rise in any of the journals. And that gave me a new strength, because we spent a lot of time in, manuscript, because you get huge number of manuscripts. And we had to do that. All of us want to publish, but there must be somebody to review it, edit it, look it from the other thing. Otherwise, we won't be able to publish it. So that's given me a new understanding of, science in a way different than what you realize as an author. So I think I've been, again, very lucky. And I got a second term there also of the first 5 years. So now I think, although if I don't have a job, I won't be unemployed, I will still be an ACS, editor-in-chief. So, and I think I would like to finish, I may not have answered Hota's question still, and, but I must say that the CSIR contribution to ICER system is a fantastic one. I'm not sure whether CSIR itself would realize, and there could be a good marketing point. CSIR is well known for their human resource development program, fellowships, etc., etc. One of the most successful things, I think many of us have come, got the CSIR fellowships, but no other country has these kind of fellowships from the government. When some visitor comes, you tell them, this started when, long time back, the government giving you fellowship. Most of the fellowships are given by the grant, the PI has to give, from there. One of the most wonderful things. Even today, if you take, well, here we started in Pune with NCL. I was, coming from the CSR background. And of course, Dr. Shivram played a very, very important role in, that one. And then Saurabh became director of ICER Calcutta. And we had Shashidhara, who was, here. Then now he's the director of National Center for Biological Sciences, came from CCMB. Gauri Shankar, another director of Mohali, he has come from, again, from CCMB and then CDFD, etc. And Amitav Das, who was the Bhavnagar thing, is doing very well. And I hope he'll become still, I think, 2 or 3 ICES, they're looking for directors. He would, have a good chance. But I think the crowning gem of all this is Dr. Nathu. He's the chairperson of 2 of the ICER board of governors. Amazing. That's really, he started in ICER Pune. And these 2 ICERs are some of the difficult ICERs. And when I've met their faculty, etc., they have great respect for the kind of mentorship, the kind of problems, institutional problems he has been able to solve. Dr. Nathu, in fact, he's, I think, the crowning glory of, the CSIR, contributing as director, as a chairman. And these positions before him, Professor Siennara was the chairman, board of governors of ISER Calcutta, then Dr. Mashelkar, and then it was the turn of Dr. Nathu. And he has done a fantastic thing, doing that. So I think at some point, and then there may be several faculty who are, from CSR labs moved. ISER Tirupati, I myself have Balraman, who came from the thing, and CSR has done a great contribution. I tend to believe that the more I see, the way in which leaders are trained within CSR system, all of us got trained. Management, trust it is put, awareness of rules. CSR purchase rules is one of the best ones, which thanks to Dr. Shivram, the very first board meeting we adopted it. Other ISERs had not done that. Always they were cribbing about why something cannot be done. And we had a, no thing, I think. So CSR has contributed very, very greatly, and, must thank CSR, for that. And of course, we have JK came from, to ISER Pune from CSR system. Hota came from that. So at the faculty level also, CSR has, contributed quite a lot. Maybe somebody should make this thing about CSR contribution to the ISERs. Now that ISERs have become a kind of at least, recognized institutions, has to be done. So, I do not want to say much more. I think all of us are waiting for, dinner. But I'm really thankful. I'm really so overwhelmed by looking at, all the people with whom I've been closely associated, coming in person. And very moved that Professor Wadia could come here and sit for the whole day, and Javed coming all the way from there. And several of my NCR colleagues with whom I have worked. And I probably, except for some new students, I can probably recognize, all the people in the audience. So thank you so much. And I hope, as I said, connection with NCL or ISER Pune it cannot be severe. And the only thing is, that after 70 years of directorship, I feel very, I don't have to worry about RTIs. That's the main, or anything else, so I can freely. And ISER Tirupati is very happy to get, Santanu, continuing at ISER Pune. But unlike ISER Pune, where I had 2 terms and could finish actually it requires 2 terms for a director to finish the whole campus. And it didn't happen in Trivandrum, it didn't happen in Calcutta, they took longer time. But in Pune, Bhopal, and Mohali, the same directors continued, it got. But I'm sure in Tirupati it would not happen. And maybe by end of this year itself, Tirupati would be complete with Santanu's great initiative and new look, etc. So by responsibility, I know it's my own feeling that I have told Shantanu that only when inauguration is done, then only I think I will inside feel that is a sense of completion of this thing at ISER Tirupati. And he's already started, interacting with faculty, etc. Similarly, I'm so happy that Dr. Sunil Bhagwat has joined here, and we all met in Trivandrum, and we also had a short, discussion together. I think it's very, very important to have good people, good meaning people to be the directors, and one who would understand education, grading policies, etc., etc. So to answer your finally, is that I got an F grade in ISER Pune. An F grade in NCL also, that's why I went to ISER
but
Pune.
I did not ask you that
So I've been
question.
repeating exams, going.